Hi guys, am I audible, visible? Hi everyone, am I audible, visible? I guess I am. Hi students. Hi. Hi everyone, good afternoon, good evening rather, good evening everyone, welcome to the session. So can you see me, hear me? I think you can see me, but tell me, can you hear me? Yes everyone, can you hear me? Okay, Tutu Ambika. Hi, good evening guys. So today it's 30th of April or 29th of April? 29th of April, then 30th, then 1st, 2nd, 3rd, 4th and finally the most awaited day 5th May will be there, and you are going to appear, right? You are going to appear in your NEET examination then. How are you all feeling guys? How are you all feeling? How are you all feeling? Yes? We all are together always, right? The team of Unacademy NEET English is always together, don't worry. Tensed? Yes, I can understand that emotion. I know you people have multiple thoughts right now, the anxiety, tension, excitement, right? You can't even wait to write your paper. You guys can't wait to, give it and to be the doctor finally, isn't it? Isn't it? I can understand your emotions, and that is why guys we are here to help you all, right? And today, right, you know that it is the 1000 question practice session, and today we are going to practice the questions in that way, in a way that you should follow for your NEET examination as well, right students? So guys, be ready for the session. I'm about to start it. So here in the description box itself you guys can see free AIMT link. So people, just keep it in your mind. Earlier we were thinking that we will just keep your test till 28th of April, but yeah, some students they demanded that they even want to practice the questions before the NEET examination. So now till 4th of May you guys can give these tests from 2 p.m. to 5:20 p.m. So that will be very good for maintaining that, for setting your body tone for your NEET examination. You know that on Sunday, that is on 5th of May, you have to sit from 2 p.m. to 5:20 p.m. in the examination hall. So you should train your body, right? You should train your body in that way only. So if you will give the test, if you will give the full syllabus mock test, obviously you will analyze your mistakes. You can just improve your mistakes. Plus you will learn that yes, you have to be, your brain will learn that it has to be active from 2 p.m. to 5:20 p.m. and it has to solve the question. So guys, right, so here you guys can see AIMT link. Okay, done bache. So you can just enroll yourself there. Okay, you can click on it and you can go to that. You can enroll here in this AIMT and you have to give the test. Okay, you have to give the test. Okay, so now this is your own channel, Unacademy NEET English. Yeah, what are you people doing? Let's make it 200K as soon as possible. Okay, so you can see the multiple videos here, see the GOC one shot ka video, right? You have your guess strategies also, and here you guys can see, right bache, that how to fill the OMR sheet. So in a very realistic way I told you that the kind of mistakes you can make while filling the OMR sheet and the things that you should avoid. Okay, so this is your channel, your own channel, so make sure you subscribe it. Okay, so let's start the session guys. Are you ready? Are you ready? So can I ask you some random questions? Can I ask you some random questions? Yeah, from my side voice is fine. I don't know what's the issue. I have changed my mic as well. Can I ask you some random questions? Okay, so which organism is going to make the red tide? Which organism is going to make the red tide? So without even the MCQs, without even the options, can you just answer that to me? Can you just answer that to me? Why are Goniolix? Very good, excellent, correct answer. So can I ask one more question? In which phenomena the genotypic and the phenotypic ratio is same, that is 1:2:1? In which of the genetic inheritance the phenotypic and the genotypic ratio is same, that is 1:2:1? That is 1:2:1. The ratio is 1:2:1. The phenotypic and the genotypic ratio is same. Guys, that is codominance and incomplete dominance. That is codominance and incomplete dominance. Okay, one more question. In the gel electrophoresis, the separation is on the basis of size or on the basis of charge? Yes, the separation is on the basis of size or on the basis of charge. What should be the correct answer? What should be the correct answer? Size. The separation is on the basis of size. So can I ask you one more question? Yes, some random questions. Okay, so in which cytoskeletal element the 9+2 arrangement of microtubule is there? The 9+2 arrangement of microtubule is there? Yes, guys. Tell me quickly. Tell me quickly. Cilia and flagella. Excellent. So guys, you guys can see the this thing here till 30th of April, means till tomorrow you guys can avail our subscription just at ₹54,999. So you know what you have to do. You have to apply this coupon code, that is ambika10, right bache? So I will start your batch right after the NEET examination. Other teachers, they have started their batches, but I will start the biology right after the NEET examination. That is from 6th of May. You will start getting my classes. Okay, because I know some students are texting me on Telegram that ma'am, when are you going to schedule your classes? I will schedule my classes from 6th of May. So this is the coupon code that you people can use, and you will get our batch just at price 5499, right bache? Right, and you know that your team is there, your amazing team is there, question practice and everything is there, right bache? Right. So now let's go, let's start, and I hope that you all will, yes, you all will give your AIMT test. Okay, so let's start the class. So are you ready? Sure? Are you ready? Ma'am, can I get 380 plus from these 100 questions? Yes, bache, yes. So let's get started. Okay, so answer one more question, that in the ETC, which electron carriers are the mobile electron carriers? Answer one more question of mine. Which electron carriers are the mobile electron carriers? Answer one more question of mine. Yes, quickly answer one more question of mine. And here you guys can see the names for the different batches. So now I'll be starting the questions. Quick, answer my question, then I'll start the questions. Quick. Cytochrome C and ubiquinone. Excellent. So here we have our first question, guys. So you people can see the questions here, and please see, understand one thing. You are going to practice 1,000. Wow, Ambika, wow. You are going to practice 1,000 questions. Okay, so let's say out of these 1,000 questions, if 400 questions are from biology, that questions are also the most expected questions. Okay, that questions are the most expected questions. Here you have your PYQs as well. So obviously, don't you think that out of 400 questions, these questions are from your complete syllabus, that out of 400 questions, at least, don't you think minimum 80 questions, they can't come in your examination? Don't you think so? You know the pattern of NEET examination. You know that PYQs are repeated. So don't you think that out of 400 questions, you guys can get minimum 80 questions? Don't you think so? Right? It's a fair calculation, isn't it? So Please be sincere when you answer the question. Read the question carefully, and I want each and everyone to respond, guys. And let's make the live learner number more than 1,000. Right? Hit the like button as well. And guys, let's make the number more than 1,000. So here you have your first question. It is from photo period affect reproduction in. Right, bache? So this is a statement from your syllabus itself. It is not from the deleted part. Right? It is a statement from other chapter. Photo period, light period, it affects the reproduction in. Yes, bache, it affects the reproduction in. Your light period, right, the photo period is going to affect the reproduction in. Yes, yes, yes. Light period affects the light period affects the reproduction in. Some students are saying 3, some are saying 1. Let me tell you, answer is 1. They are seasonal breeders. They are the seasonal breeders, right? Seasonal breeders, something related to their circadian rhythms, and definitely light is going to affect that. Isn't it? Light is going to affect that. So here your correct answer is seasonal breeders. Now let's move to the next question. Right, let's move to the next question. That dash is used extensively in biochemical and genetic work. Dash is used extensively in biochemical and genetic work. I want to see the students who are giving the correct answer. Each one of you, please participate in the discussion, and let's make the live learners more than 1,000, everyone. Quick, share the session link with your friends as well. Quick. Yes, guys, what should be the correct answer? What should be the correct answer? Dash is used extensively in biochemical and genetic work. Of course, the correct answer here is neurospora. What is the correct answer here? It is neurospora. It's a fungus, and you know that we also call it as Drosophila of plant kingdom. What do we call it? We call it as Drosophila of plant kingdom, right? So this is what you need to keep in your mind. And if you talk about the Aspergillus, so this Aspergillus, it is also known as the weed of the lab, right? It is also known as the weed of the lab. That's what you have to keep in your mind, right, students? So let's move to the next question. So which of the following plant group play an important role in plant succession on bare rock? Okay, so succession topic is also, you know that succession topic is also not included in our syllabus, but still, right, it's a repeated question. That's why I'm telling you. So you know that if I talk about the options here, so bryophyte, because mosses are included. Okay, because mosses are included. Now let's move to the next part. In majority of gymnosperms, pollination occurs with the help of, yes, pollination occurs with the help of. Yes, guys. Yes. So in majority of gymnosperms, pollination occurs with the help of, the word is gymnosperm. The word is gymnosperm. So here, obviously in gymnosperms, it will be with the help of air. So can I call it anemophily? Yes, students, can I call it anemophily? Everyone in the class, can I call it anemophily? Yes or no? Yes or no? If it is a yes, I want to see the fire emojis in the chat section. I'll be fast, don't worry guys. I will be fast. So I want to see the fire emojis in the chat section. So anemophily, that is air pollination. So you know that pollen grain needs to be lightweight. It should be non-sticky. That's what you need to keep in your mind, right? So let's move to the next question. So here you guys can see an image. So identify the given diagram and mark the correct option. This is the type of question that can come in your examination, of course. So identify the given diagram and mark the correct option. Identify the correct diagram and mark the correct option. Yes, people, quick. Everyone. Yes, everyone, quickly hit the like button and let me know. Let me know this thing. Identify the given diagram and mark the correct option. What is it? What is it? What is it? Is it? Yes. So you can see it is pinus. Am I right? It is pinus. And pinus is what? Pinus is monoecious. What is the meaning of being monoecious? You remember this trick? BMH. Bisexual monoecious hermaphrodite. Bisexual monoecious hermaphrodite. Bisexual monoecious hermaphrodite. Isn't it? Isn't it? Yes or no? So you know that, see, they are saying roots are associated with fungal hyphae. Correcto. Male gametes are motile and multiciliated, of course not. So roots are associated with fungal hyphae, and you know that here we are talking about the mycorrhiza. So when you practice one question, you learn other things also, right? So it is mycorrhiza. It is mycorrhiza. Right, bache? The symbiotic association in between the roots of the pinus and the fungal hyphae. Is that clear? Okay, now let's move to the next question. Given floral formula belongs to which family? So I hope you have seen my video where we have discussed the families in less than 15 minutes. Okay, so what should be the answer? There is a floral formula in front of you. Yes, from the families topic, that type of questions can come. That type of question can come. Anyone? So just look at this part, epicalix. You see it in the China rose as well. So it is the Malvaceae family. Which family is it? It is the Malvaceae family. Now let's move to the next question, guys. Storage of food is the main function of. Yes, we know that. Storage of food is the main function of. Guys, you know the correct answer. Yes, people, you know the correct answer. Out of these 100,000 questions, yes, you guys can expect 90 questions, right? Not just in biology, you guys can expect 180 questions in your paper as well. Why? Because they all are the repeated PYQs, right? It is all as per NCERT. So definitely you should attend this session till the end. We will provide you the PDF as well. So quickly answer my question. Storage of food is the main function of, right? And whatever questions are there from the biology, you people will see, right, bache, these questions are from the biology. You people will see at least 80 questions from this session only. At least 80 questions, minimum 80 questions I'm saying. Okay, so of course when it comes to the storage of food, right, when it comes to the storage of food, it is the parenchyma. So parenchyma mane living tissue, right, bache? It is the living tissue. Next question, which of the following is chief photosynthetic pigment? Again a very simple question. Which of the following is a chief photosynthetic pigment? Which of the following is a chief photosynthetic pigment? Yes, very good. So it is going to be chlorophyll A, which is also known as universal photosynthetic pigment, right, bache? Universal photosynthetic pigment, and I hope you remember it, right? So where is it present? In the photosystems, in the reaction center. So in the reaction center, this is the pigment that you people are going to get. So keep it in your mind, guys, keep it in your mind. It is chlorophyll A, right? So the next question, tell me, for the biosynthesis of chlorophyll, which ion do you need? Answer this question of mine, then we will move to the next question. For biosynthesis of chlorophyll, which ion do you need? For biosynthesis, the word is Moni bache, the word is biosynthesis. The word is biosynthesis. Excellent. Majority of students are giving the incorrect answer. That is why I asked you this question. When it comes to the central metal ion, that is your magnesium, students. Right, magnesium is the central metal ion, but when it comes to the biosynthesis of chlorophyll, it is your ion which is required, right? It is Fe which is required. I know this chapter is deleted, but still the statement is there in the chapter photosynthesis as well, and you should not miss it, students. Okay, you should not miss it. Clear, bache? You should not miss it. Are you getting my point? Yes. Are you getting my point? Yes, bache. Okay. So how many ATPs are required in regeneration step of Calvin cycle? Excellent. In the regeneration step, they have specified it in the regeneration step, right? You people are not pranked. You are not listening to me carefully. The word is regeneration step, guys. In the regeneration step, what should be the answer? Just for your quick revision, you are talking about the Calvin cycle, C3 cycle. So the first step will be carboxylation. Is it carboxylation? Then is it reduction? Yes or no? You tell me. I hope you remember that. Then comes the regeneration. Then what is the next? step regeneration. So in the, when it is the one turn of C3 cycle, one turn of carbon dioxide fixation, so obviously in the reduction you know that 2 ATP and 2 NADPH will be used, but when it is the regeneration, right, only 1 ATP will be used. That is why 3 is the correct answer, right? So now if I will ask you that for the formation of glucose, how many ATPs will be used in regeneration step, then what should be your answer? Yes students, then what should be your answer? If I am saying that for the formation of one glucose molecule, how many ATP will be used in regeneration step? Again, you are not listening to the question carefully. How many ATPs will be used in regeneration step? And that is 6 ATP, because 1 ATP is used in regeneration step. For the formation of one glucose, you have to take 6 turns of this cycle. So it means 6 ATP in regeneration step. I have specified the step here, guys. Okay. Now let's move to the next question. In Krebs cycle, during conversion of alpha ketogluteric acid to succinyl coenzyme A, which one is taking place? Definitely this question will come in your NEET 2024. In one form or another form, it will be there in your NEET 2024. It will be there in your NEET 2024. Yes students, I am pretty sure you know the answer. We have discussed the plant physiology in detail in our marathon session. So I believe you know the answer. Excellent. So it will be the oxidative decarboxylation. How is it possible? So it is possible because we have a trick, right? So the trick is saying ki come in and kick scooter, come in and kick scooter, fastly move on, right? Come in and kick scooter, fastly move on. So C stands for citrate, right? I stands for isocitrate. I hope you remember. And kick means alpha ketoglutarate. Please revise it with me, right? Scooty means succinyl. Okay, so succinyl. So firstly there will be the succinate, right bache? So succinate to succinyl coenzyme A. Am I right or wrong? Succinate to succinyl coenzyme A. Then fumarate, then malate, and then finally there will be the oxaloacetate. So it is the oxaloacetate which will accept your acetyl coenzyme A, which is a 2 carbon containing compound. So I hope you remember oxaloacetate 4 carbon, citrate 6 carbon, isocitrate 6 carbon, alpha ketoglutarate 5 carbon, succinate 5 carbon, succinyl coenzyme A 5 carbon, right? Then fumarate 4, sorry, this one is 4 carbon. This succinyl coenzyme A is also 4 carbon, and then rest all are 4 carbon containing compound. Do you remember that? Right? All are 4 carbon containing compound. So alpha ketoglutarate, which is a 5 carbon containing compound, it is making 4 carbon containing compound. So obviously what is happening here? Decarboxylation is taking place. And here also from isocitrate to alpha ketoglutarate also decarboxylation is taking place. Again, I am specifying this thing because this question can come in your NEET final examination. This step is of substrate level phosphorylation, right? This step is of, yes bache Sharat, this step is of right, this step is of, yes bache, substrate level phosphorylation. And here in this step there is one mistake. There is one mistake in this step. If you will tell me that mistake, you guys are going to get a chocolate. You people are going to get a chocolate. If you know the mistake, you guys will get a chocolate. There is a mistake in this step. Very good, curious facts, excellent. Obviously here, firstly, this is what I was checking. Firstly, there will be the succinate, right? Sorry, there will be the succinyl coenzyme A, and from that succinyl coenzyme A, you will get succinate. So it is the most commonly, right, most common error here in this step. So succinyl coenzyme A will form the succinate. Got it? Succinyl coenzyme A will form the succinate. So yes, you deserve one chocolate. Okay, you deserve one chocolate. So it is the step in the Krebs cycle. It is the only step in the Krebs cycle where what is going to happen? The substrate level phosphorylation will be there. Firstly, GTP will form. Immediately it will form GDP. Then again that GDP will immediately form ATP. That's what you have to remember. Okay. Okay. So happy. Yes, that kind of question can come in the NEET examination. So yes, students, are you happy? Happy or not? Arey yaar, tell me, happy or not? Happy or not? No, ma'am, we are very happy. We are very happy. You know? So, is it possible to store pollen grains of a large number of species for years in liquid nitrogen? Yes, in liquid nitrogen. At minus 196 degrees Celsius. So what is it? It is cryopreservation. So it is cryopreservation and it is the XC2 mode of conservation, right? XC2. It is a type of XC2 conservation. Okay, bache, it is a type of XC2 conservation. Clear, clear, clear. Now let's move to the next part. So recognize X and Y in the following diagram. X and Y. Hanji, X and Y. You know the answer, students. You definitely know the answer. I know you know the answer. Quick. Yes, bache. Quickly tell me. Look at the diagram very carefully. Don't get confused. Don't get confused. Look at the diagram carefully. Exactly. X is male gametes and Y is egg nucleus. Even if you are confused here, you need to focus on one thing, and that is, right, that X is the male gamete. So you can cancel these two options. Now when you look at this, definitely your Y cannot be the vegetative nucleus here, so it can be egg nucleus only. Now dash promotes rapid internode elongation in deep water rice plant. There is the elongation of the internode. Yes, students, again NCERT it is. Right, so you guys can get confused here. We have practiced it already. Dash promotes the rapid internode elongation in the deep water rice plants, and that is known as ethylene. That is known as ethylene. That is known as ethylene. Clear, bache? That is known as ethylene. Clear, bache? That is known as ethylene. Okay. Okay. Okay, so next question. Dash usually results in the growth of lateral buds, especially in the tea plantation. Growth of lateral buds. Growth of lateral buds. Again a very simple question. Growth of lateral buds. It can be done due to decapitation, that is removal of shoot tip. Why? Because that shoot right here, what is it doing? It is showing the apical dominance, right? Apical dominance. So if we will remove that apical bud, then obviously lateral growth will be there. Lateral growth will be there. That's what you have to keep in your mind, students. That is what you have to keep in your mind, students. Is that clear? Is that clear? So that is the correct answer. Clear, bache? That is the correct answer. Got it? Yes, students, got it? So that's what you have to keep in your mind. Okay, and even I know you must be thinking, why can't, right? Why can't we just apply the cytokinin? Some students must be thinking, why can't? Exactly. No, Kaushi, spraying oxygen will not be a good idea because it promotes the apical dominance. So when there is the apical dominance, lateral buds will not grow. But when it comes to the when it comes to the tea plantation, so this is done, right? The apical buds, they are removed. So here they have specified tea plantation is the thing. So we have to remove the apical bud here. That's what you have to keep in your mind. Now move to the next question. Again a statement based question it is. Statement A is telling us lateral meristems are responsible for increase in the girth in maize stem. Apical and intercalary meristem are responsible for increase in the length of the plant organs. So tell me the correct answer. Yes, bache, tell me the correct answer. Please tell me the correct answer. Yes बच्चे, lateral meristems, right? Lateral meristems, which are also known as cylindrical meristem, right? It is the cylindrical meristem. Hope you know that. They are responsible for increase in girth. Girth mane diameter, right? Girth mane diameter, you know that. So it is in the maize stem. Maize is what? Maize is a monocot. So in the monocot, you know that the lateral growth is not there. So this statement is incorrect, students. So statement A is incorrect. But yes, apical and intercalary meristem, it is responsible for the increase in length. Yes. So apical, which is present at the tips, intercalary, which is present in between the permanent tissue. So definitely it is the B, which is the correct answer. A, which is the correct answer. Next, nematophores are respiratory roots. They help to get oxygen for respiration. They help to get oxygen for respiration. Yes bache, in the case of statement based questions, one student is saying how to tackle the statement based question. In case of statement based question, read it carefully, that's all. In statement based question, it is easy. Assertion and reason can be difficult, but statement based questions are very easy. They are very easy, students. Okay, either one statement is correct or not, that's all. Done. So nematophores are the respiratory roots. They help to get oxygen for the respiration. Is it so? Is it so? Obviously nematophores, you know na, respiratory roots, we see them in the case of mangrove plants, right? Like your rhizophora, like soneraceae. So they are negatively geotropic. They are having these lenticels. Okay, what do they have? They are having these lenticels. We also call them as pneumatodes. We also call them as pneumatodes. So obviously exchange of gases will be there from these lenticels, right? In halophiles you see basically such type of roots. So why are they respiratory roots? Because they help to get oxygen for respiration. That's why the reason is justifying the assertion, and 1 is the correct answer. Any doubt, students? Any doubt? So from these questions, right, let's say if today we are going to practice 300 questions, from that 300 questions of biology, you will get at least 80 such type of question in your paper. You can mark my words bache, right? In one way or another way, these questions will be there in your paper, and we are going to have that predictor, NEET predictor paper session as well. Okay, so please be careful, students, just focus on it. Now let's move to the next question, which is again match the column kind of question. So I believe you know the answer here also, and these are the easiest kind of questions. These are the easiest kind of question. Yes bache, I am talking about both botany as well as zoology. Yes bache. See, sequoia, is it the tallest tree? Yes ma'am. Cycas is having the coralloid root. C for cycas, C for coralloid root. So here cyanobacteria is in association, right? Cyanobacteria, they will live in that roots. Okay, Pinus mycorrhiza and mango, obviously then you can relate it with the embryo sac, isn't it? So 1 with the C, you got your answer, that is 1. Right, that is 1. So now let's move to the next question. Again match the type of question, and bache, it is the most important question, right? It is from your algae part. It is the most important question. It is the most important question. Please answer it very carefully. Yes, please answer it very carefully. Match the following column and choose the correct match. So algin, carrageen, agar, parasitic algae. See, C with 2, right? C with 2. So I got my answer, right? I got my answer. So algin is obtained from brown algae. This carrageen is again from the red algae, right bache? This parasitic algae is Cephalourus, and it affects the tea plantation, right? As per my knowledge, it affects the tea plantation. I need to check it again, right? But still, right, as per my knowledge, it affects the tree plantation. So 3 is the correct answer. Let's move to the next one. Select the correct function of ABA, guys. Right, ABA, ABA, ABA. What is ABA? Yes, what is ABA? Quick people, what is ABA? ABA, abscisic acid. Abscisic acid. So abscisic acid is also known as the stress hormone, right? But it doesn't mean that this hormone is going to give stress to the plant. No, No, it's not like that. Actually, it is the hormone which protects the plant from the stress, bache, right? Whenever there is a stressful situation, this hormone will be there in the plant, and it will inhibit its the seed germination because conditions are not favorable. It will close the stomata because water scarcity is there, right? It inhibits, right? It increases the seed dormancy because obviously the conditions are not favorable for the seed growth. So A, B and D, they are the correct statements, right? So assertion: the function of sieve tube elements are regulated by the nucleus of companion cells. Sieve tube elements are enucleated at maturity, right, and they are associated with the companion cell. So what should be the correct answer here? What should be the correct answer here? Again, it's very simple. Yes, people, the function of sieve tube elements are regulated by nucleus of companion cells. So sieve tube elements are enucleated at maturity with, it's right, the statement is correct, that sieve tube elements, they are compared with the human RBC because at maturity they are enucleated. That's why, right, they are enucleated, and these elements, they are associated with the companion cell. So companion cell, the nucleus of companion cell is going to regulate the function of sieve tube elements. Is that clear? So sieve tube elements are enucleated at maturity with peripheral cytoplasm and associated with the companion cell. So assertion and reason both are true, and R is the correct explanation of A. R is the correct explanation of A. Now let's move to the next question. Match the column 1, 2 and 3 and choose the correct combination from the given option. You have to match column 1, column 2 and column 3. Yes, students, you have to match column 1, column 2 and column 3. Column 1, column 2, column 3. Primary producer, primary consumer, secondary consumer, tertiary consumer. So when it is the primary producer, without any doubt, it is the first trophic level. So A will go with 1. So you can eliminate this option. Okay, bache? And grass will be the thing. So A1N. So you can eliminate this option as well. Isn't it? You can eliminate this option as well. So it is the right way to attempt the paper. If you don't know the answer, right, you can eliminate the options as well. Okay, so next is primary consumer. Of course, out of the following, it will be at trophic level 2, 110%, right, bache? So it is going to be your grasshopper. So third is the correct answer. Third is the correct answer, right, bache? Right? So you got it right. So which is not true regarding ecosystem? Which one is not true regarding ecosystem? Which one is not true regarding ecosystem? Yes, regarding ecosystem, which one is not true? I believe you know the answer. Yes, I believe you know the answer. Which one is not true regarding ecosystem? The word is ecosystem here. Is there any cyclic exchange of material between living beings and environment? True. Self-sustainable? True. Input of energy is required? True. Cycling of energy occurs? No. You know that when energy will pass from one trophic level to another, only 10% of energy will pass, right? The energy will also be dissipated. So here, first is the correct answer, right, students? Now, identify the correct statement from the following. Yes, bache, you have to identify the correct statement from the following. Quick. You have to identify the correct statement from the following. Quick. Go for it. Which one is the correct statement here? Which one is the correct statement here? Yes. Which one is the correct statement here? Measurement of biomass in terms of fresh water is fresh weight is more accurate. Is it so? Is it so? Don't you think we consider the dry weight because there will be no seasonal fluctuation? So this is not our answer. Primary productivity depends upon the animal species. No. It depends upon that particular plant species. GPP is the amount of biomass available for consumption to carnivore. No, that will be NPP. Mass of living material in a trophic level at a particular time is called standing crop. Yes, bache, it is standing crop. BC is the thing here. So C for crop, B for biomass. right? Biomass. So if it will be the standing state, you will consider the inorganic things. Okay, okay, so 1 is the correct answer because it is the incorrect statement. Next, yes bache, next again assertion and reason based question. Answer it. Answer here. What should be the correct answer here? You know the answer, right? Mycorrhiza are considered mutualistic relationship between fungus and roots of higher plant. True. This interaction confers benefit on both interacting species, where fungus help the plant in absorption of essential nutrient while plant provides food to the fungus. See, if you will see, both the statements are correct, even the assertion is correct, even the reason is correct. Am I right? If you will see, you know that both the statements are correct. Even the assertion is right, even the reason is right. Yes or no? Yes or no? Yes or no? Definitely. But if you will see, is reason is the explanation of assertion? It's simple. They are saying mycorrhiza is considered as mutualistic relationship because one is providing the benefit to another. In the reason statement, that is what they have explained, that how are they providing the benefit to each other. So definitely 1 is the correct answer. Assertion and reason, both are true, and reason is the correct explanation of assertion. Next question, students. Right, which one of the following is not an example of hotspot in India? The word is not an example. Okay, again a very simple question. It is not an example. Not an example. Of course, Eastern Ghats are not the biodiversity hotspots. Next, how many varieties of mango has been estimated to be present in India? How many varieties of mango? Yes, it's mango, not the rice varieties. Mango, mango, mango. Mango. Definitely, 1000 will be the correct answer. Next, recessive trait is seen due to non-functional enzyme or because no enzyme is produced. A true breeding line is one that, having undergone continuous self-pollination, shows the stable trait inheritance. Now, what should be the answer? What should be the, right, what should be the answer here? You know the answer, students? You know the answer, students? Yes, even you know the answer. Recessive trait is seen due to non-functional enzyme or because of no enzyme produced. True. A true breeding line is one that, having undergone continuous self-pollination, shows the stable inheritance. True. So both the statements are correct, students. Both the statements are correct, students, right? Next question. A heterozygous round-seeded pea plant is crossed with wrinkled seeded pea plant. How many plant produces intermediate size starch grain out of 2000 progenies obtained? Right? Yes, these are the questions from genetics. Of course, these questions are from genetics. A heterozygous round seeded pea plant is crossed with wrinkle seeded. In the statement itself, they have given you the answer. A heterozygous round seeded plant, you are crossing it with this. You know that when this is the case, okay, you know that when it is the case, it is a test cross. It is a test cross. One is heterozygous dominant, one is homozygous recessive. The ratio is 1:1. One will be, means 50% will be dominant here, 50% will be recessive here. So it's not mandatory that you have to calculate. Simply they have asked how many plant they produces mediate size starch grain out of 1000. The word is intermediate now, right? The word is intermediate. One is round, right, heterozygous round, one is wrinkled. So if you will take the ratio also, what should be there? What should be there? Even if I will take it as 1:1, but is it going to show this? Is it going to show 1:1 relationship? Round seeded, but see bache, I'll tell you. Dekho, you remember pleiotropy, right? One condition is right. Do you remember pleiotropy? Pleiotropy, multiple phenotypic effects of a gene. Multiple phenotypic effects of a gene. How many of you know that? It's a very good question. Multiple phenotypic effects of a gene. Do you remember this or not? Anyone in the class? I know you know the answer, but I want an explanation here, right? Such type of questions can come in your paper, so I want an explanation here. So do you remember it or not? Tell me, do you remember pleiotropy? Do you remember pleiotropy? Multiple phenotypic effects of a gene. So you know that here, firstly they are talking about round seed and the wrinkled seed. So you know that, right? The seed shape and size of starch grain. The seed shape and size of starch grain. It shows what? It shows the pleiotropy. Right, it shows the pleiotropy. Even we have discussed it, right? Even we have discussed it. So if you remember, if it is the case, right? If it is the case, like same gene is going to control the two different traits. So heterozygous is the case. So when the seeds, right? When the seeds are having round seed shape and when they are heterozygous, so at that point of time, you will see the starch grain, they are medium. The starch grain, they are what? They are medium. What are they? They are medium. Right, what are they? They are having the medium size, intermediate size. Now how can you relate it? Again you need heterozygous. So when this, okay, sorry, again you need heterozygous. Let's say you are denoting it with RR only. Okay, right? So when you are taking this cross, look at the Punnett square. Please look at the Punnett square. So this is the thing, 50% is having the round seed shape, 50% are having wrinkled. So the one which are having the round seed shape, they will also have the, because they are heterozygous, so they also have the intermediate size. They also have the intermediate size of the starch grain. If this will be the case here, then the size of starch grain will be large. Then the size of starch grain will be large. So understand the reason here. Okay, understand the reason here. That is more important. Fine. Next, so answer is 1000 only. Answer is 1000 only. But now I told you why. So husband and wife, they have normal vision, but fathers of them were colorblind. Husband and wife, they both are having the normal vision, but their father were colorblind. So probability of their first daughter to be colorblind is, probability of their first daughter to be colorblind is, husband and wife, they are having the normal vision, but fathers of them were colorblind. Imagine that's the case. Okay, okay, sorry. Let's say colorblind father, right? So colorblind father can pass the trait to daughter. Yes, that's the possibility. Now here in the question itself, they have specified कि husband and wife, they are having the normal vision. So let's say even if wife is a carrier, husband, and even husband's father was also the colorblind, but husband's father cannot pass this defected allele to the son because they are going to pass the Y chromosome to the son. So for sure father is normal, 110% father is normal, and if mother is normal and even if she is the carrier, still there is no chance that daughter can be the color blind. Okay, right? So this will be the answer. This will be the answer because it is a recessive, sex-linked recessive disorder. Next, through dash and domestication from ancestral wild cows. No, this is not that we need. Okay, give it a try, give it a try, give it a try. Through dash and domestication, this is correct. You have to consider this question. From ancestor wild cow, we have well-known Indian breeds, Sahibal cow in. Yes, Sahibal cow in. It is not from strategies part, it is from NCERT, and you know from where it is. Through dash and domestication. So obviously through artificial selection and domestication from ancestral wild cows, we have well-known Indian breed Sahibal in Punjab. So second is the correct answer. Next, polygenic inheritance is influenced by. Polygenic inheritance is influenced by. Polygenic inheritance is influenced by. Of course, it is influenced by environment. It is influenced by environment, right? Polygenic inheritance means many genes Many genes are controlling one character. So dominant alleles, they are going to contribute there. Okay. Okay. So now let's move to the next question. In Human Genome Project, the principle of automated DNA sequencers developed by, right? Automated DNA sequencers, they were developed by, again you know the answer. Yes, again you know the answer. Again you know the answer, people. What should be the correct answer? Of course it is one, that is Frederick Sanger. So tell me, how's the Josh, how's the energy? How's the Josh, how's the energy? How are you feeling? How are you feeling? How are you feeling? Arey, send some emos, yaar. I want to judge your performance. Some are saying not good. It's okay, fine. Just revise these questions again. Revise these questions again. If you're not feeling confident, if you feel like, yaar, I'm not able to answer the question, then it means you need to revise again. So don't worry about it. You can revise, okay? You can revise. Okay? Okay, so Frederick Sanger is the answer. Now let's move to the next question. Here you have the next question. So it has been asked in the NEET 2023 paper. Answer it. Yes, students, so what is the correct answer here? What is the correct answer here? So it is 80, right, bache? It is 80, right? So it has been asked in NEET 2023. So ribosome, they have structural RNAs and they have about 80 different proteins. Done? So next question, select the incorrect statement. Yes, students, select the incorrect statement. You have to select the incorrect statement. Go for it. Go for it. Incorrect statement. So yeast is used for the production of ethanol. Yes, ma'am. Enzyme lipase is used in detergent. Yes, ma'am. Lipase removes oil stains. Yes, ma'am. Bottle juices are made clearer by use of streptokinase. No. Why? Because here we can use pectinase and streptokinase is your clot buster. It is your clot buster. So incorrect mein third will be the answer. Now next is ladybird and dragonflies are used to get rid of, again NCERT, right? It is from microbes in human welfare. Quick students, it is from microbes in human welfare. What should be the answer here? Your ladybird, they'll remove aphids. Dragonfly, they'll remove mosquitoes. Okay, next, dinoflagellate and diatoms are dash in the oceans. What are they? Dinoflagellates and diatoms, students, what are they? Quick, what are they? Dinoflagellates and diatoms. Yes, so they are the planktons. So when you use the word planktons, means they float passively. है ना? They float passively, isn't it? Isn't it? Oh, one second. They float passively, but one second. So diatoms, they are the chief producers, right? They are the chief producers of the oceans. So mainly two will be the answer. But yeah, they also float passively, but they are the chief producers. So out of this, the most appropriate option is your option number 2. My bad. It is option number 2. No doubt they float passively. You are considering them as planktons, right? So planktons, they float passively, but yes, they are the chief producers. Okay, both of them. Next, read the following names carefully and mark the correct option. Okay, so they are asking how many plants, in how many plants phycobilins are present as photosynthetic pigment. The tricky one, yes, the tricky one. But I think my speed is good. Phycobilins, where are you going to get phycobilins? Please read it carefully. Phycobilins, where will you get phycobilins? Phycobilin is the word. So obviously it is photosynthetic, but where are you going to get it? I will ask the detailed explanation here. I am going to ask the detailed explanation here. So accordingly answer the question to me. Accordingly answer the question to me. Where are you going to get it? Very good. Correct answer. In the case of red algae, you will get this pigment, and in the case of Monera, blue-green algae, that is your photosynthetic bacteria, it also contains phycobilin. Phycobilin is also water soluble. So here out of the options given here, you know that your Porphyra is the red algae, your Polysiphonia is also the red algae. That is why our correct answer is going to be 2 here. Okay, it is going to be 2 here. So P2G2 is a trick for red algae. P2G2 is a trick for red algae. Next, how many tepals and stamens are respectively found in members of Liliaceae? The word is tepals. Tepals are what? When your petals and sepals, they are united. Tepals, right? It is from Liliaceae. It is from Liliaceae. Yes, students, it is from Liliaceae. You know the answer? Yes. You know the answer. If you don't know it, then revise it. If you know it, the tepal, how many tepals are and stamens? They are fused also, na? They are fused also, na? So guys, it will be 6. It will be 6. They are fused also. Remember, here tepals, group of tepal is going to form perianth. Stamens means A, so they show the adhesion. So it is 6 and 6. It is 6 and 6. Next. In monocot stem, vascular bundles are very easy. Very easy. Even it is discussed in detail. Right? Even it is discussed in detail. Even it is discussed in detail. So what should be the correct answer here? Yes, I know you know the answer, students. What should be the correct answer here? Of course, they will be scattered. First of all, they will be scattered because it is a monocot stem. And are they going to be open? No. Why? Because they don't show the secondary growth. So is it radial? No. So it is conjoined and closed, but they will be and closed. Why? Because they don't show the secondary growth. Okay? So one more thing I want to ask you, if this is the condition, if your perianth and endometrium, they are fused, what is the word that you are going to use for this type of condition? What is the word that you people are going to use for such type of condition? Yes, what is the word that you guys are going to use for such type of condition? Guys, yes, it is epitapylus. It is not patellus. Either it is epitapylus or epiphyllus is the answer. Epitapylus or epiphyllus is the answer. Next. In Angelman's experiment, maximum accumulation of aerobic bacteria over green algae Cladophyra took place in which of the color range of white light? A very simple question. You know, na, Angelman's experiment, it talks about the action spectrum. So where will be the accumulation of aerobic bacteria? Of course, in blue and red light, because we will see the maximum photosynthesis here. Oxygen will evolve, aerobic bacteria will aggregate there. Next, identify A and B. Identify A and B. Very good. I like the question. Identify A and B. I like the question. Quick. Identify A and B. Very good. See, you are making acetyl coenzyme A from the pyruvate. So it is your which reaction? Transition reaction, link reaction. So coenzyme A is added. What will be released? Right, CO2 will be released. Yes, CO2 will definitely be released. And what about NADH2? Will it form? Yes, bache, it will form. So according to me, your answer should be 4. And what about you? What about you? According to you, what should be the answer? According to you, what should be the answer? Yes, 4 itself. CO2 is released and NADH2 will form. Next, identify the correct match with reference to the given diagram. So you remember my yoga here. Do you remember this yoga wala thing here? So I think now you guys can answer it. Yes, students, you people can answer it. You people can answer it. Guys, what happened? It is a 1000 question practice series and I cannot see the energy here. I cannot see the energy here. Right, students, what's going on? What's going on? What's going on? Are you feeling low? Nahi na? Then be fast. Right, be fast. You have to practice more question. Right, and you know this is the right way to attempt your biology paper as well. This is the right way to attempt your biology paper. Right, start with the biology, finish the biology in somewhere in 35 to 40 minutes. If you can finish it in 35 minutes, very good. Then fill the OMR sheet. As I told you, right, bache, I made a video on that as well, that how to fill the OMR sheet. Okay, how to fill the OMR sheet effectively. Do Arey yaar. Okay, do watch it. Do watch this video, this one I'm talking about, how to fill the OMR sheet effectively. So you will get the tips there as well. So start with the biology, then go for chemistry, then physics. But make sure that you use less time in the biology so that you can save time for the physics, for the chemistry calculation as well, right? You need to avoid the mistakes in the calculation. You need to learn that you have to reduce the negative marking. Okay, you have to reduce the negative marking. Clear, bache? Clear? So what should be the answer here? Your A is basically the cotyledons. Okay, this is incorrect. So B is basically what? So it is down the cotyledon. C is the root cap. Okay, D is the radical. So according to you, what should be the correct answer? It's simple. Is your C root cap? Of course. Right, of course C is the root cap. Next, an experiment was performed to study the causative factor behind sudden elongation of internode just prior to flowering in rosette plant. So it is basically yes bache, it is basically gibberellin, right? So it is the bolting, right? The internode elongation will be there prior to the flowering. Okay, internode elongation will be there prior to the flowering. So 2 is the correct answer. Next, how many of the following tissue structures are not present in dorsiventral leaf? How many of the following tissue structures are not present in? Not present is the word, students. Not present is the word. You are talking about the structure of leaf. So you know that epidermis will be there, but the such hypodermis, endodermis, pericycle, you are not going to see that. 1 2 3 4 5. So second is the correct answer, right? Second is the correct answer here. Now next, assertion and reason. Endospermic seeds are usually present in members of Solanaceae family. Reason: endosperm is not consumed completely during embryo development in members of Solanaceae family. So what should be the correct answer here? What should be the correct answer here? Quick. What should be the correct answer here? Yes, what should be the correct answer here? So endospermic seeds are usually present in members of Solanaceae family, right. So because endosperm is not, देखो endospermic seeds means albuminous seeds, the one which are having the right endosperm. Okay, so it is not consumed completely during embryonic development, that is why it is present in the seed. It's a fact. So assertion and reason both are true, and R is not the correct explanation of A. So from the families such type of questions can also come. Okay, next question, which of the following statements regarding the energy flow they are false? So total 4 statements are given, right, and they are talking about the false statement. So when you read the question, right, highlight this word, it is the false, right? They are asking about the false or the correct statement, you should highlight it. Okay, so read it and then answer it. I'm giving you time, read it and answer it, guys. Read it and answer it. They are asking about the false statement, not the right one. False statement is, बच्चे, in aquatic ecosystem is your detritus food chain is the major conduit for energy flow. Is it the major channel for energy flow? Of course not. So it is incorrect. The terrestrial ecosystem, there is a larger fraction of energy flow through grazing food chain. Means, read it again. In terrestrial ecosystem, they are saying there is a larger fraction of energy flow through grazing food chain. Actually, this terrestrial ecosystem, this terrestrial food chain is itself the example of grazing food chain, right? The detritus food chain is just connected there. So organism at each trophic level depend on those at the lower trophic level for their energy demand. It's true. The number of trophic levels in the grazing food chain is not restricted. Yes, the number of trophic levels, they are not restricted, they are, because that is how the energy flow will be. No, so 1, 2 and 4, they are false. Okay, next question, right, read the following statement. Predators keep prey population under control. Nearly 25% of all insects are known to be phytophagous. Predation, parasitism, commensalism share a common characteristics. The interacting species do not live closely together. Endoparasite live inside the host body. So how many statements are correct? How many statements are correct? Yes, bache. So I'll get a water bottle. Till then, you please answer the question. I'll go and get a water bottle. Yes, bache. How many statements are correct? So which statements are correct? They are saying endoparasite live inside the host body at different sites, obviously, right? Endoparasites are the one that will live within the host site. That is a true statement, isn't it? That is a true statement, isn't it? Predators are going to keep the population, prey population under control. That is also true. So out of these two, which one is correct? Which one is correct? Yes, you are right. Right. You are right that C is the. You are right that 3 is the correct answer. But tell me, out of these two, which one is correct? Which one is correct? Hanji, quick. Quick. B or C? Guys, you have to answer it to me. They are saying predation, parasitism and commensalism. देखो, predation may be plus minus, parasitism is also plus minus, commensalism is also plus 0. They share a common characteristic. The interacting species do not live closely together. No, it's not like that. They have to live closely together. Okay, so this one will be the correct one. Next, okay, consider following genetic disorder and how many of are the Mendelian one? Which of them are Mendelian disorders? Yes, bache, which of them are Mendelian disorder? Answer it. Which one is the Mendelian disorder? Which one is the Mendelian disorder? Yes, which one is the Mendelian disorder? Mendelian disorder. Yes, Mendelian disorder is the word. Okay, they go. Your Klinefelter is what? Klinefelter, your Turner syndrome, right? Even. What are they? So 1, 2, 3, 4, 5, 6. Yes, what are they? They are basically chromosomal disorders. So rest are your Mendelian disorders. Rest are your Mendelian disorders. Okay, they are your Mendelian disorder. Next, fill in the blanks. Regulation of lac operin by dash is referred to as dash. The regulation of lac operin by, obviously I told you in your syllabus it is the negative regulation. Why? Because your I gene, regulatory gene, is forming a repressor. Okay, so what should be the answer here? Right, it is the negative regulation. Why? Because repressor is there. So 1 is the correct answer. Next, select the incorrectly matched pair. Select the incorrectly matched pair. Quick, guys, you will get the PDF of this. You will get the PDF in the Telegram group. So which one is the incorrectly matched? Which one is the incorrectly matched pair? So adapter RNA means your tRNA. Is it inverted or L-shaped? True. Catalytic is ribosomal RNA. Smallest RNA is mRNA? No. Soluble RNA is tRNA. So second is the correct answer. Second is the correct answer. Next, which of the following are similarity between periplaneta and ferritima? Periplaneta and ferritima. Right, which of the following are the similarities between periplaneta and ferritima? Similarity. So before answering this question, right, before answering this question, you should know that ferritima belongs to phylum Annelida. Periplaneta belongs to phylum Arthropoda. Okay, so if you know it, you can answer it. Do they have open blood circular system? Open type of blood circulation system is in Arthropoda but not in ferritima. So this is not the similarity. Internal fertilization, acha. Dioecious, both are dioecious? No. This one is monoecious, so this is also not. I'll come to the similarity part. Both will have dorsal ventral nerve cord. True, bache. Indirect development means larval stages will be there. Do you consider larval stages in earthworm? That is also the question. So now when it comes to the internal fertilization, do they have that? You can recall the cockroach structure also, right? If you don't know anything, you can recall the cockroach part also, right? You can just recall the cockroach, the development. Okay, the development. So guys, in both of them, see, if you have to talk about the similarities, right, in their case, yes, double ventral cord is the similarity. Okay, but the internal fertilization is not in both the cases. Okay, it is not in both the cases. Indirect development is also not in both the cases. So when it comes to the similarity, it will be your only D. Okay, it can be your, it is your only D. Okay, next. Earthworm have no skeleton, but during the burrowing, the anterior head end become turgid and act as hydroskeleton. I know earthworm is not the part of your syllabus, right? But sometimes, you know, they ask the question. So try to answer this. Try to answer this. Yes, bache, cockroach ka session will also be there. Don't worry. Earthworm have no skeleton, but during burrowing, the anterior end become turgid and act as hydroskeleton. It is due to quick, answer it. Yes, answer it guys. Just a minute. Yes, bache. They are saying earthworm they don't have, right? Earthworm have no. It contains no skeleton, but still, bache, still. Obviously, why is it so? Because of blood. Setae. Setae is for the locomotion. Is it the gut paralysis? Definitely not. Definitely not. Then what should be the correct answer? What should be the correct answer? Yes, everyone. What should be the correct answer here? Hydroskeleton, kind of. So here it will be coelomic fluid. Okay, here it will be coelomic fluid. Now guys, can I show you something interesting? Can I show you something interesting? Okay, so first of all, do check it, mistakes to avoid while filling the OMR sheet, and soon, see, be ready for it. Most predicted NEET 2024 paper, right? Be ready for it. Most predicted NEET 2024 ka paper, right? So here obviously you will get that, right? You will get that questions, that 200 questions of course, okay, and it will be your most predicted paper. Imagine, it will be your most predicted paper. And I still remember that yes, we have to revise the entire biology once again. So after it you will get an idea. You will get an idea that where are you guys making the mistake. So you have to put that in the comment section. I will pick that topics and I will take that in the one shot of the biology. So after it I will take another one shot video. So tomorrow I hope you know that we will be discussing all the disorders, because from disorders 2 to 3 questions. How many questions are, I don't know why the screen doesn't work when I use it in this way. Okay, so see, I'll tell you. Tomorrow at 11 AM we have the session all disorders of biology in one shot. You know that when it comes to the disorders, at least 3 to 4 questions from disorders will come in your syllabus. Human physiology say, from your genetics also. So whatever diseases are given, whatever disorders are given in your NCERT, even from human health and disease, whatever is given, even from STIs, from your reproductive health, whatever points are given, I will pick that up, and here we are going to finish it. So this session will be for around 2 hours or less than 2 hours. So less than 2 hours matlab at least you can consider 4 to 5 questions. So basically we will prepare for 20 marks tomorrow separately. This is the one thing. Now tomorrow itself you have this session, because we know that you people were demanding the physics sessions more and more. So Yawar sir is going to take the session with all high weightage chapters. Okay, bache. Now here the most predicted NEET ka paper, just take it, just practice it on 1st of May. Then you will get my sessions, like you will have my session on this biology one shot, where all the most important topics will be catered. Then the diagram based revision sessions will also be there, because from the diagram itself we get so many questions. Okay, so one will be the complete biology revision with the help of diagrams, one will be the complete biology revisions with the most important topics. Is that clear? Right. And for the cockroach also, for the biology also, for the cockroach also, for the biology also, for the frog also, you will get a session of less than 60 minutes. Okay, I will cover one main topics in one shot. Okay, so now you have your physics ka paper. This organic chemistry ka 90% questions will come from this. So same way mein you will get the biology paper also. It's not like that you are not going to get the biology paper, you will also get it. So here most predicted NEET 2024 paper will also be there. Right bache, and you will also get the biology ka revision, biology ka paper prediction also. Fine. Okay, so let's start the question practice again. Let's move to the next question. Which of the following are incorrect for platyhelminths? Which of the following are incorrect for platyhelminths? Quick, answer it to me. Yes, bache. Which of the following are incorrect for platyhelminths? Right, so they are saying that they have the dorsal ventral flat body. Yes, bache, hooks and suckers are in the parasitic form. All are bilateral and triploblastic, but all are endoparasite? No. They are free living also, and you remember planaria, it's a free living platyhelminth. So of course one is the solution here. Next, testes of cockroach located in. Such type of questions can come in paper, right? Bache, testes of cockroach, they are located in, right? Testes of cockroach, they are located in. Quick, bache. Testes of cockroach, they are located in. It's a very obvious question. Obviously 4 to 6 abdominal segment, right? 4 to 6 abdominal segment. Now let's move to the next question. When frog is on land and then respiration takes place only by, frog is on land, then respiration will be from, yes people, then respiration will be from, frog is on land. Frog is on land. So when frog is on land, skin, buccal cavity and lungs, all the three, right? From all the three. And guys, first of all, hit the like button here. Yes, do you want this session? If it is a yes, quickly hit the like button and you have to click on this, that is notify me. Quick, hit the like button right now. Right now just hit the like button. I want to see the increase in the likes here, and click on this notification on. So tomorrow at 11 AM I'll be taking your session. Okay, yeah, I will share the PDF with you guys. Quickly hit the like button. Hit the like button guys, and click on this button, that is notify me. You will get the notification and you have to attend this session because we will complete all the disorders in one shot. All the disorders with their explanation, right? We are going to cover it, and we will be starting with the chemical control and coordination. We will start from that chapter because in that chapter hormones, their disorder, everything is written now. So that's how we have to start. But I'll be very fast, but if I will be very fast, na technical Guruji, then you will not be able to understand anything. I have to explain it as well, because from one question the another kind of questions can also come. Okay, so quickly hit the like button guys, and here click on this button, that is notify me. You will get the notification of this session tomorrow. Quick. You are very bad, right? Some more likes, very good. Some more likes, want to see you all there, and the session will be for less than 2 hours. Okay, I'm not going to waste your time as well. Okay, chalo. So let's go. Vasoconstriction causes. Vasoconstriction causes. Guys, quick. Very good, Leharika. Vasoconstriction causes. What does vasoconstriction means? To constrict the blood vessel, that is definitely going to increase the blood pressure, isn't it? That is definitely going to increase the blood pressure. So here this should be the correct answer. Then fluid nature of plasma membrane is important in, fluidity. Yes, fluid nature of plasma membrane is important in. Fluidity is the word. Definitely that is for the endocytosis, for the secretion, for the cell growth as well. So if you remember, plasma membrane is a phospholipid bilayer. So these phospholipids, they can just exchange their position in their vicinity, right, in the same leaflet. This is what fluidity is. So for the growth, for the secretion, for endocytosis, it is important. Let's move to the next question guys. Read the following 4 statements, right, and then you have to find out which statements are wrong. So again in the paper, when you get such questions, right, again in the paper, when you get such question, please highlight this thing. Wrong is the word. Okay, wrong is the word. So how many of the above statements are wrong, right? Read it again, read it carefully. Read it carefully. Is in amphibia and reptile incomplete double circulation present? Yes. In arthropod and mollusca, open circulatory system is there. In joint diastole stage, semilunar valves are closed. Yes or no? Okay, the hepatic portal vein carries blood from intestine to the kidney, not to the kidney, to the liver. So this is the correct answer. Right, bache? This is the correct answer. Joint diastole में semilunar valves, they are closed, they are not open. They are not open. Next, each human kidney has nearly, okay, simple. Each human kidney has nearly, simple. Yes, bache, Anuradha bache, Akshita, Pranav, exactly, near about 1 million nephrons. Next question. Who explained that cells divide and new cells are formed from pre-existing cell? Omnis cellula e cellulae. Omnis cellula e cellulae. Omnis cellula e cellulae. Omnis cellula e cellulae. So it is the Rudolph Virchow. It is the Rudolph Virchow. Right, bache? What's the correct answer? It is the Rudolph Virchow. Next. Which of the following gland is divided into two parts, central medulla and peripheral cortex? Which of the following gland is divided into two parts, central is medulla and peripheral cortex? I think I have repeated it so many times. These three are having this. So out of all the options given here below, this will be your correct answer. Right? Your adrenal gland is also having the adrenal medulla, adrenal cortex. Ovary is also having the same. Okay, now let's move to the next question. Subunit of ATS ribosomes yeah, are. Subunit of ATS ribosome are. Yes, students. Subunits of ATS ribosome are ATS, 60S, 40S. Right? What is the S here? It is the sedimentation coefficient. It is the sedimentation coefficient and it is nothing but it is the Swedberg unit. It is nothing but it is the Swedberg unit. Got it? Next, ball and socket joint can be seen in. Ball and socket joint can be seen in. The word is ball and socket joint can be seen in. Right? It can be seen in. The word is ball and socket joint. One question will definitely come from the joints. One question will definitely come from the joints. Okay, so of course it is in the shoulders. Right, bache? DNA replication and centriole duplication occurs in which phase of the cell cycle? DNA replication and centriole duplication occurs in which phase of the cell cycle? It is your examiner's favorite question. Right, the duplication of centriole, and so we know that it is the S phase. It is the S phase. Now let's move to the next question. During pregnancy, most of the major organ systems are formed by the end of, yes, but in the pregnancy, right, the major organ system, they are formed by the end of 12 weeks of pregnancy, isn't it? Isn't it? That is your first trimester. That is your first trimester, isn't it? What should be the answer? By the end of 20 week or by the end of 3 weeks? During pregnancy, major parts, most of the major organs, they are formed by somewhere near about 24 weeks. Guys, you really need to check it. You really need to check it. You really need to check it. Right, the first is the correct answer here, by the end of 12 weeks, first trimester. Then which of the following coats cover testes all around? Which of the following coats cover testes all around? There is a trick here again. Yes, bache. The coverings of the testes. You remember the covering of the testes? We have tunica, vaginalis, albugina, vasculosa. Do you remember that? So they are saying which of the following coats cover testes all around, means like this, right? Complete covering is the word here, right? This is the trick for the coverings: tunica, vaginalis, albugina, vasculosa. But which one is going to complete it all around? Which one is going to complete it all around? And that is your tunica albugina. That is your tunica albugina, right bache? That is your tunica albugina. Next, gap between meiosis and meiosis is meiosis 1 and meiosis 2 is known as, gap between meiosis 1 and meiosis 2 is known as, I believe you know the answer, one of the easiest question, it is the interkinesis. So in interkinesis there will be no DNA replication. Okay, next question, which one of the following bond is formed between N-terminal and C-terminal in the proteins? N-terminal and C-terminal in the proteins. Which bond will form there? Yes, bache, which bond will form there? Which bond will form there? N-terminal and C-terminal. N-terminal and C-terminal. Peptide bond is your CONH na. So they are asking which bond will form between N-terminal and C-terminal in proteins. Dekho, overall I was expecting this. Dekho, when you write down, you know that we have mainly L amino acid in our body, right? L amino acid in our body. Basically you have N, right? Amino group is there, right? You remember na? Do you remember that? Right, we say that mainly we have the L amino acid in our body, mainly this is what we discussed. Now when you talk about the bonding, right, and this L amino acid means this is how, right, the first amino acid will be having its NH2 group free, if you remember that. Okay, so here you have CONH, that's how the bond will be na, that is how the bond will be. This is your next amino acid, this is your next amino acid. So you know that peptide bond will form like this. Okay, so the water will release and you will be left with CONH. Okay, you will be left with CONH. That is what you guys are going to get. You will be left with CONH. This is your peptide bond. Am I right or wrong? So that, but now they are saying which of the following bond is formed between N-terminal and C-terminal. They are not asking that how amino acids are going to connect. Let's say you have a chain of protein, yeah, polypeptide. One side is free N-terminal, another is free C-terminal. So this is how they are asking, right? This is the question here. So understand the question first. Understand the question first. So peptide bond will form in between the two, right? Peptide bond will form in between what? It is going to form in between, yes, your N and C, right? But in the amino acid, but they are saying which one of the following bond is formed between N-terminal and C-terminal in proteins. So answer it, check it, right? Check it. Just check this out. So this is your homework, right? I'm giving you two options, either peptide or ionic. What should be the answer here? What should be the answer here? Okay, so this is your homework. You will answer it to me. Okay, next question. Antivenom and anti-serum are the examples of passive immunization. Antivenom, which is given to patient, contains preformed antibodies against snake venom, right? Against snake venom. Now what should be the answer here? What should be the answer here? Yes, people, anti-venom and anti-serum, they are examples of passive immunization. The reason they are mentioning here is that anti-venom, which is given to patient, contain preformed antibodies. Firstly, tell me, are they example of passive immunization? Yes. Why? Because here preformed antibodies are inserted in our body, right? It will give us the effect immediately. It will not be long lasting, but it will give us the effect immediately. So yes, reason is explaining the assertion. So 1 will be the correct answer here. Next, read the following 4 statements, and again you have to mention incorrect statement. So while answering such question, please highlight the word correct and incorrect. Please highlight the word correct and incorrect. So guys, answer it to me. Read the following statements, following 4 statement, and answer it to me. Yes bachche, answer it to me. Quick. Yes bachche, what should be the answer here? Yes bachche, what should be the correct answer here? Incorrect statement. Annelids possess longitudinal muscles only which help in locomotion, right? So do they just have longitudinal muscle? They have the circular muscle also, one statement. Closed type circulatory system is present, true. Nephridia do not help in excretion and osmoregulation, false. Members of phylum Annelida are triploblastic, metamerically segment and acelomate, this is also false. Okay, so Annelida possess longitudinal muscles only, this is also false. So 1, 2, 3. 3 statements are false statements. So second is the correct answer. Second is the correct answer. Done. Move to the next question. Which statement is true about the diagram given above? Yes, which statement is true about the diagram given above? So it is from structural organization in animals, guys. It is from structural organization in animals. Is it composed of tall and cylinder cells? No. No, they are flat and irregular. Is it the example of compound epithelium? Yes, bachche. Right. So is it found in nephron? No, because it is the compound squamous epithelium. Their main function is to form diffusion boundary? No, because that is the function of simple squamous epithelium, but it is having the limited role in secretion and absorption. It is having the limited role in secretion and absorption because it is mainly for the protection. Next, which statement is false? Which statement is false? Yes, bachche. Which statement is false? They are asking false statement. They are asking false statement. Obviously, chordae tendineae found in auricle, they prevent the backflow of blood. Are chordae tendineae in the auricle? You remember the ventricles here, these papillary muscles here, and they are having these chordae tendineae which are attached to that, you know, flaps, bicuspid, tricuspid mein. So this 3 is the incorrect one. This 3 is the incorrect one. Next question. Synaptic cleft is always present in the chemical synapses, right? Secondly, electrical potential difference across the resting plasma membrane is called as the resting potential. So what should be the answer here? 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 Both the statements are correct. Yes, bachche. Because electrical potential difference across the resting plasma membrane is the resting potential. So synaptic cleft is always present in chemical synapses. Next question. Which of the following is present in pons? Hanji. Which of the following is present in pons? You know na? It is the part of hind brain. Do you remember this? It is the part of hind brain. If you remember, hind brain is having 3 parts. The trick is physics, chemistry, maths. So cerebellum, pons viruli, medulla oblongata. So obviously pneumotaxic center. Okay, pneumotaxic center is present there. Next, FRC, functional residual capacity is. FRC, functional residual capacity is. Functional residual capacity is. Yes, everyone, I believe you know the answer. Functional residual capacity is. Yes, functional residual capacity is, do you remember the trick? देखो, you have inspiratory capacity, expiratory capacity, total lung capacity, sorry, vital capacity, total lung capacity, functional residual capacity. So here you have tidal volume, tidal volume, tidal volume, tidal volume. You can write down inspiratory capacity, expiratory capacity, vital capacity, total lung capacity, and functional residual capacity. So here keep the tidal volume common. So when it is the inspiratory capacity, inspiratory reserve volume. When it is the expiratory capacity, you have to add the expiratory reserve volume. When it is the vital capacity, you have the inspiratory reserve volume and expiratory reserve volume. When it is the total, then you have to even consider the residual volume, right? So when it is the residual capacity, residual volume, and expiratory reserve volume, that is how you have to remember it. So FRC means residual volume, and this 3 is the correct answer, right bache? Next question. Okay, again, points about red fibers, right? You have to find the correct points about the red fibers. Yes bache, correct points about the red fibers. Quick, how many points are correct about the red fibers of muscles? How many points are correct about the red fibers? They are red, they are red. Yes bache, they are red. Reddish appearance will be there. Okay, so do they have myoglobin content? Yes bache. Aerobic process for ATP reproduction? Yes bache. So out of mitochondria and sarcoplasmic reticulum, which one is more? Which one is more? Yes bache, which one is more? Tell me. You are right, this is the correct statement. So in red muscle fiber, which thing will be more? Which thing will be more? Red muscle fibers में which thing will be more? Yes bache, they are rich in myoglobin content and they are rich in mitochondria. In their case, right, this is not there. Sarcoplasmic reticulum is comparatively low. ठीक है बच्चे? So A, B, D and E, these 4 statements are the correct one. These 4 statements are the correct one. Now move to the next question. Which of the following method of contraception is effective only up to a maximum period of 6 months following parturition? After parturition up to 6 months, right? After parturition up to 6 months. Yes बच्चे, up to 6 months. Exactly, it is your lactational amenorrhea, because during this time the gonadotropins are not there, but yeah, the prolactin is high. Okay, next question. Which of the following have been found as very effective emergency contraceptive? The word is very effective emergency contraceptive. Very effective emergency, the word is emergency here. Okay, the word is emergency here. Which one of the following is the emergency contraceptive? Yes students, yes students. So of course progesterone, estrogen combinations and IUDs. Okay, so match the column 1 with column 2. Match the column 1 with column 2. It is on the basis of their size. Virus, bacteria, mycoplasma. I think we all know this, right? I think we all know this, isn't it? So 1 will go with B. So eliminate these 2 options, right? RBC is 7 micrometer. 3 will go with C. So you can eliminate this option. So bacteria is basically, yes bache, okay, my bad. 3 will go with D. So yes, this is the option. So bacteria is basically somewhere 3 to, okay, and viruses are right bache. Okay, so what should be the correct answer? What should be the correct answer? Second is the correct answer here, right? Mycoplasma, you know na, smallest living cell. So next, how many of the following are non-phagocytotic? The word is non-phagocytotic. Dekho, you know that monocytes, your neutrophils and your macrophages, they are phagocytotic, isn't it? They are phagocytotic, isn't it? So only your D lymphocyte is not phagocytotic. So 1 will be the correct answer, right bache? Only your B lymphocyte will not be phagocytotic, so 1 will be the correct answer, right? So after the formation of product in the bioreactor, it undergoes through separation and purification process before a finished product is ready for marketing. These processes are collectively referred to as, yes, these processes are collectively referred to as, obviously it is the downstream processing. What will be the correct answer here? It will be the downstream processing. Next, due to ampicillin resistant gene, one is able to select a transformed cell in the presence of ampicillin. So it is the example of, these are the questions which you just have to hear and answer. Obviously selectable marker. Move to the next question. PCR stands for, again very simple, polymerase chain reaction it is, right? Next question, the proteins encoded by the genes Cry1AC and Cry2AB. Again, it is also an obvious question. Cry1AC and Cry2AB, exactly, Cry1AC and Cry2AB, cotton bollworms. Next, identify the correct statement with respect to the development of transgenic animals for the study of disease. Identify the correct statement with respect to the development of transgenic animals, right, for the study of disease. They are designed to increase our understanding of role of genes in disease. They are developed as model for human diseases. They exist for Alzheimer's, so all of these will be the correct answer. Next, select the false statement with respect to the epithelial tissue. Quick, select the false statement with respect to the epithelial tissue. Yes, bache. False statement. The word is false statement. Bache, is it going to form middle structure part of organs? Never. Right, epithelial, it is mainly for the covering, for the protection. So definitely, right, which one? Fourth is the correct answer. Next, capillaries which run parallel to loop of Henle. Right, if you are going to make mistake here, you will be punished for sure. You guys will be punished. It is something that easy. Of course, it is the vasa recta. Next, when a neuron is in the resting state, that is not conducting any impulse, the axonal membrane, right, now it is in a resting state, it is not conducting any impulse, okay, it is not conducting any impulse, it is not conducting any impulse. It is not conducting any impulse. Obviously, it is more permeable to potassium and nearly impermeable to sodium ions. Next question. Select incorrect match. You have to select the incorrect match. Incorrect match. Is WBC is the cell without nucleus? No, ma'am. Done. Again, match the column type of question. Now this speed is fine, right? This speed is fine, everyone. This speed is fine. If it is fine, just send the fire emojis in the chat section quick. So after physics, there will be the chemistry revision, and then there will be, sorry, after biology, then chemistry, then physics revision will be there. So guys, answer it to me quickly. Answer it to me quickly. Right? Are you feeling confident? The speed is fine. Are you feeling confident? Speed is fine. Then answer it, yeah. Tell me. It's good, right? Okay, so match the following: growth hormone, luteinizing hormone, prolactin and FSH. So growth hormone, wow bhai, don't you think it's easy, right? Don't you think it's easy? See, I will just see A is with 3, right, in all the option. Growth hormone is diabetogenic hormone. Okay, fine, we got it. What about the luteinizing hormone? You know that it will stimulate your, right, is it going to stimulate your Sertoli cell? That will be done by FSH. So your D will go for 2, right? And what about your luteinizing hormone? It is for the ovulation for sure, right? So if you know B is with 1, that is obvious, you got your answer. Next, by the contraction in diaphragm, the volume of thoracic chamber increases in the, there is a contraction is diaphragm, right? The peripheral muscles of the diaphragm, they will contract. So obviously the volume will increase towards the anterior posterior axis, Anterior posterior axis, yes ma'am. So if there will be the contraction of external internal muscle, then there will be the change in the dorsal ventral axis. Volume will change in the dorsal ventral axis. Okay. Next, match the following and choose the correct answer. Okay, match the following and choose the correct answer. Match the following and choose the correct answer. Yes, oogenesis, fertilization, implantation, functional maturation of sperms, and oogenesis. Yes, bache. So where will the fertilization occur? In the ampulla, of course, right? Implantation will be there in the uterus, definitely. Maturation of sperm will be in the epididymis. So oogenesis is associated with the ovary. So what should be the correct answer? A with 3, B with 1, C with 4, D with 2. You got your answer, right? You got your answer. Next question, right? Statement 1 and statement 2. ER divides the endoplasmic reticulum, divides the intercellular space into 2 distinct compartments. RER involved in protein synthesis and excretion. So now tell me, what should be the answer? What should be the answer? Both are correct, right? Both are correct, right? Endoplasmic reticulum, membrane bound it is, na? It will divide the intracellular space into 2 distinct compartments, right bache? Yes bache, it will divide it, definitely. So RER, rough endoplasmic reticulum, because ribosome is associated, so it is playing role in protein synthesis. Next, agriculture and human settlement was started about. Agriculture and human settlement was started about. Human settlement and agriculture, again it is from evolution, so yes, 10,000 years back. Next, malignant tumors exhibit metastasis. Contact inhibition is the property of malignant tumor. Very easy. Contact inhibition is the property of malignant tumor. Yes, yes, yes. Malignancy, right? Cancerous tumor, M, metastasis will be there. Means this tissue will invade the different locations in the body. Clear bache? Right, so contact inhibition is the property of malignant tumor? No, it is not. Right, so statement 1 is correct, but not second is incorrect. Right, baches? 1 is correct, second is incorrect. Next, which one of the following option gives the correct categorization of 6 animals according to the type of nitrogenous waste? It's a good question. It's a good question. Read it. It's a good question and answer it. It's a good question and answer it quickly. They are saying that which one, right, gives the which one is a monotalic, ureotalic, or uricotalic? They go, pigeon and human is definitely not a monotalic. Frog and lizard, lizard are also not a monotalic. Okay, then ureotalic. Cockroach is definitely uricotalic, so this cannot be your option, right? So you are left with 1. So aquatic amphibian, frog and human, pigeon, lizards, and cockroach. So 1 is the correct answer. Next, analysis of urine helps in clinical diagnosis of many metabolic disorder. Reason, glycosuria is an indicative of diabetes insipidus. Again, it's a very simple question. Again, it's a very simple question, people. It is a very simple question. See, the second statement is incorrect because glycosuria, it indicates diabetes mellitus, not diabetes insipidus. Right, bache? So reason is false. But analysis of urine helps in clinical diagnosis of many metabolic disorder. 3 should be the answer. Next, a certain patient is suspected to be suffering from acquired immunodeficiency syndrome, that is AIDS. So which diagnostic technique will you recommend for its detection? Yes. Guys, quick. Very good, very good. Out of all the options here, ELISA is the answer. But always remember, it is the Western blotting that will be confirmatory. Okay, it is the Western blotting that will be confirmatory. Understood? Understood. Next question, next question, next question. Column 1, column 2. Column 1, column 2. Match it. Yes, match it. Column 1 with column 2. Just match it. Column 1 with column 2. Just match it. Again, easy. Yes, bache. See, PCR amplification of gene, 1 with D. Eliminate these two options. Bioreactor is for the large scale culture. 2 with A. Yes, bache. Gene gun to induce the alien gene in the host DNA, of course. 3 with, go with B. You got your answer. Eco R1 is the restriction endonuclease, and it will recognize GAA-TTC. Remember, it will recognize gatus. It will recognize gatus. It will recognize gatus. It will recognize gatus. ठीक है बच्चे? Okay. So which is phosphorylated lipid? Which is phosphorylated lipid? Which one is phosphorylated lipid? Again a very simple question. Phospholipid, basically in simple words, it is phospholipid, lecithin. Okay, next question. Predators act as conduit for energy transfer across trophic level. Predator keep prey population under control. Again, this is a very simple type of assertion and reason. Again, it's a very simple type of assertion and reason. Yes, bache? Yes, bache? देखो, this statement is correct that predators, they act as channels for the energy transfer across the trophic level, because predator, right, the one that will feed on some another organism, that is how the energy will transfer. Secondly, they keep the prey population under control, that is also true, but reason is not explaining the assertion, so that's why second will be the correct answer. That is why second will be the correct answer. Clear, bache? Clear? Then the pyramid of biomass in sea is generally inverted. The biomass of fishes far exceeds of phytoplankton. The pyramid of biomass in sea is generally inverted. The biomass of fishes far exceeds that of phytoplanktons. Right? They far exceeds that of phytoplanktons. They far exceeds that of phytoplankton. Yes, bache? Pyramid of biomass in sea is generally inverted. That's a true statement, okay? That's a true statement, right, bache? So biomass of fishes far exceeds that of phytoplanktons. They are saying biomass of fishes far exceeds that of phytoplanktons. Yes, bache? Yes. Why this pyramid is in biomass is inverted? These are the phytoplanktons. They are supporting the organisms which are having more biomass than them. That's why this is the correct answer. So that is why first is the correct statement. First is the correct answer here, because reason is explaining assertion. Next question: what is the percentage of power in the incident solar radiation? Yes, in the incident solar radiation, what is the photosynthetically active? Right, what is the amount of that? That is 2, less than 50%, okay? That is 2, less than 50%. So observe the given pie chart representing biodiversity of different vertebrates. And guys, again, just for your quick reminder, today this is what we are going to see. Right, all the disorders of biology in one shot, and that will be in less than 2 hours, and that session will be very effective. Right, why? Because 3 to 4 questions minimum they can come from the disorder. So what you have to do, click here. Alright, let's make it 100 plus right now, and see, notify me, click on it, notification on. So you will get an update about it. Okay, so just hit the like button. So tomorrow I want to see you all in this session, that is at 11 AM in the morning. Then there will be the physics class as well. Okay, and tomorrow I will also put one video to revise the photosynthesis in less than 30 minutes. Good, great, to revise the photosynthesis in less than 30 minutes. Okay, done. So just have a look, guys. So what should be the correct answer here? What should be the correct answer here? So A is fishes, of course, then mammals, and your C will be amphibians, because they are same, and here it will be birds. So 1 is the correct answer. Next, Match column 1 and select the correct option again. Match column 1 and select the correct option again. Yes, bache. So remember that example based session, right? In that example based session we have revised it. Yes, people. So dodo, 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 Mauritius, Mauritius. Yes, bache. Stellar sea cow, Russia, D with 1. Right, so dodo is basically what? It is Mauritius, right? Mauritius. So obviously you can eliminate these two options. So quagga, quagga, quagga is for, yes, is it Australia or Africa? Is it Australia or Africa? Which one should be the correct answer? Yes, you know the answer, guys. Answer it to me quickly. Answer it to me quickly. Answer it to me quickly. What should be the correct answer? The quagga will go for Africa. Thylacine will go for, yes, bache, Australia. So 3 should be the correct answer. Dodo, Mauritius, quagga, Africa. Thylacine will go for your this Australia, stellar sea cow, Russia. Okay, 3 is the correct answer. Okay. Next, this, right? This. Okay. So it is from the organism and population. Is it from the organism and population? So this is very important. This is very important, right? One question or two questions from this population interaction can come. Protocorporation, commensalism, amensalism, parasitism. Hanji. So where are you going to see the protocorporation, commensalism, amensalism and parasitism? Yes, students. Yes, what should be the, I'm giving you the time to revise it. So sorry, to read it carefully. So tell me. See bache, protocorporation, hermit crab and sea anemone. Commensalism will be with your sea anemone and clownfish. Amensalism, commensalism you know na, +0. Amensalism mein -0 will be there. So -0 will be in the case of, yes bache, it will be in the case of, yes. Parasitism is Parthenium. So what should be the answer here? What should be the answer? Is it 3 or 4? Protocorporation is your hermit crab. Commensalism is your sea anemone and clownfish. Amensalism is Parthenium, right? Parthenium, because it is the intruder. Okay, so it is not getting affected. It will affect 1. So C with 1. Yes, I got my answer. C with 1, right? Amensalism with 1. So obviously parasitism will go for this cuckoo and crow, and that is the example of brood parasitism. Next. Which of the following are the cause of biodiversity loss? Which of the following are the cause of biodiversity loss? So you know the answer, right? It is HOAC. You know the answer, right? It is HOAC. Right, bache? It is HOAC. Okay, so habitat loss and fragmentation, overexploitation, alien species invasion and co-extinction. So obviously it will be 1 and 2. It will be 1 and 2. So means 4th will be the correct answer. 4th will be the correct answer. Then emasculation of flower done during, emasculation of flowers done during, emasculation of flower done during, yes, emasculation is the removal of anther. Yes, bache, it is the removal of anther, and that will be done when stamens are immature. Why? Because once they will be mature, they will shed their pollen grain that can fall on the stigma, and here we want to avoid that. Okay, right? So next question. In which case female has a pair of dissimilar chromosomes and, right, dissimilar chromosome, and male 2 similar chromosomes. Okay, female is basically heterogametic. We know the answer here again. Don't you think that will be birds? Birds. Next, which of the following option is not true? Which of the following option is not true? Quick guys, which of the following option is not true? Guys, we do not have time for break and to chill because it is 29th today. Hai na? It is 29th today, right? And tomorrow 30th, then 1st, 2nd, 3rd, 4th and 5th. Okay, so we cannot We cannot waste our time, right? You will get the break. Right after this, your Wazeem sir is going to start the class. Let me contact him, right? So we'll tell you about it. But as of now, focus here. Focus. These questions are very easy. Okay, these questions are very, very, very easy. And trust me, your NEET paper is also going to be easy. It's just that you need to understand that you have to reduce the negative marking and you have to manage the physics, chemistry, biology all together. Okay, so which of the following is not true? Answer it to me. Which of the following is not true? Mendel experiment has the large sampling size. Yes. In pea plants, axil flower position is a dominant trait. Yes. Mendel selected 14 true breeding pea plant varieties. Hanji. Mendel conducted hybridization experiment on sweet pea. No, it was pea. That is your Pisum sativa. Okay, the sweet pea will be your Lathyrus species. Sweet pea will be your Lathyrus species. Next, the discontinuously synthesized fragments are joined together by. Yes, bache, they are going to join together by obviously molecular glue, that is your DNA ligase. Molecular glue, that is your DNA ligase. Next, select the incorrect match. Select the incorrect match. Select the incorrect match. Again, Z gene is going to give you beta galactosidase, Y will give you permease, A will give you transacetylase. Y is going to give you the repressor, but that is as per NCRT. That is as per NCRT. If you will study the same topic in the higher classes, you will find that I gene also form activator. But okay, that is not the part of our syllabus. So that's why we are marking the 4 here. Next, in Human Genome Project, the total estimated cost of the project would be approximately $90 billion US. Okay, so lac operin consists of one regulatory gene. I does not refer to inducer, rather it is. Okay, so what should be the correct answer? What should be the correct answer? Guys, this statement is incorrect, but this one is correct. 3 is the correct answer. It is not 90 billion US dollar. It is not 90 billion US dollar. Okay, it is not 90 billion US dollar. Got it? Next question. A E. coli cell with completely radioactive DNA was allowed to grow in culture medium containing non-radioactive isotopes. In second generation, find out the percentage of radioactive and non-radioactive DNA. They are saying an E. coli cell with completely radioactive DNA, okay bache, was allowed to grow in a culture, then you have shifted it to the medium containing non-radioactive. So second generation, mein what will you see? What will you see in the second generation? The word is second generation here. The word is second generation here. You know na how that replication is? Is it the semi-conservative mode? It is the semi-conservative mode. So it will be 50-50. 50% will be radioactive, 50% will be non-radioactive. Next, which one of the following is most crucial step in Calvin cycle? Which one of the following is most crucial step in Calvin cycle? Most crucial step. Dekho, Calvin cycle means C3 cycle. Carboxylation is very crucial. Why? Because you know that ribisco is having two natures. It can also oxygenate, right? But it should not. That will be a wasteful process. That is the photorespiration. So carboxylation should occur here. Done, bache? Most abundant enzyme on earth is? Most abundant enzyme on earth is again ribisco, ribulose, bisphosphate carboxylase oxygenase. So ribulose it is. Ulose means ketose is the functional group here. Okay, ketose is the functional group here. Next, how many statements are true about the glycolysis? How many statements are true about the glycolysis? Right? How many statements are true about the glycolysis? Yes, bache. Yes, bache. True about glycolysis. It occurs in the cytoplasm of cell. Yes, ma'am. Present in all living organisms. Yes, ma'am. Glucose undergoes partial oxidation to form 2 molecules of pyruvic acid. Yes, ma'am. In anaerobic organism, it is the only process. No, ma'am. Actually, in the anaerobic organism, it is the only process. So, how many statements are true? 3 statements. Right? So, glycolysis is oxygen dependent or oxygen independent? Tell me. Glycolysis is oxygen dependent or oxygen independent? Hanji, yes, bache, Tharun Kumar, you will get that questions in your paper for sure. For sure, it will be more than 50. Hanji, answer my question. Glycolysis is oxygen dependent or oxygen independent process? Independent means without oxygen. Independent means without oxygen. Next, bache, consider the following reaction. RQ for this process. They are asking about the RQ value. Very simple it is. Respiratory question. Volume of CO2 evolved to the volume of oxygen consumed. Very easy it is. 1. Why? Because here you have the glucose. Okay. So it is 1. So 6. 1 it will be. Understood? Next, acceptor of acetyl coenzyme in Krebs cycle. We just discussed that. Acceptor of acetyl coenzyme in Krebs cycle. Acceptor of acetyl coenzyme in Krebs cycle. You know that, guys. You know that. Yes, bache. Who's going to accept the CO2? Of course, your oxaloacetate. The spelling error is there. It is oxaloacetate or oxaloacetic acid. Okay? So, can we revise Krebs cycle once again? Can we revise Krebs cycle once again? Yes, students. Can we revise Krebs cycle once again? Right? So, what is the trick? Come in and kick, scooter, fastly move on. Come in and kick, scooter, fastly move on. So guys, answer one question of mine. I'm not going to explain it. So fumarate is a 4-carbon compound or is it a 5-carbon compound? Fumarate, is it a 4-carbon compound or is it a 5-carbon compound? Fumarate, is it a 4-carbon compound or 5-carbon compound? Let me ask you the questions randomly. 4-carbon, 5-carbon, 4-carbon, 5-carbon. 4-carbon, 5-carbon, 4-carbon compound, done. Okay, so how many steps of substrate level phosphorylation will be there? How many steps of substrate level phosphorylation will be there in the Krebs cycle? Quick, guys. How many steps of substrate level phosphorylation will be there? Only one step. Which step? Can you just tell me? It's a break for you guys, right? It's a kind of break. You answer my questions. Obviously, one step, succinyl coenzyme A2 succinate. Okay, only in the Krebs cycle, right, bache? How many NADH will form? Arey, I am getting this message, yaar. It's a fun message. Wait, Anubhav is sending something. Anji, you tell me, how many NADH will form? I am not talking about the link reaction. I am just talking about the Krebs cycle right now. Okay. How many NADH will form? I am talking about one Krebs cycle. Of course, three steps. 3 steps. 3 NADH. 3 NADH. I am talking about only one round of the Krebs cycle. 3 NADH. So, can you tell me the steps also? Hanji Ankit, can you tell me the steps also? Isocitrate to alpha ketoglutarate to your succinyl coenzyme A also. And one more step is there. No, do not just go for the step number. Okay. So, Anubhav, what are you expecting us to do? Hanji, Anubhav, what are you expecting us to do? Right? This message is not going to hurt my feeling because that is a fact also. Okay? Even we have a family to take care. है ना? We have a family to take care, even we have to feed them, we are also the provider, so we need to earn as well. But whatever we can do as a noble cause, we are doing it. Okay, so from YouTube, if you are watching my lecture, I'm not going to get any money, I'm just justifying my job. Fine. So yes, even we want to earn the money, we have to be, we have a family to take care as well, na? And if you are ready, no ma'am, you don't earn money, you just teach free of cost, we will take care of your family, then okay, I'll be very happy. Ma'am, these questions will come for NEET. Yes, bache neha, because they all are PYQs. We have covered many previous year questions. And if you will check the paper, right, you will get many questions from the previous year papers only. Okay? Anubhav, that's not your cup of tea. Okay? So expect something that you can do. Okay, so now guys, let's move to the next question. Okay, so in where that Krebs cycle will occur? What is that location, the site of Krebs cycle? What is the site of that Krebs cycle? Site of that Krebs cycle. Be very specific. Array, be very specific. Mitochondrial matrix. Mitochondrial matrix. It is mitochondrial matrix. Okay. Okay. So all the enzymes of Krebs cycle, are they in the mitochondrial matrix? All the enzymes are in mitochondrial matrix? Yes, ma'am, except succinate dehydrogenase. Except succinate dehydrogenase. Right? Except succinate dehydrogenase. Understood? 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 Except succinate dehydrogenase. Why? Because this enzyme is there in the inner membrane of the mitochondria. Okay, it is just like a connecting link in between the Krebs cycle and your ETC. Now let's move to the next question. In pineapple, flowering and fruit set up are synchronized by dash. Ethylene increases number of dash, and complete the above statement by choosing the correct option there. Again, the easy one. Again, the easy one. Easy one. Quick, quick, quick. What should be the correct answer? The pineapple, flowering and fruit setup are synchronized by ethylene, and ethylene increases the number of female flowers and fruits. So second is the correct answer. So natural cytokinins are synthesized in the regions where rapid cell division occur. Okay, natural cytokines are synthesized in the regions where rapid cell division occur. Cytokine promotes nutrient mobilization, which promote leaf senescence. Is it so? Read both the statements carefully. Bache, they are saying ki cytokinin, which promotes the cell division, is it going to promote leaf senescence? Of course not. Of course not. So statement 1 is true, but statement 2 is false. Again, cytokinin, it provides the Richmond-Lang effect. That is, it delays senescence. Okay, it delays senescence. It delays senescence. Understood? Next question. Next question, with respect of the given graph, identify the wrong statement. Most important topic right now. Most important topic. Most important topic. Hanji, with the respect given graph, identify the wrong statement. Hanji, identify the wrong statement. Quick. Quick, quick, quick. Done? Dekho, this is related to the enzyme. Yes. Is it related to the activation energy? Yes. You know that A is the activation energy without enzyme. B is the activation energy with enzyme, and substrate is having more energy, product is having less energy. It means the energy is released. So it is basically the exothermic reaction. So okay, so you have to find the wrong statement. So yes, this is the incorrect one. A is showing activation energy without enzyme, correct. B is showing activation energy with enzyme, correct. Graph is showing exothermic reaction, correct. But is it showing endothermic reaction? No. For endothermic reaction, graph will be like this: substrate or product. Next question. Biodiversity refers to Biodiversity, it refers to, yes bache, quick. Biodiversity refers to wow, the simplest question, isn't it? It is the number and type of organisms present on the earth. And the thing that you discussed now, that there are more than 1.5 million organisms on our earth. So you consider only eukaryotes there, not the prokaryotes. Okay. Now let's move to the next question. Consider the following 4 statements and select the option which includes all the correct ones only. Okay, correct one only. Correct one only. Yes bache, it's easy. They go, archaebacteria differ from other bacteria in having a different cell wall structure. True. Bacteria shows the most extensive metabolic diversity. True. Cyanobacteria have chlorophyll B similar to green plants. It should be A. So false. Mycoplasma can survive without oxygen. True. So A, B and D. Done. Next. Deuteromycetes is known as fungi imperfecti. In Deuteromycetes, only the asexual phase is known. So you know that yes, Deuteromycetes is the fungi imperfecti because it doesn't have the sexual stages. It has the asexual stage only. So obviously, one it is. That reason is explaining the assertion as well. Next question. Gametes of Phaeophyce are. Very good. Gametes of Phaeophyce are. Very important. From algae, 1 or 2 questions you guys are going to get. Gametes of Phaeophyce are. Gametes of Phaeophyce are. Yes people, gametes of Phaeophyce are. Gametes of Phaeophyce are. You remember? Lateral, 2 lateral flagella they have, but it is pyriform. It is pyriform. Next, in dorsiventral leaf location of palisade parenchyma and phloem are. Right, in dorsiventral leaf means the leaf of dicots. Okay, so palisade parenchyma and phloem is towards the location of palisade parenchyma. What will be the location of palisade parenchyma? Palisade is this one. Okay, and spongy is this one. So palisade parenchyma is towards the adaxial side and phloem is towards the abaxial side. Okay, it is towards the abaxial side. So which one is upper, which one is lower? Which one is upper epidermis, which one is lower? Answer it to me. Which one is upper epidermis and which one is lower epidermis? Answer it to me. Yes students, answer it to me. Very good. Next question. Next question. Cells of grass leaves, they help in minimizing Okay, the cells which will help in minimizing transpiration are, of course, bulliform cells, also known as motor cells. Because of them, there is the rolling of grass leaf. Understood? Next, consider the following 4 statements and select the option which include all the 4 ones. Right? Which include all the 4 ones. Yes, bache, that will include all the 4 ones. Consider the following statement and select the option which includes all the correct ones only. Read the statements carefully. Innermost layer of cortex is called pericycle. No, it's endodermis. Okay. So you can cancel these 2 options. The tangential wall and the radial walls of the endodermal cells of roots have deposition of a waxy material, subrin. Okay. Initiation of lateral roots take place from the pericycle. Yes bache. Pith is larger and well developed in monocot roots. Monocot roots mein, is it well developed? Is it well developed? Yes. So what can you say? Is it your A is not the correct answer? Is it B, C and D, or is it C and D? B is also true, right? Subrin deposition is there, right? Subrin deposition is there, right? So it is B, C and D. Next question. Which one of the following is an example of opposite phyllotaxy? Opposite phyllotaxy. Just this is the one node and that is how the leaves are growing. Opposite phyllotaxy. The word is opposite. So obviously calotropis. Opposite. Opposite. Calotropis. Next. Mango fruit is developed from. Mango fruit is developed from. Yes, students. Mango fruit is developed from. Is it a true fruit? Hanji. It is developed from monocarpillary superior ovary. Monocarpillary superior ovary. Single cotyledon of monocot seed is called. Single cotyledon of monocot seed is called scutellum. You remember? It is scutellum. And alluron is that proteinaceous layer. It is also triploid. Remember that. And there is epiblast also. Epiblast is a reduced. Right? There is epiblast also. Epiblast is what? It is the reduced cotyledon. Okay, it is the reduced cotyledon. Next, geitonogamy is functionally cross-pollination and genetically self-pollination. Geitonogamy involves pollinating agents, but pollen grains come from same plant. You remember na, geitonogamy, functionally cross-pollination and genetically self-pollination, because it involves pollinating agent, but pollen grains come from same plant. Obviously, if a plant is monoecious but the flowers are dioecious here, so obviously pollen grain from this will go to this. Hai na? So what's the point here? Both the statements, they are correct. Next, endosperm development precedes embryo development. The endosperm of angiosperm plant is, endosperm here will be, yes bache, you will get the PDF in the Telegram group, our official Telegram group of Unacademy NEET English. Right, you have to join that group and you will get the PDF. And again students, once again I'm telling you, you can check any video of mine. See, this is the link for free all India mock test. Do not miss that. Right, earlier we were thinking to keep the mock test on Sunday only, but students, they want more and more practice. That is why till 4th of May you guys will get a chance to write a paper. That paper is as per NCERT bache. You will get a chance to write that paper, and that too free of cost. Okay, so this is your all India mock test. Enroll there, and if you are enrolled, make sure that you don't miss the exam, and your exam is from 2 PM to 5:20 PM. Okay, so that's why we are saying that you guys are going to get NEET ka feel, and you will train your brain in such a way that it has to sit from 2 PM to 5:20 PM for the exams. Okay bache? Done bache? Right? So be the part of this AIMT, and till tomorrow only you will get the subscription just at ₹54999. So right after the NEET examination, I will be starting the classes in my batches. Okay bache? Okay. Now endosperm development is just before the embryo development. So endosperm of angiospermic plant is triploid, right? Next, select match column 1 with column 2. Match column 1 with column 2. Match column 1 with column 2. Single ovule in the ovary. Where are you going to see that? Or pollen grain is covered with mucus in the aquatic plants. So you can take the example of Zostera, right? Because here you will see hydrophily and it is the marine grass. The spelling mistake is there. Safe place for egg laying, such kind of condition you will see in this Amorphophallus also. Flower with foul odor in the Rafflesia. So D with R and single ovule will be there in the A with Q, B with P, C with S, D with R. Second is the correct answer. Then let's move to the next question. Ribbit popper hypothesis, species area relationship, hotspot, global species diversity. Another important topic. Right? Another important topic. Please don't miss it. Right? Right. So, ribbit popper hypothesis, it was given by yes students, it was given by quick. It was given by yes. Okay, so hotspots, C with the normal bear and global species diversity, this. So second is the correct answer. Next question. Next question. Select the incorrect match for the interspecific interaction. Obviously commensalism, it is incorrect, it should be plus and 0. Next, of the incident solar radiation, less than 50% of it is photosynthetically active radiation. Plants capture only 1 to 5% of the PAR. So, answer the question. Yes, students, answer the question here. Answer the question here. So, you know that A is true, but B is false. Plant can, right? A is true, B is false. Right, out of that 50% radiation, plant can use only, right, 2 to 10%, or you can say that 1 to 5% of YR, 2 to 10% of PR. So this reason is incorrect, but yes, assertion is true. Next, genes which code for a pair of contrasting characters are known as, which code for a pair of contrasting traits, which code for a pair of contrasting traits. Quick. Quick. So yes, here the correct answer is going to be alleles, isn't it? Alleles. Select the set of incorrect statement. Yes, bache. Set of incorrect statement. Is the lifespan of fruit flies 2 months? Is it 2 months or 1 month? Is it 2 months or 1 month? Incorrect statement they are asking. Erich von Schirach was one of the rediscoverers of Mendelian work. Yes. Chromosomal theory of inheritance was given by Sutton and Bovary. Yes. But what about the lifespan of fruit fly? What about the lifespan of fruit fly? They are asking incorrect statement, not the correct one. So yes, 1 is the correct statement, because Drosophila is not going to live for 2 months. 2 months means how many weeks? 8 weeks. Of course not possible. Next question. In human being, chromosome 1 has dash genes, Y has dash genes. Again, it is from HGP, Human Genome Project. So how many genes are there in chromosome 1? 2968, and B will be having 231. Next, first stable product of Calvin cycle. First stable product of Calvin cycle is phosphoglycerate. Yes, bache. So primary acceptor is a ketose sugar. Hanji, so what should be the correct answer here? Yes, what should be the correct answer here? First stable product of Calvin cycle is phosphoglycerate. They are saying first stable product of Calvin cycle is phosphoglycerate. Yes, bache, it's true. Right, bache, primary carbon dioxide acceptor is a ketose sugar. You know na, ribulose 1,5 bisphosphate it is. Right, it is ribulose 1,5 bisphosphate. So because it is ribulose 1,5 bisphosphate, so of course it is what? It's a ketose sugar. So definitely both the statements are correct, but reason is not explaining the assertion. The reason is not explaining the assertion. Next, the hormone responsible for apical dominance. The hormone responsible for apical dominance. Which hormone is responsible for apical dominance? That is auxin. Next, according to R.H. Whittaker 5 kingdom classification, the unicellular eukaryotic organisms were placed in. As per that R.H. Whittaker classification, as per R.H. Whittaker, no doubt we study chlorella in plant kingdom, but as per him it should be in protista, because protista they are having what? They are having the unicellular eukaryotes. Okay, they are having the unicellular eukaryotes. So this is the correct answer here. Clear, bache? Next, bache, which of the following is erroneous pair? Hanji, answer this. Yes, bache. Quick. Yes, they are asking erroneous pair. I hope you know that. It means incorrect pair. Incorrect pair. And they are asking it from the examples. They are asking it from the examples. So Sphenopsida, sphagnum, it is incorrect. Do revise it. Next, bache, the given diagram represents the stomatal apparatus in dicots and monocots. Identify A, B and C. Quick. The given diagrams, they represent stomatal apparatus in dicots and monocots. So identify A, B and C. Identify A, B and C. Quick. Obviously the solution is what? See, this is the epidermal cell, B is the subsidiary cells, accessory cells they are, C is the guard cell. Actually bache, yes bache, you can see the arrows are not so proper, but yes, this is the guard cell, and here also this one is the guard cell. So C is the guard cell. So you can cancel these two options. Then your C, okay, this A is guard cell ki surrounding mein epidermal cells, right? This B is the subsidiary cell, so 1 is the correct answer. Okay, next, match the column 1, 2 and 3 and choose the correct combination from the options given below. From the options given below. Yes bache. So are you going to find such, so is it radial arrangement? Okay, so acha, you have to match it. So this one is your conjoint closed, A with 3, okay, B is your conjoint open, and C is your radial. And now you know that radial will be in the root, right? Radial will be in the root. So C with 1 with M. Okay, I got my answer. So A will go with this 3 and will go with this K. Okay, B will go with 2 and then it will go with dicot stem, of course. So that is how it is. That is how it is. So 1 or 2 questions will come from this part. Next bache. Okay, so it is related to estivation. So correct match, Hanji. Correct match, quick. You have to select the correct match. Even in the families video we have revised it. This is the correct match, right? So vexillary is there in the case of beans. Okay, so standard vexillum, wings or LA, keel and carina, which are few. So this one is correct. This one is correct. Next, which of the following is a correct floral formula of a zygomorphic? The word is zygomorphic. Okay, the word is zygomorphic. So if you understand zygomorphic, that is bisexual, you will directly eliminate this option. Then they are saying bisexual. This will be the sign for being bisexual. Then they are saying hypogynous. Hypogynous means your ovary is superior, then it will be like this. Okay, you can cancel this option. Then they are saying okay, 5 united sepals, 5 united sepals, 5 free petals. Dekho, if it will be united means like this, bracket sign will be there. Okay, petals are free. 5 free petals means like this. 5 epipetalous. Epipetalous means the androecium is fused, right? And stamens are free. So this is the correct option here. Third is the correct option here. So you just pray to God that you will get question like this. So if you understand the symbol, then you can answer it. Next, the only type of pollination in which genetically different type of pollen reaches to the stigma. Genetically different type of pollen reaches to the stigma. Xenogamy, that is your cross pollination. Xenogamy, that is your cross pollination. Are you getting my point? Next bache, a file-like rasping organ radula is found in. Very easy. A file-like rasping organ radula is found in. After it, your Wasim sir is about to start the session in next 10 minutes. Yes bache, Wasim sir is about to start the session in next 10 minutes. So tell me. Yes everyone, tell me. What should be the answer here? Obviously it will be right in mollusca na. So out of all, CPI is the example of mollusca, right? Next question, the skeleton of sponges is made up of spicules. Supportive spicules lines the spongiocele. Achha ji, is it so? You know that who's going to line the spongiocele? Who's going to line the spongiocele? Spongiocele, that cavity is lined by choanocytes. The choanoderm will form. The cavity is lined by choanocytes, right? So this is what you guys can see. Okay, this is what you people can see. So right, A is correct, R is false. Next, an example of heterocrine gland is, heterocrine gland माने composite gland, heterocrine gland माने composite gland. Okay, so then it is also known as mixed gland. Okay, so it is pancreas. What's that? It is pancreas. What's that? Pancreas. Now read the following statement. Read the following statement. Quick. Read the following statement. So what do you need to figure out? You need to figure out the incorrect statement. So yes, connective tissue are most abundant and widely distributed in the body. Okay. Connective tissue help in linking and supporting. Okay. Adipose tissue is type of dense connective. No. Right. And connective tissue doesn't secrete its matrix. It is not like that. Okay. So third is the correct answer, right? C and D, they are the incorrect options. C and D, they are the incorrect options, right? C and D, they are the incorrect options. Next, during hibernation and estivation, frog respires through. During hibernation and estivation, frog respire through. Very important it is. Very important it is. Yes, but during hibernation, that is your winter sleep, and in your summer sleep, frog respire through skin. A type of chest pain caused by reduced flow to the heart. Chest pain is the word. Chest pain is the word. It's a hint, bache. Chest pain is the word. It's a hint. So it is angina pectoris. It is angina pectoris. So 70 to 80% of the electrolytes are reabsorbed in which segment? 70 to 80% of the electrolytes, they are reabsorbed in which segment? Of course, it is PCT, proximal convoluted tubule. So tell me the type of epithelium present here. Tell me the type of epithelium present here. Quick. Yes, bache. It's proximal convoluted tubule. So it is brush bordered cuboidal epithelium. Please do not make the mistake here. It is brush bordered cuboidal epithelium, right? What is the meaning of brush bordered? The cells are having microvilli, right? Cells are having microvilli. Clear, bache? Next, the cerebrum wraps around a structure called, very good, again directly from NCERT, and that structure is a major coordinating center. That structure is a major coordinating center for the sensory and motor signaling. So that is thalamus. Next, which one of the following is incorrect regarding neurons? Which one of the following is incorrect regarding neurons? Quick, guys. Which one of the following is incorrect regarding neurons? Axon is a long fiber, the distal end is branched. Yes, bache, it's like this, na? Short fiber which branch repeatedly and project fibers from the cell body also contain nasal granules. Yes, your dendrites are also having nasal granule, very important. Neuron is a microscopic structure having three major parts, true. Cell body and axon, they contain cytoplasm with all cell organelles and no. In the axon, bache, you don't have all the cell organelles. You don't have the nasal granules in the axon. You don't have your mitochondria here in the axon. So this is what you need to remember. So second is the incorrect option. Second is the incorrect option. Clear, bache? So if for some reason vasa differentia in the human reproductive system gets blocked, vasa differentia, so sperms, gametes will not be transported from, vasa differentia is blocked. Yes, bache? Vasa differentia, that muscular duct, read the question carefully. It is not vasa efferentia, it is vasa differentia. It is not vasa efferentia, it is vasa differentia. So it will be epididymis to ejaculatory duct. Remember, testis, testicular lobes, seminiferous tubules, then here it is the retitestis, then vasa efferentia, then epididymis, and then there will be the vas deferens. So obviously from epididymis to ejaculatory duct it will not pass. Okay, it will not pass. Next, match column 1 with column 2. Yes students, match column 1 with column 2. Match column 1 with column 2. Follicular phase, follicular phase, right? Baki menstrual phase, breaking of endometrium without any doubt. Ovulatory phase में release of secondary oocyte, again without any doubt, right? Okay, right? So follicular phase में fully mature Graafian follicle, luteal means secretory. So third is the correct answer. Next, natural method of contraception include, natural method of contraception include, yes bachche, natural method of contraception include, exactly, 1 coitus interruptus and periodic abstinence. Next, this is a method of birth control, this is a method of birth control, again a very simple question, IUDs. Next, which one of the following is not a property of cancerous cell? Not a property of cancerous cells. Obviously they will have excessive mitosis, abnormal mitosis, uncontrolled mitosis, but they do not have the contact inhibition. Contact inhibition is the property of the normal cells, right bache? Next question, match the correct one. Rhinovirus, Haemophilus influenzae, Wicheria bancrofti and Amoeba histolytica, right? So which one is going to affect the lungs? You know that pneumonia. That will be done. Acha, you have to match the correct one. Okay. My bad. So this is incorrect because it is also monogenetic, right bache? So Wicheria bancrofti, female culex will spread it. Okay, so 3 is the correct option. Done. So rhinovirus is not going to affect the lung. Pneumonia will not last for 3 to 7 days only. So of course this is the correct answer, filariasis. Right, your Wicheria bancrofti, it will cause filariasis, elephantiasis, and culex will spread it. Next, wings of pigeon, mosquito and bat show. Wings of pigeon, mosquito and bat, they show. Wings of pigeon, mosquito and bat, you know that their structure is different but their function is same. So it is the convergent evolution. AC is the trick. Convergent evolution means analogous organs. Convergent evolution means analogous organs. Okay? Convergent means analogous organs. Next, in Hardy-Wingber principle, allelic frequency in a population are stable and is constant from generation to generation. Acha. Okay? In Hardy-Wingber principle, the gene pool remains constant. So what is the correct answer? What is the correct answer? In Hardy-Wingber principle, allelic frequency, P plus Q, allelic frequency, they are stable and is constant from generation to generation. We say na, totally condensed. Condensation will start in prophase, but it will be totally condensed in metaphase. Next question, which of the following structure resembles a cart wheel? Hooray. Voice not audible? Huh? Seriously? Hello? Now. Is it fine? No? Okay, 1 minute, 5 minutes. Give me some time. Now is it fine? Now okay? I think it's okay. ठीक है ना? Now fine ना? अरे just wait. The following structure resembles a cart wheel. Okay? Done? Centriole 9 plus 0. It resembles a cartwheel like structure. Okay, so 9 plus 0 means center mein there will be no microtubules, there will be other proteins. Periphery mein you will see A plus B plus C like this. Okay, that is how it is. 9 plus 0, so triplet will be arranged at the periphery. So 9 peripheral triplets and there will be nothing in the center. Okay, okay, next question. Algae have cell wall made up of cellulose, galactans and mannans and minerals like calcium carbonate. In plant cell wall it is having cellulose, pectin and proteins. Obviously. Easy, easy. It's very easy. It's very easy. What should be the answer? It's very easy. What should be the answer? It's very easy. Okay. Am I audible? I'm audible now. Wazeem sir is going to take the class. Don't worry. So genetic material is absent, nuclear membrane absent, mesosome present, circular DNA is also present. Huh? I hope you enjoyed the biology part. Now your Wazeem sir is going to start the session. Do not forget to give the AIMT test. And moreover, bache, you know that tomorrow I will be taking this disorders ka session. And then there is a NEET predictor session where the entire team, okay, see, most predicted NEET paper, that is on 1st of May. Okay, so make sure you like it. Make sure you click on this thing, that is notify me. Okay, chalo, bye. Wazeem sir is about to start. I will share the PDF in the Telegram group. Okay, bye.
Can you hear me, guys? Am I audible to you? Can you all hear me? Can you all hear me? Let me know in the chats quickly, quickly. Can you all hear me? All right. So, people, good evening and what's up? How are you all doing? All good, all good, all good. Someone is saying, sir, wait for 5 minutes break, please. Is that? Do you wanna wait for like 5 minutes? Do you want to wait for 5 minutes? Is that? Let me know in the chats quickly. I need to turn one light off. There are some electricity issues. So I think it should be fine, the dark mode. Yes. So how many questions are you done with in your biology? Can you let me know in the chats? How many questions are you done with in biology? I would want to know in the chats quickly. I would want to know in the chats quickly. Quickly in the chats, people. Just give me a second and we shall be starting, right? There will be no break. There will be no break. Just give me a second and we shall be starting. Okay, so you did not keep count. All right, I believe more than 300 questions. Yeah? Because it's almost been 3 hours. Ambika ma'am was taking the session. So absolutely, I believe it will be more than 3 hours. I mean, it will be more than 300 questions for sure. So should we start? Should we start? Should we start? Should we start, people? Are you all ready for the chemistry? Are you all ready for the chemistry? Quickly. Are you all ready for the chemistry? Quickly in the chats. What happened? Am I perfectly audible, visible? People are saying it's live is cut. Can't you all hear me? Can't you see me? I think I am perfectly audible, visible, yeah? All right. No, I think I am audible, visible properly. There was some glitch basically. I think that's fixed. Yeah, that glitch is fixed. That glitch is fixed. Where is bio session? Bio session you can see now at the end of this session, right? At the end of the session, you can see your bio. Okay? Yes, live is on, live is on, live is on. Live is on, people, live is on. Live is on. Perfect. So let's get going then. Let's get started, yeah? Let's get going, let's get started with the first question of the chemistry. So my dear students, the first question with which I'm starting, I believe you all can kill this easily. Yeah, I believe you all can kill this easily. K2Cr2O7 in acidic medium, right? Your K2Cr2O7 in acidic medium, it gets converted into Cr tri-positive. Remember this. Perfect. When we talk about your KMNO4 in acidic medium gets converted into Mn di-positive. These are the two things which you need to remember over here. Perfect. These are the two things which you need to remember over here. So K2Cr2O7 in acidic medium, it gets converted into Cr tri-positive. I would want you guys to remember this KMNO4 as well. And do remember in acidic medium, it gets converted into Mn di-positive. Very simple. Yeah? Very simple and basic. This was our first question, which is very simpler one. Look at this particular question, people. Carefully have a look on this particular session. Carefully have a look on this particular session. It's again one simple question. On the basis of standard electrode potential of redox couples given below, find out which of the following is the strongest oxidizing agent. Find out which of the following is the strongest oxidizing agent. My dear students, oxidizing agent itself undergoes reduction. You should know it. Oxidizing agent itself undergoes reduction, right? And let me tell you, the one which undergoes reduction easily, that behaves like a strong oxidizing agent. Now, which one undergoes reduction easily? The one which has got more SRP. More the SRP, more the tendency to undergo reduction, better the oxidizing agent, right? Now, if I ask you to have a look on all these values which are given to us, check it out, people. Fe tri positive, Fe di positive. So +3 to +2, decrease in the oxidation state. Decrease in the oxidation state means reduction. So this is your SRP value, right? I2 gives I negative. So 0 to -1. 0 to -1, again decrease in oxidation state. So this is SRP. +2 to 0, decrease in the oxidation state, SRP. +1 to 0, decrease in the oxidation state, again SRP, right? Now, if I ask you to compare all these values, where do you see the SRP value maximum? Where do you see the SRP value maximum? Which one is the maximum SRP value? As I can see, this 0.8, it is the maximum SRP value. It is the maximum SRP value. More the SRP, more the tendency to undergo reduction, better the oxidizing agent. So it is Ag positive over here, which is going to behave like the strong oxidizing agent. I believe it's clear to everyone. Right? A very simple and basic question. We have solved these sort of questions many a times. Moving on to one more basic question. Look at this. Look at this, people. Look at this. The solubility of CuBr is given to us. We just have to calculate its Ksp value. We have to get the value of solubility product. So my dear students, we are starting with what? We are starting with CuBr. And let me tell you the CuBr, how it undergoes dissociation. Cu positive aqueous plus Br negative aqueous. Perfect. If I say the solubility of CuBr is S, that means concentration of Cu positive will be S. This will be also S. Now, if I ask you, how do we write the Ksp expression? Ksp expression is written like this. Copper positive concentration raised by stoichiometric coefficient. This is Br negative concentration raised by stoichiometric coefficient. So I would say Ksp is nothing. This value is S. This value is S. So that makes it S square, right? Let me tell you the value of S. The value of solubility of CuBr is given, right? That is 2 into 10 raised power minus 4. So it is 2 raised into 10 raised power minus 4 raised power 2. The value comes out to be 4 into 10 raised power minus 8. Is there any option as 4 into 10 raised power minus 8? Yes, I can see it's option A, which is going to be the correct answer of this particular question, right? Right, people? Right, people? Is that clear to everyone? I believe this is clear to everyone. Yeah? Perfect, guys. Let's move on to one more question. Look at this particular question. My dear students, all the questions which we have come up with in the today's session, expect 90 plus questions directly or indirectly in the upcoming NEET examination. Trust me on that, right? Trust me on that. Okay, we have got a reaction which is reversible in nature. Now what we are doing exactly, we are increasing the pressure. Now what will happen due to the increase in pressure? My dear students, if you look at the reaction carefully, it is N2 gas plus 3 times H2 gas, right? It gives 2 times NH3 gas. If you look at this particular reaction, on the reactant side, we have got 3 plus 1, 4 gaseous moles. On the product side, we have got 2 gaseous moles. So when the reaction proceeds in the forward direction, I would say gaseous moles are decreasing. If the gaseous moles are decreasing, that means pressure is decreasing in the forward direction. Similarly, in the backward direction, gaseous moles are increasing. And if gaseous moles are increasing, that means pressure will be increasing. Now, what we are doing, as per the question, we are trying to increase the pressure. If we are trying to increase the pressure, equilibrium will try to decrease it. And in which direction pressure is decreasing? In forward direction pressure is decreasing. So I would say with the increase in pressure, equilibrium shifts in the forward direction. I believe this is clear to every one of you. Yeah? Right? Moving on to one more. Look at this particular question carefully, guys. It's again a simple question, and expect these sort of questions to be in your NEET 2024 when it comes to the chapter equilibrium. Look at the question carefully, guys. You are given with a reaction that is 2 times SO3 gas. Let me tell you, it's an equilibrium with 2 times SO2 gas plus what? Plus O2 gas. This is the reaction which we have. Right? Someone is saying why bio is not visible. You can see bio at the end of the session now, right? You can see bio at the end of the session. When the session ends, then only all the sessions will come together, right? Okay. So my dear students, this particular reaction as per the question is carried in a vessel which has got volume of 1 liter, right? Initially at time t is equal to 0 as per the question. We are starting with 1 mole of SO3. We are starting with 1 mole of SO3. Then what happens? Equilibrium is established in the reaction. What happens? Equilibrium is established in the reaction. My dear students, I'm assuming that at equilibrium, out of 1 mole of reactant, let's assume out of 1 mole of reactant, 2x moles got dissociated. So I'll be left with 1 minus 2x. I'll be left with 1 minus 2x moles of reactant at equilibrium. Now we use the stoichiometry. Since 2 moles are giving me 2 moles, so 1 mole will give 1 mole, 2x will give 2x. As simple as that. 2 moles are giving me 1 moles over here. So 1 mole of SO3 gives me 1 by 2 moles. So 2x will give 1 by 2 multiplied by 2x. That comes out be x. So I got the moles of each reactant product present in the container at equilibrium, right? I got the moles of each reactant product present in the container at equilibrium. Now, my dear students, it is given that at equilibrium 0.6 moles of SO2 were formed, right? Number of moles of SO2 at equilibrium. This thing is given to me. How much is it? It is 0.6. Now, if I ask you, number of moles of SO2 at equilibrium, we know it already, that's 2x. So 2x value is basically 0.6. That means x value is your 0.3. X value is your 0.3. Since you got the x value, so 1 minus 2x. 2x is 0.6. So 1 minus 0.6 is 0.4. 2x value, you already know that is 0.6. X value, you got to know that's 0.3. So these are the moles of reactants products in the container at equilibrium. Now, people, if you divide all these terms by the total volume of the vessel in liters, what do I get? I will be getting the concentration of each reactant product at equilibrium. And my dear students, once I get the concentration of each reactant product at equilibrium, I can write the Kc expression. Kc expression is going to be concentration of SO2 at equilibrium raised by stoichiometric coefficient, concentration of O2 at equilibrium raised by stoichiometric coefficient divided by concentration of SO3 at equilibrium raised to the power stoichiometric coefficient. So from here, we can easily get the value of Kc, right? Kc will be concentration of SO2 at equilibrium, that is 0.6, so it is 0.6 ka square multiplied by O2 concentration is 0.3 raised power 1 divided by SO3 concentration is 0.4 whole raised power stoichiometric coefficient 2. Now it is just the matter of calculation and get the value of Kc from here, right? Right, people? Right, people? Is it clear? Is it clear? Is it clear? Is it clear? I believe this is clear to everyone and you can easily solve this sort of equation. Yeah? Perfect. Now, moving on to one more question. Moving on to one more question. All right, look at this particular question, guys, from the chapter, let's see, from the chapter solution. Which of the following 0.1 molar solution will show maximum boiling point? Maximum boiling point. The first solution is the solution of NaCl. The second solution is the solution of copper chloride, right? Third one is magnesium sulfate. Third one is magnesium sulfate. Fourth one is chromium sulfate. Fourth one is chromium sulfate. Fourth one is chromium sulfate, right? My dear students, as far as you would be knowing, delta TB, elevation in boiling point. Elevation in boiling point is directly proportional to number of ions furnished. Number of ions furnished. Number of ions furnished from the electrolyte which undergoes dissociation. Now, if I ask you, this NaCl, it gives Na positive plus Cl negative. So this NaCl is furnishing 2 ions. CuCl2, it gives Cu di positive plus 2 times Cl negative. It's furnishing 3 ions. MgSO4, it gives Mg di positive plus SO4 di negative. It's furnishing 2 ions. Cr2SO4 whole thrice. Cr2SO4 whole thrice. It gives 2 times Cr tri positive plus 3 times SO4 di negative. So 3 plus 2, it gives 5 ions. More the ions furnished by the electrolyte, more is going to be the what? More is going to be the boiling point of the solution. So it's absolutely option D, which is going to be the correct answer of this particular question, right? As simple as that. As simple as that, right? Perfect. Let's move on to one more question. Let's move on to one more question. My dear students, this is one theoretical question which I believe you can easily kill it. I believe you can easily kill this question. Quickly. I believe you can easily kill this question. Which is the correct, what is correct regarding the thermal stability of hydrides of group 15? We are given with the hydrides of group 15 starting with NH3. Then we have got PH3. Then we have got what? ASH3. Right? These are the hydrides of group 15. We have to talk about their thermal stability. Right? My dear students, just remember one thing. Thermal stability is directly proportional to bond dissociation energy. Bond dissociation energy. Okay. So when you move from nitrogen to phosphorus to arsenic, what happens to their size? Size increases. If size increases, bond dissociation energy automatically decreases. And if bond dissociation energy decreases, I would say thermal stability will decrease. So which will be having maximum thermal stability? It's NH3 which will be having maximum thermal stability. So it has to be option 1. That has to be the correct answer of this particular question. I believe this is done and dusted, right? Perfect, people? Perfect, perfect, perfect. All right, if you remember this question I gave you in the last session as well, right? This is important. That's why I have mentioned it again here. And let me see whether you remember this question or not. Let me see whether you remember this question or not. Quickly. Quickly, people. Quickly, people. Everyone. The number of Cl double bond O. The number of Cl double bond O bonds in hypochlorous, chlorous, chloric and perchloric acid will be. What do you think about this one? What do you think about this one? This question you can only solve, my dear students, if you remember the structures of these compounds. And let me tell you, these are the structures of these compounds. This is your hypochlorous, this is chlorous, this is chloric, this is perchloric. There is one bond here, there are two bonds here, there are three bonds here, 3 to 5 on 6. So 6 has to be the correct answer of this particular question. This sort of equation you can only solve. You can only solve if you remember the structures of these compounds. And I would highly suggest you guys to remember the structures of these compounds. Right? Okay, one more similar sort of question. Right? Which you can kill. This is the easy kill question, I call it. Easy kill question. Easy kill question, people. Easy kill question. What is the answer of this question? What is the IUPAC name of it? First of all, you'll be writing potassium. You'll be writing potassium. Then you'll be writing trioxalato, right? Trioxalato. Then you'll be writing cobaltate, right? Its oxidation state here is +3. So it is going to be potassium trioxalato cobaltate 3, right? So it has to be option 4, which has to be the correct answer of this particular question. Yeah? It has to be option 4. Again, this is the question which I gave you in the yesterday's session. I felt this is important. That's why I gave you this question here again. That's why I gave you this question here again. Okay. Now, people, look at this particular one. An organic compound undergoes first order decomposition. If the time taken for 60% decomposition is 540 seconds, find the time required for 90% decomposition. Find the time required for 90% decomposition. Expect this sort of equation as well. Expect this sort of equation as well. Will you be able to solve this? Will you be able to solve this, people? Will you be able to solve this? Yeah? Right? Check it out. Check it out. It's a simple question, guys. As per the question, we have got the organic compound which is undergoing first order decomposition. Let's say this is the first order reaction which we have. I'm assuming its rate constant is K. It's a first order reaction as per the question. Let's assume initially at time t is equal to 0, the initial concentration of this particular reactant is A0. This is 0. This is 0. Initially at time t is equal to 0. Now, as per the equation is concerned, if the time taken for 60% completion, time taken for 60% completion is given to us as 540 seconds, right? It's given to us as 540 seconds. It's given to us as 540 seconds. What do we have to calculate? We have to calculate the time taken to complete 90% of the reaction. 90% of the reaction. Now, my dear students, if you look carefully, I believe you will be remembering this particular result. Time taken to complete X% of the first order reaction is nothing but 2.303 divided by K. It is a log of 100 divided by 100 minus X. I hope you remember this result. Time taken to complete X% of the first order reaction, right? Time taken to complete X% of the first order reaction. Time taken to complete X% of the first order reaction. Do you remember this? What is the value of X here? Quickly. What is the value of X here, people? See, over here, if I use this particular condition, condition, X value here is 60 because 60% of the reaction is getting completed. X value is 60 here, right? So I'll say time taken to complete 60% of the reaction is equal to 2.303 divided by K. It's going to be log of 100 divided by 100 minus 60 comes out to be 40. So it just has to be equal 10 by 4. Well, T 60% is given to me as 540. 540 is nothing but 2.303 divided by K. It is a log of 10 by 4. Log of 10 by 4 will be log of 10 minus log of 4. Now, people, it's just a matter of calculation. I'll say K is equal to 2.303 divided by 540. Log of 10 is nothing but 1. This is 0.6 and 1 minus 0.6 is nothing but 0.4. So you just have to multiply it with 0.4. You'll be getting the rate constant from this particular expression, right? You will be getting the rate constant from this particular expression. Am I clear? Am I clear till here? Yes? Can you let me know in the chats? Am I clear till here? You're right. So from here, you got the rate constant. Right? You got the rate constant. Now, what you'll be doing again? My dear students, again, you have to calculate time taken to complete 90% of the reaction. So what I'll be doing? In this particular expression, I'll take the value of X as 90. So it will be T 90%, which we have to calculate, is equal to 2.303 divided by K value we have already got. That is 2.303 multiplied by 0.4 divided by 540. It is a log of 100 divided by 100 minus X. X here is 90, so 100 minus 90 is 10. So, right? So this becomes log of 10. Log of 10 is 1. So remove this all. So what do I get? 2.303 cancelled. Right? This gets cancelled. It is coming out to be 540 divided by 0.4 seconds. Now just solve this particular term and identify your answer from here. Is it clear? This is a bit lengthy question, but this sort of a question can be asked. It is a bit lengthy question, but it can be asked. Did you get it? People? Perfect. I'm moving on to one more question. So first of all, the answer of this particular question will come out to be 1350 seconds. 1350 seconds. I'm moving on to one more question. One more simple question I'm giving you. Even I have solved this sort of a question. I felt this question again important, so came up with this question again, right? So that everyone would again solve this particular question. My dear students, if you look at this question carefully, the volume of hydrogen liberated at STP by treating 2.4 grams of magnesium with the excess of hydrochloric acid. So as per the question, you are making magnesium to react with HCl. And let me tell you, when magnesium reacts with HCl, it leads to the formation of MgCl2. And with that, H2 gas is liberated. This is your reaction, correct? Now, my dear students, we have got 2.4 grams of magnesium. So if I ask you how many moles of magnesium we have, I'll say given mass of magnesium divided by molar mass of magnesium. So I have got basically 0.1 moles of magnesium, right? I have got 0.1 moles of magnesium. Now, if you look carefully, stoichiometric coefficient here is 1. Stoichiometric coefficient here is also 1. So can I say 1 mole of magnesium, when it reacts with HCl, it leads to the formation of 1 mole of H2. Therefore, 0.1 moles of magnesium upon treatment with HCl will lead to the formation of 0.1 moles of H2. So I got to know that these many moles of H2 are liberated. But am I supposed to calculate moles or volume? I am supposed to calculate volume. So volume of H2 liberated will be equal moles of H2 liberated multiplied by 22.4 liters. The value finally comes out to be 2.24 liters. So I would say 2.24 liters of H2 will be liberated at the end. So the answer of this particular question will come out to be option C. Right? Yes? Perfect. Let's move on to one more question. People. Look at this particular question. I believe you can easily solve this one. This question. Try to solve it. What do you think? The question is, a solution is prepared by adding 2 grams of X to 1 mole of water. So basically, imagine as if you have got a container And in this particular container, we have got 2 grams of X, 2 grams of X, and with 2 grams of X, we have got 1 mole of water. 1 mole of water means 18 grams of water. 18 grams of water. What am I supposed to calculate? I'm supposed to calculate the mass percentage of X. So, how do I calculate the mass percentage? How do I calculate the mass percentage of X here? It will be equal. Mass percentage of X will be equal. Mass of X in grams divided by total mass of the solution, that is 2 plus 18 grams, multiplied by what? Multiplied by 100. Solve it and you're done. Solve it and you're done. Yeah? Correct? Again, one simple and basic question this is. That's all. A lot of people are saying it has to be option 4. And if you're saying it has to be option 4, definitely it will be option 4. So option 4 is the correct answer of this particular question. Right, people? Someone is saying, how can I see the previous bio video? Previous bio video, you can only check, that will be visible to you when the session ends. When the session ends completely, then only your bio session will be visible to you. Yeah? Perfect. I'm moving on, people, to one more question. Look at this particular question. Look at this particular question. Again, the question from the chapter kinetics. Can you solve it? Can you solve it? This is again one very important question, right? You would have solved it many a times, I believe. Perfect. You would have solved it many a times, I believe. A first order reaction. You have got a first order reaction, A gives B. Perfect. Its rate constant is given to me as 10 raised power minus 2 second inverse. 10 raised power minus 2 second inverse. Perfect. Let's say initially at time t is equal to 0, the amount of reactant was 20 grams. Amount of reactant was 20 grams. At what time the amount of reactant left will be 5 grams? At what time the amount of reactant left will be 5 grams? At what time amount of reactant left will be 5 grams? So I can use the integrated rate equation. I can use the integrated rate equation. I can say T is equal to 2.303 divided by K. K is nothing but 10 raised power minus 2. It is going to be log of A naught divided by 80, right? What is A naught and what is 80? A naught here is 20, 80 here is 5. So 5, 1, 5, 4. So it finally comes out to be log of 4. Log of 4 is nothing but 0.6. So finally, it's a matter of calculation. It is 2.303 multiplied by 0.6 multiplied by 10 raised power 2. Solve this and get the value of time from here. You'll be getting the time in seconds. Again, one basic question this is. Again, one basic question this is. I believe you can solve this sort of a question if it comes. Okay. I'm moving on to one more question. Let me see if you can solve this one. You should be in a position to solve this one, right? You should be in a position to solve this one, people. Quickly. What do you think about this one? When an electron is excited from ground state to what? To fifth orbit. From ground state to fifth orbit. The number of spectral lines obtained in Bohr's spectrum of hydrogen. Now, if I ask you how do I calculate the maximum spectral lines obtained? The maximum spectral lines obtained is calculated by the formula N2 minus N1, and this has to be N2 minus N1 plus 1 divided by 2. I believe you already have the value of N1 and N2. Now, N2 here is 5, N1 is 1. So this is 5 minus 1 plus 1 divided by 2. So minus 1 plus 1 cancel. 5 4s are 20. So 20 divided by 2 comes out to be 10. So I would say in total 10 spectral lines will be obtained when the electron will be making a transition from, right? When the electron will be transitioning between 1 to 5. I hope I'm clear to everyone. And this sort of equation, right, it is frequently asked. So it will be asked again. Even this sort of equation, have an eye on this as well. It is just, I want you guys to remember one thing. See, you have got Lyman series, you have got Balmer series, you have got Lyman, Balmer, you have got Paschen, you have got bracket, you have got fund. You have got bracket, you have got fund. Right? These are the series which we have. I'm not talking about Humphrey. Just remember one thing. This is U, this is V, this is I, this is I, this is I. This stands for ultraviolet. Perfect. This stands for visible spectrum. This stands for infrared. This stands for infrared. Or you can say this stands for far infrared. Infrared only basically. Right? Accordingly, you can match. Whatever is asked, you can match it. In this particular question, I was supposed to check which of the following series for transitions in the spectrum of hydrogen atom falls in visible spectrum. So it is Balmer which will be falling in the visible spectrum. And let me tell you, this is the asked question. This is the asked question. Let me tell you that. This is the asked question. Okay, you should be knowing it. You should be knowing it. One more question, guys, which I liked, right? Which I solved in the last session as well. I would want to solve this question again because this sort of a question can be asked, right? This I found important. This I found important. Can you give it a try? The question is, you are given with a reaction. The reaction is A plus B gives products. A plus B gives products. As per the question, order with respect to A as well as B is 1. Order with respect to A as well as B is 1. Order with respect to A as well as B is 1. Right? So if I write the rate law is going to be R is equal to K, concentration of A raised power 1, concentration of B raised power 1. This is your rate law. Perfect. Now people, in this rate law, if you fit the data of experiment number 1, experiment number 1 says that rate of the reaction is 0.1 is equal to K. Concentration of A is how much? It is 20 raised power 1. Concentration of B is how much? It is 0.5 raised power 1. This one equation which I got. Fit the second experiment data into this rate law. So this has to be 0.4 is equal to K. Concentration of A. Concentration of A is X raised power 1. Concentration of B is 0.5 raised power 1. This equation number 2. Fit the third experiment data. This is 0.8 is equal to K. Concentration of A is 40 here, raised power 1. Concentration of B is Y here, raised power 1. So how many equations I got? I got 3 equations. Now, my dear students, from these 3 equations, you have to calculate the desired quantities, right? So what do? So first thing, if I divide equation 2 with equation 1. If I divide equation 2 with equation 1, 0.4 divided by 0.1 comes out to be 4 only. K and K gets cancelled. 0.5 cancelled. So this will be X divided by 20. So from this particular option, X value will come out to be 80, right? So X is gone. X is over. We have calculated X, right? Now it is the time to calculate Y. It is the time to calculate Y. And when you divide other 2 equations, the value of Y over here, you'll be getting exactly as 2. So it's option A, which is going to be the correct answer of this particular question, right? Which is going to be the correct answer of this particular question. I am moving on to one more. My dear students, look at this particular question from the chapter Thermochemistry. Look at this particular question from the chapter Thermochemistry. Enthalpies of formation of CCl4, H2O, carbon dioxide and HCl, they are given to me. The magnitude of enthalpy of the reaction given below. The magnitude of enthalpy of the reaction given below. This is the balanced reaction that's given to me. You should be knowing standard enthalpy of reaction will be equal. How do we calculate standard enthalpy of reaction? In terms of standard enthalpies of formations of reactants and products. So let me tell you, enthalpy of reaction is nothing but enthalpy of formation of products minus enthalpy of formation of reactants. So I'll start with the product. Its stoichiometric coefficient is 1. It is 1 multiplied by standard enthalpy of formation of what? Carbon dioxide plus coefficient here is 4 multiplied by standard enthalpy of formation of what? It is HCl. So I wrote the standard enthalpy of formation of products minus what is the standard enthalpy of formation of reactants? It's 1 multiplied by standard enthalpy of formation of what? CCl4. Perfect. Plus this is 2 times standard enthalpy of formation of what? Standard enthalpy of formation of H2O. I believe all these parameters are given to you as per the question is concerned. Check it out, people. Check it out, people. What is the standard enthalpy of formation of carbon dioxide? It has to be given to me. It is minus 349. Minus 349 plus 4 multiplied by standard enthalpy of formation of HCl. Formation of HCl is given to us as minus 92. So this minus 92. Perfect. Minus 92 minus standard enthalpy of formation of CCl4. How much is that? That is minus 105. Minus 105 plus it's going to be 2 times standard enthalpy of formation of water. That's also given to me. And how much is that? That is minus 242. Can I say it's again the matter of calculation only and you are done? Perfect. But be careful with one thing. Be careful with one thing. You are asked about the magnitude of enthalpy of reaction. If the answer from here comes out to be negative, you are not supposed to write that negative sign. Because the question itself is saying that you have to calculate the magnitude of enthalpy of reaction, nothing else. I believe I'm clear. I believe I'm clear, yeah? Somebody is saying, where is the bio session? Bio session you can see now once this complete stream is over, right? Once your physics session is also over after this, then only you can see the bio session. Yeah? Okay? because we are taking the session from different places. I'm taking the session from Kashmir. Ambika ma'am was taking the session from Chandigarh. Sorry, from Noida. Perfect. We are not in the same studio. That's why it happens. Okay. Perfecto. I'm moving on, people, to a few more questions. Let's see. Let's see what all other questions I have in the box. Let's see what all other questions I have in the box. This sort of a question, many a times you would have killed. I believe that. Right? So the correct increasing order of ionic radius. Okay. How exactly you're going to solve this? You know, in case of isoelectronic species, in case of isoelectronic species, how do you check the size? You just check the number of protons. More the protons, lesser the size. In simpler words, I can say it like this. More the protons, lesser the size. Or you can say more the atomic number, lesser the size. Okay, more the atomic number, lesser the size. All these here, they are isoelectronic species. They have got same number of electrons. 18 electrons here, 18 electrons here. All of them are having 18 electrons, right? Now, talk about their atomic number. Atomic number of calcium, you know, that is 20. Potassium, it's 19. Chlorine, it's 17. Sulfur, it is 16, right? This is the atomic number, I must say. More the atomic number, lesser the size. More the atomic number, lesser the size, right? More the atomic number, lesser the size. So I believe it's option C only, which is the correct answer of this particular question as well. So I expect this sort of a question as well. Perfect. Perfect, people. I am moving on to one more. Look at this particular question. The species having maximum number of lone pairs of electron on the central atom. Now the point is, how do we check the lone pairs on the central atom? If I take any one, let's say I'm taking SF4. Let's say I'm taking SF4 into consideration first. Sulfur here is the central atom, right? Sulfur here is the central atom. So if I ask you how many valence electrons are with sulfur, there are 6 valence electrons with sulfur. How many surrounding atoms it has? It has 4 surrounding atoms. Number of surrounding atoms is equal to number of sigma bonds. Number of surrounding atoms is equal to number of sigma bonds. So I must say sulfur had 6 valence electrons and it has utilized 4 of the valence electrons to form 4 sigma bonds. So there'll be 2 electrons left as such on sulfur in the form of a lone pair. So there is 1 lone pair on sulfur. If I talk about XCF4, if you calculate it, you'll find there'll be 2 lone pairs on xenon. Perfect. There'll be 2 lone pairs on xenon. If I talk about ClO3 negative, when you calculate it, when you check it, there'll be 1 lone pair on chlorine. If you talk about I3 negative can be written as I2 negative. What you'll observe when you check it out, you'll find 3 lone pairs on the central iodine. So my dear students, what is the answer of this particular question, right? It's going to be this one, which is going to be the correct answer of the question. I believe it's clear. So it was a very simple basic question, which you already killed. Now, expect this sort of a question for sure. I'm not pretty much sure whether you have solved this sort of a question or not, right? If you have not, have an eye, have a look how exactly I'm going to solve this sort of a question. The molar solubility of CaF2. First of all, how CaF2 undergoes dissociation? It will be Ca di positive plus 2 times F negative aqueous, right? This is your CaF2. Perfect. Let's say its solubility is S. This will be S. This will be 2S because the stoichiometric coefficient here is 2. Perfect. Now this CaF2 is being introduced in NaF. Now, NaF is a strong electrolyte. It would have undergone dissociation.
Let me know in the chats if I am perfectly audible and visible to each and every one. Okay. Just a second. Guys, let me know in the chats if I'm perfectly audible and visible to each and every one. Okay. Okay, great. Just a second. We are starting. Okay, just let me fix everything and we'll start. Okay, okay. Just a second, people. Okay, let me fix this over here and we will start. So actually, we have an electricity issue over here. That's the reason Wasim sir couldn't continue, but he will come at the end. Wasim sir will come at the end. Okay. So till then, I will be taking the session and then Wasim sir will continue. Okay. Okay, great, 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 great. Now, guys, let's start this session. I want everybody of you to let me know in the chats if I am perfectly audible and visible to each and everyone. Okay, because we have to kill rest of the questions over here. Okay. And if Wasim sir is watching, I ask Wasim sir, please tell everybody in the Telegram that I have joined. So I will be starting the physics portion. So after me, Wasim sir will come and he will continue. Okay, because he's having some inverter problems. Okay, in his studio. So let's start our session over here. Let's take a look at the first question that we have exactly on the screen. So what is mentioned over here is that we have two equally charged identical metal spheres. Let me make it over here. Everybody out there, listen to me very, very, very carefully. We have got two metal spheres over here. Let's suppose this is the first metal sphere that I am making over here. Okay, this is the first metal sphere. Let's suppose this is the first metal sphere. Okay, and we have got one more metal sphere. In the question it says that you have got two metal spheres. This is the first metal sphere and this is the second metal sphere. It says this is A and this is, we say, B. Charge on this is Q, charge on this is Q, and separation between this and this is, we say, let's suppose, R. Now, my dear friends, can we say in this particular case how much is the force between the two? We can clearly say, sir, force is K Q1 Q2 divided by R square. Okay, so we can exactly say over here force is K Q1 Q2 by R square, and instead of Q1 and Q2, I'll write over here this is K Q square divided by, we say, R square. Okay, this is the force between these two. Now what does the question actually say over here? What does the question actually say over here? We say two equally identical metal spheres A and B repel each other with a force F. Okay, and then it says another uncharged sphere C is touched with A. It says that we are bringing one more uncharged sphere. Okay, that is this C, and it says we are touching this C with A. Okay, and then placed midway between A and B. When this uncharged sphere C, is touched with A, then it is placed between midway of A and B. Let me show you over here. See, we say this will be A over here and this will be B over here. Okay, this is B and this is A. We are placing this midway. My dear friends, initially charge on A was Q. Now we say, once we touch this uncharged sphere with this A, now charge will be shared. Q by 2 is on this one. Q by 2 is on this one. Because this A sphere will actually share the charge. Okay, it will share the charge with C. So charge will be shared. Okay, now distance between these two is R by 2, and this distance is also R by 2. What we are supposed to find out in this particular question is that we have to find how much is the force on C, how much is the net force on C. Everybody out there, listen to me very carefully. See. My dear friends, charge on B is already, we know it is Q. Now listen to me very carefully. We say over here, if we talk about this sphere C, guys, see, we know, sir, this charge, this A sphere will repel this sphere C in this direction. Okay, so I say force on this C will be simply, I say this force will be force on C due to A. So A sphere will repel this sphere C with FCA. Similarly, on this sphere C, this B will apply force. I say in this direction, that is FCB. Okay. Now let's calculate FCA first. If I just try to find out FCA, FCA is the force between, we say sphere C and sphere A. So can I say this will be K, Q1. What is the value of Q1? That is Q by 2. What is the value of Q2? That is also Q by 2, because it is between A and C. Divided by, what is the distance between these two? That is R by 2 whole square. Okay. Now, if you solve it, my dear friends, it will come out to be, that is K Q square divided by 4. And then we have R square by 4, and 4 will cancel. So it will be K Q square divided by 4. Okay. Sorry, divided by R square. And simply we call this as F. Why? Because we know KQ square divided by R square is F. Okay. Now this force, we got it. Let's find the force FCB. We say, sir, force FCB will be simply, that is the force between sphere C and force between sphere B over here. So that will be K Q1. What is the value of Q1 over here? That is Q by 2. So I can write over here, this is Q by 2. Okay. And what is the value of this Q2 over here? That is, we say, sir, Q over here. Okay. Divided by, how much is the distance between these two spheres? That is R square divided by 4, because R by 2 whole square. Okay. Now this will be K Q square divided by 2 into R square by 4. 2 ones, 2 twos. So this will become 2F. This will become 2F. As simple as that. This will become 2F. This will become 2F. Okay. Okay. Yes, sir. Now, my dear friends, listen to me very carefully. Listen to me very carefully over here. Listen to me very carefully. See, now we say force on this sphere C. Force on this sphere C. I can say this is the sphere C over here. Force in this direction is, we say, sir, F. Okay. Why? Why? Because FCA is acting in this direction, and FCA we got it as F. And force in this direction is, we say, sir, 2F. Why? Because FCB we got as 2F. Okay. Now, how much will be the net force in this case? We say this 2F minus F. That is, we say, sir, F towards sphere. F towards sphere A. F towards sphere A. So answer option 1 is the correct option. Let me know in the chats, everybody out there, is this question crystalline clear? Is this question crystalline clear? Everybody out there, let me know in the chats if this question is crystalline clear. All the people out there. Everybody, guys, let me know in the chats, is this crystalline clear? Is this crystalline clear? Okay. Everyone means everyone. Then we will move on to the next question. Okay. Okay. Great. We will be sharing the PDF in our Telegram channel. I want you guys to join that as soon as possible. Okay. We will be sharing the PDF in our Telegram channel. I want you guys to join the Telegram channel as soon as possible. Okay. That is t.me Unacademy NEET English. Now we have got the second question over here, which is a very good question that's mentioned on the screen. Everybody over here, look at the screen. Look at the screen. You will get to know. Now see. We have over here two small spheres, each having charge Q, are suspended by an insulated thread. We have got, this is a charge Q and this is a sphere, another sphere. Okay. So each of having charge Q, this is charge Q, this is also having the charge Q. Okay. Are suspended by an insulated thread of length L. This length is exactly L. From the hook, the arrangement is taken to a space where there is no gravitational force. Guys, see. If there is no gravitational force, mg means the force which acts on this in the downward direction, that force is 0. Can I say they will elongate and they will behave, it will look something like this. This will be the sphere and this will be the sphere. Guys, listen to me very carefully. I am saying that in the downward direction, what is the force acting on the charge? That is mg. And in this direction, what is the force? That is electrostatic force. We say on this also, we say electrostatic force will act and mg acts in the downward direction. Tell me one thing. If I say this mg downward force is 0, so can I say they will elongate and it will look something like this afterwards? Yes. Now on this charge, force electrostatic will act in this direction. Force electrostatic will act in this direction. This length is L, this length is L. Now guys, can I say this is we say string over here. This is we say string over here. And in the string you have got tension. In the string you have got tension force. Same is the case in this one. So we say here this will be the tension. This will be the tension. Now tell me one thing. Tell me one thing in this case. In the question it is asked that the angle between the two threads. You guys are supposed to find the angle between the two threads and tension in the string. Now see guys, we say the force, this charge sphere will remain at equilibrium over here. Can I say in this particular case, sir, tension will be simply electrostatic force? And we can say, sir, what will be the value of electrostatic force? I can say that is K Q1. That is charge on this is Q, so K Q square divided by distance between these two. That is, we say, sir, 2L whole square. 2L whole square. So can I say this will be K Q square divided by, this will be 4L square, 4L square, KQ square divided by 4L square. This will be the tension force. And it has asked what is the angle between the two strings. Guys, how much is this angle? This is 180 degrees. So angle between the two strings is 180 degrees. Tension in this case is how much? KQ square divided by 4L square. And we say angle in this case is, we say 180 degrees. Angle in this case is 180 degrees. Okay. Next question, not this one. Okay, this one. This is an important question. We say, sir, this is important. We say this is important. I want you guys to listen to me very, very, very carefully over here. Let's suppose you have got these charges, okay? This is plus Q, plus Q, plus Q. And this is till infinity. You guys are supposed to find the net electric field at the origin because of all of these charges. See, how do I solve this particular question? We say, sir, net electric field at the origin will be, it will be, because of the first charge, it is KQ divided by, because of the first charge, it is KQ divided by, we say 1 square. Why? Because first charge is placed at 1 meters. Then we say KQ divided by 2 square. Then we say KQ divided by 4 square. Okay. This is because of this charge. Okay. And I'll directly write over here. This will be simply KQ divided by 2 square is 4. 4 square is 16 plus KQ divided by 8 square is, we say, 64 and so on. So we said this is the net electric field because of all of these charges at the origin. Now, if I ask you, what can you take common over here? You say, sir, I can take KQ common. What remains over here? This is 1 plus 1 by 4 plus 1 by 16 plus 1 by 64 plus so on. And I have told you so many times in my classes that this is what we call the GP series. This is what we call the GP series. And in order to find the sum of all of this, we have a formula. S is equal to A upon 1 minus R. So A in this case is first term, that is 1 upon 1 minus. What is R? R means common ratio. How to find common ratio? Take any term. Let's suppose I take 1 by 16. Divide it with the term which comes before it. That is 1 by 4. 4 ones are 4 fours are. So common ratio is 1 by 4. So it is 1 minus 1 by 4. So sum in this case will be 1 upon 4 into 1 is 4 minus 1 is 3 by 4. That is 4 by 3. 4 by 3. Okay. Now, we say instead of all of this, you can put 4 by 3. So we can write over here, net electric field is KQ into, we say, sir, 4 by 3. KQ 4 by 3. And further also you can write it, guys. We know what is the value of K. K is equal to 1 by 4 pi epsilon naught. Okay. Then we say Q into 4 by 3. Now this 4 and this 4 will cancel out over here. So we can further write it is Q upon 3 pi epsilon naught. Q upon 3 pi epsilon naught. Is this clear? Let's move on to the next question. Next question we have over here. We have got a ring in this particular case. Okay, this is a charged ring. Okay. And my dear friends, in this case, we say, you guys are supposed to find the horizontal and vertical component of the electric field. Horizontal and vertical component of the electric field. Listen to me very carefully. Guys, this is a cord. I have told you so many times, that electric field because of the cord at this point is how much? That is root 2k lambda by r. Root 2k lambda by r. Okay. Now tell me one thing. Have I told you this before? If I say, let's suppose this is a root 2k lambda by r, we are supposed to find its horizontal component and vertical component. Guys, tell me one thing. Can we use this logic? If we have one force over here, F, if we have another force over here, F, okay, we say a resultant in this case will be under root of 2F. Resultant in this case will be under root of 2F. Because whenever the angle between the two vectors is 90, we say in that case resultant is 2F. Now tell me here, we'll use the same logic. This is under root of k, root 2k lambda by r. Can I say its horizontal component will be k lambda by r and this will be also k lambda by r. So this will be its horizontal component and this will be its vertical component. Okay. And let me know in the chats if this is crystalline clear. Let me know in the chats if this is crystalline clear. Everybody out there. Everybody out there. Now, this is, we say, sir, semicircular ring over here. Half of this contains positive charge, half of this contains negative charge. I ask you to find the net electric field at this point. How can you find? Guys, because of this ring, see, you can divide this into two chords. Guys, see, because of this part of the ring, electric field will be towards the ring. Okay, guys, this because of this chord. We know because of the chord, electric field is how much? We say this is under root 2k lambda by r. Okay. And why am I showing the direction towards the chord? Because it is having the negative charge. Okay. Now you have got positively charged chord over here. Because of the positively charged chord, we say electric field is away from the chord. And this will be also under root 2k lambda divided by r. Okay. Because of this chord, we say electric field is under root 2k lambda by r away from the charge. Because of this chord, it is root 2k lambda by r. Now guys, tell me one thing. Don't you think that they will be having the resultant something like this? Everybody tell me in the chats, is this crystalline clear? Is this crystalline clear over here? Okay. Now here, angle between these two is 90 degrees. Remember this? So we say, guys, if we have got one vector in this direction, if you have got another vector in this direction, so angle is 90 degrees. How much is the resultant? Can I say resultant in this case will be under root of whatever is this mentioned over here, that is root 2k lambda by r. Okay. So we can simply say that is 2k lambda by r. And direction is towards the right. Okay. I hope this one is clear. Next question. Okay. This is an important question. I will mark over here. This is an important question. Okay. Everybody. Guys, see. I have got over here a line charge, infinitely long charged rod. Okay. I have got an infinitely long charged rod. And there is a charge placed over here. You can clearly see. Charge is minus q. Okay. Charge is given as minus q. Charge in this case is given as minus q. Okay. And I am given some velocity to this particular charge over here. Okay. Now, my dear friends, this charge will start perform the circular motion. My question to you is, it is mentioned, I give velocity v to the charged particle at r distance from this line, from this infinitely charged wire. It will follow the circular paths. Okay. We are supposed to find how much velocity should we give to it so that it moves in a complete circle. Okay. Now guys, see. Here we use one concept. If a particle performs circular motion, like this negative charge is performing circular motion, we say centripetal force will act over here. Sir, centripetal force will act. Okay. I'll write FC. We say FC is the centripetal force that will act. And that centripetal force will be equals to electrostatic force. Guys, if you have got a charge over here, we say on this charge, this rod will attract, this wire will attract it towards itself because rod has got negative charge. So we say that will be equal to electrostatic force. Keep this thing in your mind. When it comes to the centripetal force, we can write it simply. This is mv square divided by r. M is the mass of charge, v is the velocity, r is the radius, is equal. And electrostatic force will be simply q times of E. And here, guys, see, here electric field will be produced because of this wire, infinitely long wire. So can I say in this particular case, sir, because of infinitely long wire, electric field is simply that is 2k lambda by r. If you have studied this, if we have got an infinitely long charged wire, because of that electric field is 2k lambda by r. Okay, so I can write over here, this is mv square divided by r. It will be equals to q into 2k lambda divided by r. R and r will cancel. R and r will cancel. So v square will be simply 2kq lambda divided by m. So v I can find over here. Now take the root. So it will be under root of, we say, sir, 2k lambda q divided by, we say, m over here. This will be the velocity of that charge. Is this crystalline clear? This will be the velocity of that charge. Is this crystalline clear? Let me know in the chats, everybody out there. Guys, still over here, is this crystalline clear? All the people out there, let me know in the chats. Guys, just a second. There is some electric. It is heavily raining over here. That's the reason we are getting the electricity issue over. Okay. Guys, is it clear? Is it clear now? Okay. Now see. It is heavily raining over here in Kashmir. That's the reason we are getting this connectivity issue. Okay. Okay, great. Now see, in the question it is mentioned that, okay, in the question it is mentioned that, we have got a sphere over here. Okay. Not a sphere, this is a cylinder. It says that I'll write over here, if flux through the curved surface, is phi, if flux through this curved surface is phi, then find flux through plane surface. Then you guys are supposed to find the flux through plane surface. This is the question. Now listen to me very carefully. Over here. Guys, we say here, if I write flux through a closed surface, we say, sir, that is q divided by epsilon naught. Okay. And when it comes to the closed surface over here, closed surface means cylinder. Cylinder is made up of two plane surfaces, this one, and then you have got the curved surface. So that will be flux through We say two plane surfaces plus flux through one curved surface. Okay. Now, can I write over here, this is Q divided by epsilon naught will be flux through two plane surfaces plus flux through one curved surface. Guys, flux through the curved surface is given as phi naught. Okay, this is phi naught. So I'll write over here, this is simply phi naught. So can I write, sir, flux through two plane surfaces will be simply this flux through two plane surfaces. It will be Q divided by epsilon naught minus, we say, phi naught. So now we can say flux through one plane surface, it will be 1/2 of, we say, Q divided by epsilon naught minus, we say, phi naught. So this will be the flux through one plane surface. Plane surface means this plane surface. Okay. The other one is the curved surface. Okay. I hope this is clear. Okay. Cool. Let's talk about this question. This is an important question. I want you guys to keep your mind over here. See, in this particular question, what is given is that, listen to me very carefully. It says that a charge plus Q in an electric field of charge plus Q, you have got this plus Q charge over here. Okay. Another charge is carried from A to B. Okay, in this direction. Then it is taken from A to C in this direction. And then it is taken from A to D in this direction. And then it is taken from A to E, something like this. We are supposed to find the work done. Work done will be minimum along path A. Let's first find work done. There is a formula for work, and that is Q delta V. Okay. Now, guys, listen to me very carefully. We say work done in this case will be simply work done in this case, or don't write Q delta V, write work done is equal to change in potential energy. This I have told you in the marathons. When it comes to the work done, it is simply change in potential energy. That is U final minus U initial. Let's suppose when we took the charge from A point to B point. Okay. We say charge is over here and finally charge is over here. When initially charge is at this point, can you tell me how much is the potential energy? You say, sir, that will be finally when it is at over here, you say it is K Q1. Q1 is Q, Q2 is Q square divided by distance between these two is R. So this is R minus. When charge was initially over here, we say it will be also K Q square divided by R. So work done will be 0. Similarly, if you find the work done along this path, that will also come out to be 0. Work done along this path, that will also come out to be 0. So we say option D is the correct option in this case. That is 0 along each path. Okay. 0 along each path. Okay. Next. Let's move on to, we say, this particular question we have got over here. Let's move on to this particular question. Not this one. Yeah, this one. Okay. Now, guys, in this particular case, we are given basically two condensers. Two condensers means two capacitors over here. Okay. We are given two condensers means two capacitors over here. Two condensers means two capacitors. Okay. One is capacity having capacity C, another is capacity having capacitance C by 2. Okay. And in this case, the battery V is connected over here. What we are supposed to calculate in this particular case, the work done in charging both the condensers will be, both the capacitors will be in this particular case. Now, guys, see. You have got one capacitor over here, that is C. Another is, we say, C by 2. We are supposed to find how much should be the work done in charging both the capacitors. Guys, remember one thing. Remember one thing. The amount of work done this battery will do over here, that work done will be only stored as energy of the capacitors. Okay. Now, can you tell me how much is the equivalent capacitance of these two? Sir, they are in, we say, parallel. We say C equivalent in this case will be simply, that is when the two capacitors are in parallel, we say C plus C by 2, C plus C by 2. So that will be simply, that is 3C divided by 2. Okay. This will be the equivalent capacitance. Now, how to find the work done? Sir, we know work done will be stored as energy of the capacitor, and that is 1 by 2 C equivalent into V square. So that is 1 by 2. What is the value of C equivalent in this case? Guys, you have got the two capacitors. Their equivalent capacitances, we say 3C by 2. So I'll write over here that is 3C divided by 2 into V square. So this will be simply 3C V square divided by 4. 1 by 4 CV square. Oh, sorry, 3 by 4 CV square. This is the amount of energy. This is the amount of work done in charging the capacitors. Okay. This was a question which was asked in NEET 2017. A battery is charged by a capacitor. A battery is charged by a battery. The battery is removed with an identical uncharged capacitor. Okay, let me show you what does the question say. See. I'll tell you. We have got a capacitor over here. Okay. And this capacitor is charged by a battery. Okay. Which is having the potential difference V. Now, how much will be the energy stored in the capacitor? U is equal to 1 by 2 CV square. Okay. Now we are taking this capacitor. Okay, we are taking this capacitor and we are connecting it with an uncharged identical capacitor. Okay, remove this battery, then connect the capacitor with an uncharged identical capacitor. Now guys, see, they will share the charge Q by 2 and Q by 2. If I say Q was the charge initially on this one, if I say Q was the charge initially on this one, finally they will share the charge between the charged and uncharged capacitor. Okay. Now we are supposed to find how much is the final energy in this particular case. Capacitance of this is C, this is C. Okay. If they share the charge Q by 2 and Q by 2, their potential will be also shared V by 2 and V by 2 over here. So we say finally energy in this case will be U final, I can write 1 by 2. 1 by 2. Guys, finally energy. C and C are in parallel. So C plus C is 2C. I'll write over here 1 by 2 into 2C. Okay, because 1 by 2 CV square. C equivalent is 2C. V square. How much is the potential difference in this particular case? Sir, potential difference across both is V by 2. So it will be V by 2 whole square. So can I write it something like this? Can I write it something like this? It is simply this and this will cancel. So this is CV square divided by 4. CV square by 4. And further we can write it, sir, 1/2 of 1 by 2 CV square. So 1 by 2 CV square is what? 1 by 2 CV square is what? That is initial energy initially. So I can say this will be simply 1/2 of, this will be simply 1/2 of, that is UI. Okay, this is UF. So final energy will decrease by the factor of 2, will decrease by the factor of 2. Okay. Is this clear and crystal till over here? Let me know in the chats, everybody out there. Okay. Okay, one more type of question we have got from the capacitors over here. Guys, see. If you ever see, listen to me very carefully. If you ever see, let's suppose we have got a point over here, point P, and we have got point Q over here. Okay. You have to find the equivalent capacitance between P and Q. Mark this point. See, if this is P, this will be also P. They will be having the same potential. If the wire is directly connected, this is P. This point will be also having the P potential. Okay. Now, this is Q point. We say this point does not have the potential Q. Why? Because capacitor came in between, this capacitor. Mark this point as X. So we say this will be also X and this will be also X. Now, how many points are in this entire branch over here? You say, sir, 3. Which one? P, X and Q. So mark it over here. P, X and Q. Now, if you take a look over here, First capacitor is between P and X, so let me make it over here. Second capacitor is between X and P, so it will be also between P and X. It's one and the same thing. Third capacitor is also between P and X. Third capacitor is also between P and X. Fourth and fifth capacitor are between X and X. So these two will short circuit over here. Short circuit. Okay, you can remove. Next is between X and Q. Next is between X and Q. This is C, 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 C. Now tell me how to find the equivalent capacitance. Whenever the capacitors are connected in parallel, their equivalent capacitance will be C1 plus C2 plus C3. So their net is 3C over here. And then you have got one more capacitor, that is C. Now here, how to find C equivalent? C equivalent will be simply, if the two capacitors are in series. So this will be 3C into C upon 3C plus C. 3C plus C. C1, C2 upon C1 plus C2. As simple as that. As simple as that. Okay. So we say this will be simply over here, sir, that is 3C square divided by 4C. C and square will get cancelled out over here. That is 3C divided by 4. 3C by 4 is the answer in this particular case. Okay. Great, 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 great. Now, let's move on to this particular question we have got over here. Let's move on to this question. Find the ratio of the resistance of A and B. Guys, listen to me very carefully. You have got VI graph over here. You have got VI graph over here. Remember one thing always. Remember one thing always. Whenever you see the VI graph, listen to me very carefully. I'll write over here. In case of a VI graph, we say slope is directly proportional to the resistance. If it was IV graph, then it was slope is inversely proportional to the resistance in that case. Okay. Now guys, tell me one thing. See, I say this A line is making 30 degrees over here. We say this angle will be 60 degrees. So this wire A is making 60 degrees with the x-axis. So tell me, if I write over here slope of A divided by, I say, slope of B, that will be what? Slope means tan theta. So over here, I'll write tan of 60 degree because A wire is making 60 degrees divided by, we say, tan of 30 degrees because another is making 30 degrees. So tan 60 is how much? I guess it is root 3. Okay. And tan 30 is 1 by root 3. Please do check it on your own, if I'm not making any mistake. So I say this will be simply a root 3 into, we say, root 3. That will become 3 by 1. So answer in this case is 3:1. Okay. Answer in this case is 3:1. Is it correct? Let me know in the chats, everybody out there. Okay, next. Okay, now you have got this. In this case, you are supposed to find VA minus VB. This is how it actually looks like. You have got point A, you have got point B over here. You are supposed to find VA minus VB, potential difference. Guys, see, I have told you always, whenever, if I say the direction of the current is this, means this will be positive terminal, this will be negative. This is positive and this is negative. Why? Because current always flow from higher terminal to the negative terminal, higher to lower. So in this particular case, listen to me very carefully. This is a good question actually. Here you have got 1 ampere current in this direction. In the lower direction, you have got 3 ampere current. Don't you think that, sir, 4 ampere will be coming from this side? That only is getting divided. Yes. So 4 ampere is here also. Now see. 2 ampere is coming from this side. So here it will be 2 ampere. So means 2 ampere would have been coming from this side. See over here. This 2 ampere and this 2 ampere are joining together. So 4 is coming out. Okay. Now guys, see one thing. So this is positive, this is negative. This is positive and negative. And here this is positive and this is negative. In order to solve this type of question, I have told you, you guys are supposed to select the path. So select this path from A to B. Okay. I'll write over here, select the path select the path A to B, select the path A to B. Okay, so I am selecting the path over here from A to B. Now, my dear friends, listen to me very carefully. We say when it comes to the point A, first we'll write the potential of A, that is VA. First we'll write the potential of A, that is VA. Then in case of this wire, in case of this resistance over here, how do we write the potential difference? We say, sir, it is I into R. V is equal to IR. Guys, how much is I over here? 2 into 4. That is, we say, sir, 8. 2 into 4, that is 8 volts. Okay. Because 4 is the resistance, 2 is the current. So we write over here then 8 volts. Now, we are moving from higher to lower, plus to minus. So we take this as negative, minus 8 volts. Okay. Then comes next battery. In case of battery, you have got 3 volts potential difference. So we are moving from lower to higher. We take this as positive, plus 3 volts. Okay. Then come over here. In case of this resistance, how to find potential difference? That is I into R. I is 2, R is 2. 2 into 2 is 4. So we write 4 volts over here. And we are moving from positive to negative. So this will be minus 4 volts. Whenever we move from higher potential to lower potential, we take as minus 4 volts. Okay. Then come over here. In case of this, we say V is equal to I into R. I is how much? That is 2 ampere. R is how much? That is 1. 2 into 1 is 2. And we are moving from minus to plus because this is the direction of the path. So it is plus 2. It is plus 2 is equal to plus 2 volts is equal to, we say VB. Finally, we reach the point VB. Okay. Let me know in the chats if this is crystalline clear to each and everyone, guys. Let me know in the chats. Is this crystalline clear to each and everyone? So what we can say, sir, VA minus VB will come out to be 7 volts. As simple as that. VA minus VB in this case is coming out to be 7 volts over here. Okay. Okay. Guys, simple. It is not that. In order to solve this type of problem, the first thing you guys are supposed to do is select the path. If you are selecting the path from A to B, then you have to first write the potential of A. Then potential difference of this resistance. Okay. If you are moving from higher potential to lower potential, like this is the direction of the path, then you take its potential drop as negative. Here you are moving from lower to higher, it you take its potential drop as positive. That is the logic what I am saying. Okay. Now, in this particular case, you have to find the current. Find. This is another question which is a good question. Here you guys are supposed to find the current in this branch over here. You have to find the current over here. Okay. Now guys, if you have to find the current in this particular wire, over here, how will you find the current? Listen to me very carefully. Let me show you. We basically do one thing. We redraw the circuit. Let me show you how do we exactly redraw the circuit. I will tell you. So we write it something like this. You basically interchange the position. over here. That's it. I'll tell you. See. So you make it something like this. This is the resistance and this is the battery over here. This is one more resistance. This is the battery over here. Okay. And then you have got this battery over here, that is 8 volts. So this is how do we exactly redraw the circuit. See, you have got this circuit over here. I interchange the position of this resistance and this battery. Okay. It's one and the same thing. Okay. So this is 20 volts battery over here. And this is 5 ohm resistance. Okay. And this is 4 ohm resistance. And this is 10 volt battery. And this is 8 volt battery. Now, my dear friends, listen to me very carefully. What we are supposed to do in this particular case is that we assume 0 volt potential at this point. Let's assume 0 volt potential at this point. It will not create any difference. Okay. Now, if this is 0 volt, could you please tell me how much this will be? This will be minus 10 volts. Everybody knows that. Guys, we say this potential minus this potential has to be equal to this potential difference. Okay. Like we have got, this is 10 volts. If I say this is 0 volts, this has to be minus 10. Why? Because 0 minus of minus 10 will be equal to plus 10. That is the logic we use over here. Now tell me, if this is 0 volt over here, if this is 0 volt over here, we say, sir, this will be also 0 volt. Yes. How much will be potential of this point? This will be, sir, plus 8 volt. We say this is also plus 8 volt. Okay. We say this is also plus 8 volt. Why? Because the wire is going straight till over here. Now guys, tell me one thing. If this is 0 volt over here, how much is the potential difference of this point? That is 20 volt plus 20. Because this 20 minus 0 has to be equal to this 20. Yes, we are getting. Now, you have got this resistance over here. You were supposed to find the current in this resistance only. Okay, in this resistance. Now, take this resistance. Take this resistance. I'll make it over here. Okay, its resistance is 5 ohms. And we say this is plus 8 volts and this is plus 20 volts. Okay. How to find the current? We say, sir, current I is equal to V divided by R. Okay. And this is actually delta V. Delta V means higher potential minus lower potential. That is 20 minus 8 divided by 5. That is 12 divided by 5 ampere. 12 by 5 ampere is the answer in this case. Okay. So current here in this branch will be 12 by 5 ampere. Okay. Yes, yes, yes. Okay. Okay, we have got this question over here. This is from the magnetism chapter. We have got this question over here. Look at this question. We have got an infinitely long wire. We have got an infinitely long wire, wire 1 and wire 2. And we are supposed to find, let's suppose the current is flowing something like this. We are supposed to find magnetic field at point B because of these two wires, this one and this one. We are supposed to find magnetic field because of these two wires. Okay. Now see. Listen to me very carefully. Guys, we say, if we talk about the magnetic field, let's talk about the net magnetic field over here. We say net magnetic field, it will be, let's first find because of this wire. Okay. So at this point, we are supposed to find, we say one angle is 45 degrees and another angle will be, sir, 90 degrees. Why? Because another end of this wire is at infinity. So magnetic field will be mu naught I divided by 4 pi r. Then we say sine of 45 plus sine of 90. We know magnetic field because of the wire is mu naught I by 4 pi r sine of theta 1 plus theta 2. In this case, it is theta 1 is 45, theta 2 is 90. Okay. So this is simply net magnetic field will be, this is because of one wire. But how many wires we have over here? We have 2. So we say multiply it with 2. So it will be simply 2 mu naught I divided by 4 pi r. 2 mu naught I divided by 4 pi r. Then sine of 45 is 1 by root 2. Sine of 90 is 1. So this will be the answer in this particular case. Okay. Okay, you have got one more over here. Guys, see. I'll tell you. In this particular case, you have got a wire in which current I is flowing. Okay. Then you have got a square over here. And you have got one more wire. See. This structure is basically made up of 5 wires. We say, I have told you many times, if we have got a wire and the point where you are supposed to find a magnetic field that lies along the axis of the wire, like on this axis, we say magnetic field because of this wire at this point will be 0. Okay, because it lies on its axis. Similarly, magnetic field because of this wire at this point is also 0. Why? Because this point is also lying on its axis. As simple as that. So magnetic field because of first and second is 0. Now what is there? There is this square. Now we have studied before, sir, magnetic field because of the square. How much is the magnetic field because of the square? So that will be simply net magnetic field over here. That will be because of square. That is 2 root 2 mu naught I divided by pi into L. Remember this expression directly. If you have got a square, Okay, this square, it is made up of 4 wires in which current is flowing. We say magnetic field at the center will be how much? 2 root 2 mu naught I divided by pi into L. 2 root 2 mu naught I divided by pi into L. This will be the magnetic field because of the square. Now come over here. See, in this case, guys, listen to me very carefully. Magnetic field because of this wire at center will be 0. Why? Because this point is on the axis of the wire. Similarly, magnetic field at the center because of this wire also is 0. Because this point center is on the axis of this wire also. Now, my dear friends, listen to me very carefully. If I say magnetic field because of this wire, how to find the direction? Direction because of, see, place your thumb along the direction of current, place your thumb along the direction of current, then curl your fingers. Okay. So we say that will be inside. Because of this wire, magnetic field here is inside. Because of this wire, magnetic field here is inside. But because of this wire, magnetic field here will be outside. Because of this wire, magnetic field here will be outside. So we say net magnetic field will be, we say 0. Net magnetic field in this case is 0 because here the current gets divided in different directions. So that's why. Okay. Now, there is a question over here, which is from chapter EMI. What is the question? See, I'll tell you what is the question over here. Everybody listen to me very carefully. Guys, see. I have to find the flux. Find the flux through the square loop. I am supposed to find the flux through the square loop. This is the square loop, okay? I am supposed to find the flux through the square loop. Listen to me very carefully. Guys, when it comes to the flux, we say because of this wire, magnetic field through this square loop will be variable. Because as you keep on moving away from the wire, magnetic field will keep on decreasing. So we say magnetic field here is something different. Okay, magnetic field is changing. Okay. Now what we can do in this particular case is we'll take a small strip over here, which is at x distance. This small strip is at x distance. And the width of this is, let's suppose, dx. We can find the flux through this strip. Then we'll integrate and we'll find the total flux. Guys, we cannot find the flux through the entire loop directly. Why? Because magnetic field is variable. But magnetic field will be constant through this strip. So let's find the flux. d phi is equal to, how to find the magnetic flux? Flux is equal to magnetic field into area, B into A. So it will be simply B into area. Okay, area here will be dA. Area here will be dA. Guys, flux through this will be BdA. BdA. dA is the area. So we say, sir, d phi I can write over here B. What is the area of this strip? Length of the strip is L and width is dx. So L dx. Total flux will be phi is equal to integration of BL dx. Now, listen to me very carefully. Tell me one thing. Because of this wire, infinitely long wire, at we say x distance, how much is the magnetic field? Everybody knows that. Sir, it will be integration of magnetic field will be mu naught I divided by 2 pi x L dx. 2 pi x L dx. Okay. Now, guys, in this particular case, what we can do is phi is equal to. We say what we can take common, that is mu naught I into L divided by 2 pi. Then we say integration of a to a plus L 1 by x dx, because x is variable over here. Now what is this a? A means this from here to here, it is a, and L is the length of this side. So if you integrate from a to a plus L, we say you can find the flux through this entire square. Okay. So now integration of 1 by x is simply log of x. So answer in this case will be mu naught I into L divided by 2 pi. We say log of x from a to a plus L. So this will be the answer in this particular case. Then finally you can say mu naught I into L divided by 2 pi. log of, we say, A plus L divided by A. This is the final answer. Okay, this is the flux over here. Okay, this is the flux over here. One more thing, one more important question from this one, which I want to tell you in this particular case, okay, which I want to tell you in this particular case. See, here, my dear friends, here we say over here, you have got a ring, okay, you have got a ring over here. And then you have got a magnet over here. This magnet is moving away from the ring. You guys are supposed to tell me the direction of current in the ring. Remember, the coil will never want that magnet should go away from the ring. So it will create an imaginary magnet inside it. So we say this will be the North Pole, this will be the South Pole. We said this imaginary magnet will try to attract this magnet. The coil never wants that magnetic field should decrease through it, nor the magnetic field should increase through it. If magnet is coming close to the coil, in that case, coil will repel it. If magnet is going away from the coil, in that case, coil will attract it. Okay. Now, guys, we are supposed to find the direction of the current with respect to the observer. There is a logic over here. If the observer sees North Pole in the coil, okay, then we say current in the coil is anti-clockwise. And if the observer sees South Pole in the coil directly, then we say current is clockwise. Here, observer is directly able to see North Pole in the coil, so current will be anti-clockwise. As simple as that. Okay. A question on the concept of BLV. You have got a magnetic field over here. Okay. And in this magnetic field, a rod is moving with a velocity v. You have to find the EMF. See, we say EMF is equal to BLV. Whenever the rod is placed in a magnetic field, its EMF is simply BLV. Now, B is, and most importantly, all these three should be mutually perpendicular. Now, guys, listen to me very carefully. This L has got two components. One is, we say, L cos of theta, L cos 60. Another one is, we say, L sine of 60. Now, the question is, which component you guys are supposed to take? See, L cos 60 is parallel to V, so it will not create the EMF. We say L sine 60, velocity and magnetic field are perpendicular, so we say it is BVL sine of 60 over here. Okay. Now, a question from the transformers. What is this? The primary and secondary coil of a transformer are having the 50 and 1500 turns. Okay. So we say number of turns in primary coil in the transformer is 50. And number of turns in secondary coil and transformer is, we say, sir, 1500. Okay. If the magnetic flux linked with the primary coil is phi naught plus 4t. So we say flux through primary coil is given as phi naught plus 4t. Okay. You guys are supposed to find the output voltage, V output. You guys are supposed to find the output voltage. Guys, you might have studied in the transformers, there is a formula. What is that? That is output voltage is equal to number of turns in secondary coil upon number of turns in primary coil into V input, Okay. I hope you might have seen this particular formula before. Okay. Now, in this particular case, listen to me very carefully. We say, sir, V input we don't know. How to find V input? Guys, input voltage we can find, that is d phi divided by dt. According to the Faraday's law, if you differentiate flux, you will get voltage. Okay, EMF. So we say flux For phi naught, differentiation is 0 plus. For 4t, it is 4. V input is 4. Now, number of turns in secondary coil is 1500 divided by, we say 50 into, we say, sir, 4. Okay. 0, 0. Okay. 5 3s are 15. 5 zeros are 400, 120 volts. V output in this case is 120 volts. Okay. Okay. V output in this case is 160 volts. Cool. Now, okay. This is an important question which I want to tell you over here. Okay, this one. This is a question from modern physics. An electron and a photon have the same energy. An electron and a photon have the same energy. Okay. An electron and photon have the same energy. E. In the range of few electron volts. In the range of few electron volts. The ratio of the de Broglie wavelength associated with the electron and wavelength of the photon is. Okay. Okay. Cool. Now, guys, listen to me very carefully. Listen to me very carefully. We are given in this case, we say a photon. A photon. Okay. Can you tell me how to find the energy of a photon? We say energy of a photon is simply hc divided by lambda. Okay. hc divided by lambda. Okay. Cool. Now, how to find the lambda of photon over here? It will be simply, that is hc divided by E. This is the wavelength of the photon. Okay. Now, let's talk about an electron. We have got an electron also over here. How to find the de Broglie wavelength of an electron? We have studied this. We say, sir, lambda is equal to h upon under root of 2m into, we say, sir, E. 2m ke instead of kinetic energy, we write E over here. Okay. Now, guys, listen to me very carefully. If I say this is lambda of electron, if I say I have to find in this particular case, guys, I have to find the ratio of de Broglie wavelength associated with an electron and the wavelength of the photon. I have to find lambda E divided by lambda pH. What will be that? Lambda of electron in this case will be simply h upon under root of 2m into E divided by lambda of photon is how much? We say, sir, hc divided by, we say, E. hc divided by E. Okay. Now, what I can write in this particular case is h and h will cancel over here. What I can write in this particular case is this will be simply E upon we say 2m whole raised power 1 by 2 into 1 by c. If you simplify this, it will be E upon 2m, okay, whole raised power 1 by 2, whole raised power 1 by 2 will be root into 1 by c over here. Just simplify this and you will basically get it over here. Okay. Okay. So this was the question which was asked in, we say IIT JEE 2021 and NEET 2016. That's the reason I have put up this over here. Option A. Yes. Okay, there is a question from, okay, first we'll talk about this one. There is a question from semiconductors. Okay. This was the question which was actually asked in NEET 2017. It says that which one of the following diode is forward biased? Guys, I have told you so many times this is p-type semiconductor, this is n-type semiconductor. Okay, p-type and n-type semiconductor. If p-type is connected to higher potential, n-type is connected to lower potential, in that case we say diode is forward biased. Here, P type is connected to us 3 volts, N type is connected to 5 volts. So this is higher potential and this is lower potential. Means this is reverse biased. Okay. And here, this one is, we say, forward biased. Why? Because P side is connected to higher potential, 0 volts. N side is connected to -2, that is lower potential. So when it comes to this one, this is P and this is N. This is also reverse bias. This is also reverse bias. So option second is the forward bias in this case. Okay. One more question which was asked in 2004. Okay. Now see, this is P type and this is N type. So we say P side is connected to 5 -5 and N side is connected to -12. So this is forward bias. Here, this is 0 volts. This is P side and this is N side. So N side is connected to -10 P. This is also forward bias. Here, this is 0 volts because this is grounded over here. This is P side and this is N side. N side is connected to +5. P side is connected to 0. So this is reverse bias. Then we have got this one. This is P side and this is N side. P side is connected to higher potential. This is also forward biased. In this case, which one is reverse biased? We say option C. NEET 2016, this question. Okay. What does this ask in the question? What have they asked in the question? Given the two diodes, okay, the current flowing and the resistance are 1. Okay. See, guys, this is, we say, sir, higher potential. Higher potential is connected to N side. Okay, this is N side. This is N side. And this is P side. This is P side and this is N side. So we say this is reverse bias. We say this is reverse bias. Reverse bias. If the diode is in reverse bias, remember that this behaves like an open wire. Open wire means no current will flow through this part. Okay. So current will directly flow through this. Now current in this case will be I is equal to V divided by R. V is how much? V is 10 volts over here. Okay. R in this case is how much? R, this resistance, that is 2. Now don't consider this. Why? Because this is open wire, current will not flow over here. 2 plus 2 is 4. 10 by 4. Okay. That is how much? 2.5 ampere. 2.5 ampere. Okay. Okay, this question. This was the question which was asked in JEE Mains 2021. Okay. Tell me the answer of this particular question over here. Okay. If you can think of something over here. Tell me what should be the answer. Read the question very carefully and let me know the answer in this particular case. See, in Bohr's atomic model of hydrogen, let kinetic energy, potential energy, and total energy be the kinetic, potential, and total of the electron respectively. Choose the correct option. We have to choose the correct option in this particular case. We have to choose the correct option over here. Guys, see. I'll tell you. We say here, when it comes to the total energy, listen to me very carefully. I'll write over here. When it comes to the total energy, we say, sir, total energy is inversely proportional to n square. If you have seen the energy of an electron revolving in the nth orbit, how much is that? That is inversely proportional to, we say, n square over here. Okay. Now, my dear friends, kinetic energy is inversely proportional to, we say, n square. And potential energy is also, we say, inversely proportional to -1 by n square. What is n? N is the number of orbits. Okay. N is the number of orbits. For first orbit, n is 1. For second orbit, in actual reality, how much is the energy of an electron in nth orbit? -13.6 z square by n square. Okay. Kinetic energy is just positive. Now see, potential energy, see, total energy and potential energy are negative. Okay. Now, guys, listen to me very carefully. If I say, choose the correct option when an electron undergoes transition to higher level, means if n is increasing. Transition to higher level means n is moving from 1 to 2, 2 to 3, something like that. If n is increasing, we said this kind total energy will decrease, but it decreases in negative amount. It decreases in negative amount. If something decreases in negative amount, means overall that is increasing. So we say energy, total energy increases. Same is the case with potential energy. Potential energy will also increase. Okay, so potential and kinetic energy will increase, and we say potential and total energy will increase. We say kinetic energy will decrease. Option B. So as n increases, kinetic energy directly decreases. So option B is the correct option. This was the question which was asked in JEE Mains 2021. Okay, one more question which was asked in JEE Mains 2021. Okay, let's suppose we are given an electron moving with a speed v and a photon moving with a speed c have the same de Broglie wavelength. The ratio of kinetic energy of electron as to that of the photon is see. Here it says that when the speed v have de Broglie wavelength of electron in this case is given as de Broglie wavelength of photon. Okay. If I talk about for electron, it is h divided by mv. De Broglie wavelength of an electron is h by mv. If I talk about, sir, photon, it is simply hc divided by ep, energy of photon, because energy of photon is hc by lambda. Okay. Now, I want you guys to basically equate these two because they are equal. So you can write h by mv is equal to hc divided by ep, energy of photon. Now, guys, tell me one thing. As we know one thing, what is that? We have studied energy E is equal to 1 by 2 mv square. Energy of an electron, if it is moving with the speed v, its energy is 1 by 2 mv square. Okay. Now, can we do one thing? Take this 2e over here, divide by v will be equal to mv. Now, put this mv in this equation over here. So what I'll get over here, that is h divided by, instead of mv, we'll write 2e divided by v is equal to, we say hc divided by ep. Okay. Now guys, take this h and h will cancel. Take this in the numerator. I can write it something like this. Over here, I'll write it. So this will be simply, I want you guys to take this v over here. Okay. So this will be v upon 2ee. 2ee will be equals to, that is c divided by ep. So now we can write energy of electron divided by energy of photon. Energy of electron divided by energy of photon will be equal to V by 2C. That is V by 2C. Okay, this one. Just a second, guys. Hello. Hola, Gus. How are you? أنا كورن pause مماز باقي كي بتوكس ولا تلفون كرا. Guys, Wasim sir is joining. Okay, Wasim sir is joining. Most probably I'll join afterwards once again. Karab pause. Stop, huh? You see setup.
Huh? Is I'm live. Hello, Yawar. Take care. Bye bye. Okay. Guys, can you hear me now? Can you hear me? There are some electricity issues going on here because it's raining continuously from past 4-5 days, right? and it's the red alert for flood here. Okay, so quickly, can you let me know in the chats? I don't know how long the screen will work. Okay, I'm getting it charged. Yeah. So can you all hear me? Everyone? Everyone in the chats, can you all hear me? Quickly, fire in the chats. Quickly, fire in the chats, quickly. Quickly, people, fire in the chats. Everybody in the chats. Okay, nice. So let's get going. Let's get started with the same question, right? Which we were doing basically. Yeah. So look at this particular question, guys. Look at this particular question carefully. As per this question, the molar solubility of CaF2 is given in 0.1 molar solution of NaF. Okay, the molar solubility of CaF2 whose KSP is given in 0.1 molar solution of NaF will be. How do we deal with such kind of the question? Try to understand very carefully because this sort of a question is frequently asked in the NEET or even JEE examination. Trust me on that. So people, if I particularly talk about CaF2, first of all, you should know how this CaF2 undergoes dissociation. It undergoes dissociation as Ca di positive aqueous plus 2 times F negative aqueous, right? Let's assume its solubility is S. This is going to be S and this is going to be 2 times S. Why 2S? Because stoichiometric coefficient here is 2. Stoichiometric coefficient here is 2. Now, my dear students, since we are introducing the CaF2 into NaF, and NaF being the strong electrolyte, it will get completely dissociated into its ions, which is Na positive and F negative. Now, my dear students, if I ask you, what is the concentration of NaF we are keeping here? As per the question, it's 0.1 molar. So initially, this will be 0.1 molar. This is going to be 0. This is going to be 0. Now, this NaF will undergo complete dissociation. I'll be left with nothing. Now, 1 mole gives 1 mole. 0.1 gives 0.1 molar. Similarly, 1 gives 1. So 0.1 gives 0.1 molar. Right? Now, if you look carefully, I would say in the same container. What do we have? In the same container, we have got a weak electrolyte and the strong electrolyte. Both are having one ion in common. And which one is that common ion? That is F negative. Now, if I ask you, what will be the concentration, total concentration of the common ion? It is going to be 2S plus 0.1. And 2S plus 0.1, due to common ion effect, it is just going to be 0.1 molar. Correct? Now, my dear students, after this, if I want to write the expression for KSP of this CaF2, expression for KSP of CaF2, it's going to be concentration of calcium di positive raised by its stoichiometric coefficient 1, concentration of F negative raised by its stoichiometric coefficient, that's 2, right? KSP value as per the question is given to me as 5.3 into 10 raised power minus 11 is equal to calcium di positive solubility is, its concentration is S. F negative concentration, I told you it is 0.1. 0.1 means 10 raised power minus 1 whole raised power 2. Correct? So S value from here will come out to be 5.3 into 10 raised power minus 11 divided by 10 raised power minus 2. So S value will be nothing but 5.3 into 10 raised power minus 9, and this is going to be moles per liter. Now, if I ask you which option is matching here, it is 5.3 into 10 raised power minus 9. So it has to be option 4, which is the correct answer of this particular question. If it is clear, let me know in the chats quickly. If it is clear, let me know in the chats quickly, people. If it is clear, let me know in the chats quickly. Yeah? I believe it's clear to everybody. I believe it's clear to everybody. My dear students, let me tell you, this sort of a question is frequently asked. It is frequently asked. Right? It is frequently asked. Let me tell you that. It is frequently asked. Okay? I am moving ahead and let's talk about few more important questions. This question, I believe every one of you would be solving on your own. You need not to use pen and paper here, right? You need not to use pen and paper here. You can let me know in the its answer in the chats quickly. Reason for lengthenoid contraction. What is the reason for lanthanide contraction? You should know it. It is the negligible screening effect of f orbitals, right? I'm not discussing it. You must be knowing about this. You must be knowing about this. Okay? Moving ahead, people. Moving ahead. Moving ahead. Just give me a second. Just give me a second. All right. What about this sort of a question? What about this sort of a question? What about this sort of a question? In which of the following pairs are both the ions colored? Are both the ions colored in aqueous solution? My dear students, how do you check whether the ions in the aqueous solution are going to be colored or not? How do you check that? You just need to check whether there are unpaired electrons present or not. If there are unpaired electrons present, directly you can mark them as colored. Directly you can mark them as the colored. Can you let me know what do you think about this particular question? Quickly in the chats. Quickly in the chats, can you let me know what do you think about this particular question? What should be the answer of this question? Quickly. My dear students, if I select the last option, if I talk about the outermost configuration of nickel, iron, cobalt, nickel, it is iron, cobalt, nickel. It is 3d8 4s2, correct? But as per the question, it's nickel di positive. So you need to take 2 electrons out. When you take 2 electrons out, it becomes 3d8 configuration. So 3d8 configuration will be simply like this. 1, 2, 3, 4, 5, 6, 7, 8. Yes, there are 2 unpaired electrons here. If I talk about titanium tripositive, scandium titanium, 3d2 4s2. But this is tripositive, so you have to take 3 electrons out. When you take 3 electrons out, the configuration becomes 3d1. The configuration becomes 3d1. So there'll be 1 unpaired electron here. Absolutely, there'll be 1 unpaired electron here. Perfect. And wherever you see the unpaired electrons, you are going to mark that as colored. So what is going to be the correct answer of this particular question? The correct answer of this particular question has to be option D, in which both the ions will be having unpaired electrons. I hope I'm clear to everyone. Now people, this question also you need not to use pen and paper. You should be knowing it already. You should be knowing it already. Of the following transition metals, the maximum number of oxidation states are exhibited by manganese, right? Manganese shows maximum oxidation states among all. Perfect. This is again very simple and basic one. No need for the explanation of this particular stuff, right? Okay. What about the electronic configuration of copper? Electronic configuration of copper. You know, copper and chromium, they show exceptional configurations. You need to remember them both. So if I talk about chromium and copper, right? Copper is nothing but 3d10 4s1 outermost configuration. This is nothing but 3d5 4s1. These are the configurations of your chromium and copper. Anything can be asked. Anything can be asked, people. So if I talk about copper, it is 3d10 4s1 and 3d10 4s1 is here. So it has to be option B, which is the correct answer of the question. Remember these two as well. Remember these two as well, people. Remember these two as well. I am moving on to one more question. The name of the complex ion. What is the name of this particular complex ion? FeCN6 trinegative. Again a simpler one, right? Again a simpler one. Easy kill question. Easy kill question. Easy kill question. First of all, if I ask you what is the oxidation state of iron here, it is +3. It is +3, right? Oxidation state of iron here is +3. So it is going to be hexacyanido. It is first of all going to be hexacyanido. Then it's going to be ferrate, and 3 is the oxidation state. And at the end, you are going to write iron, right? So what is going to be the name? It is going to be hexacyanido ferrate 3 ion. So it has to be option D, which is going to be the correct answer of this particular question as well. Correct? Yes, there will be some of the questions of organic as well. No need to worry. Just wait. Just wait, people. Just wait. Just wait. All right. This question, I believe, you all would be knowing already. Right? You are given with the ligands. You have to arrange them on the basis of their ligand field strength. Ligand field strength. You know, Cn negative is the strongest ligand which we have among all. Right? So it has to be option B or option D. It has to be option B or option D. It has to be option B or option D. It has to be option B or option D. So what do you think is the correct answer of this question? Is it option B or option D? Quickly. Guys, I hope you remember that series in coordination. Absolutely, it's going to be option D, right? Just a memory-based question, nothing else. Just a memory-based question, nothing else. Okay. The geometry and the magnetic behavior of NiCO4. We are given with NiCO4 here, right? Nickel here is in zero oxidation state. Perfect. So what is the outermost configuration of nickel? Iron, cobalt, nickel. 3D8, 4s2. So it is 3D8, 4s2. It is 3D8, 4s2. How do I make this configuration here? Let's check it out. 1, 2, 3, 4, 5, 6, 7, and 8. Perfect. And this is 4s2 here. This is 4s2 here. Perfect. These are the p orbitals. These are your p orbitals. Now, my dear students, look at the ligand. Is it weak field or strong field ligand? It's a strong field ligand. If it's a strong field ligand, what is it going to do? What is it going to do? Quickly. What is it going to do? Is it going to cause the pairing? Absolutely, my dear students, it is going to cause the pairing of electrons. So one of the electron will go here, one of the electron will go here. So after pairing, what will be the configuration finally? After pairing, it's going to be 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. This is S containing 0 electrons and this is P containing 0 electrons, right? Perfect. Now look at the number of ligands here, 4. Perfect. Look at the number of ligands here, 4. So what is going to happen? Hybridization will take place. Which hybridization? Perfect. This is S. sp3 hybridization will take place here. sp3 hybridization. And sp3 hybridization means the geometry is going to be tetrahedral as per hybridization. The geometry is tetrahedral as per hybridization, number 1. Number 2. Is there any unpaired electron? There is no unpaired electron. Pairing has been done. If there is no unpaired electron, the species is supposed to be diamagnetic. The species is supposed to be diamagnetic. I believe this is clear to everyone. You can solve this sort of equation. You can solve this sort of equation on your own. So what is the answer of this question? It has to be tetrahedral geometry and diamagnetic in nature. This sort of equation you can easily, right, get in the upcoming examination for sure, people. For sure. For sure. Yes? Now, my dear students, look at one more. Look at one more. The question is, which is diamagnetic? Can you check it out? Which is diamagnetic? Can you check it out, people, which is diamagnetic? So you need to see where will you find 0 unpaired electrons. Where will you find 0 unpaired electrons? First of all, this is your strong field ligand. This is your strong field ligand. Strong field ligand causes pairing of electrons. So there are high chances in these two. There will be 0 unpaired electrons. There are high chances. Now I'll be checking among these two, right? See, these are weak field ligands, so no pairing is going to happen. So there will be unpaired electrons. I'm not going to check them. So check A and C. Check A and C. If I talk about C here, nickel here is in dipositive state for sure. Nickel here is in dipositive state. Iron, cobalt, nickel. 3D8, 4s2 has to be the configuration, but this is nickel dipositive, so take 2 electrons out. So this is 4s0. This is 4s0. So my dear students, I'll be making 3D8 configuration here. So 1, 2, 3, 4, 5, 6, 7, 8. This is 3D8. This is 4s0. This is 4p0. Perfect. Now, this is a strong field ligand. It's a strong field ligand. If it is a strong field ligand, what's going to happen? Pairing is going to happen, right? Pairing of electrons is going to happen. So one of the electron will shift here. When one of the electrons shifts here, so how this pairing is going to look like? 1, 2, 3, 4, 5, 6, 7, 8. This is vacant now. This is vacant, and these 3 are vacant. Perfect, right? So there are 4 Cn negatives attached, right? So 4 orbitals will participate in the hybridization. So this is 1, 2, 3, and 4. So hybridization is dsp2 here. Perfect. Hybridization is dsp2. At the same time, is there any unpaired electron? There is no unpaired electron. If there is no unpaired electron, it has to be diamagnetic for sure. It has to be diamagnetic for sure. That's something which I was supposed to check. Which of the following is diamagnetic in nature? It's option C, which is diamagnetic in nature. Right? Perfect, guys. Let's move on. Let me see if we can solve other questions as well. This is one more question which we have. NiCO4, NiCN4 dinegative, NiCl4 dinegative. The hybridization states of nickel atoms are respectively. How do you check the hybridizations? First of all, NiCO4. I think we are done with NiCO4 already. That's sp3, right? This is something which I've discussed already. That's sp3. right? This is sp3, so I need not to check it again. I need not to check it again. So this one is sp3. This one is sp3. Perfect. Sp3. So this cannot be the answer. Perfect. Now, NiCN4 dinegative. I think we are done with NiCN4 dinegative as well. NiCN4 dinegative, we are done. NiCN4 dinegative, we are done. It is dsp2. It is dsp2. We need not to check it again. It is dsp2. So first one is sp3. Second one is dsp2. sp3, dsp2. So it is option A or option B. We have to check the last one basically. We have to check the last one. We have to check the last one. It is direct, guys. Here the ligand is weak field. If it is weak field, it's not going to cause any pairing. If it is not going to cause any pairing, the hybridization involved is not going to be dsp2. It is going to be sp3. It is going to be sp3. So it has to be option C. I'm sorry. It has to be option B, which is going to be the correct answer of this particular question. Right? This sort of a question you can easily get in the examination for sure. Right, people? Okay, moving ahead. Moving ahead. Just give me a second. Moving ahead. All right. Look at this particular question. Look at this particular question, guys. Quickly. Time taken to complete 87.5% of the reaction is the time required for the reaction to undergo 87.5% completion. And T 50% is the time required for the reaction to undergo 50% completion. The relation between T 87.5 and T 50% for the first order reaction will be. Let me give you some direct expressions for first order. You need not to go for the derivations and stuff. In case of first order reactions, do remember, time taken to complete 75% of the first order reaction is always equal to 2 times the time taken to complete 50%. Time taken to complete 87.5% of the first order reaction is equal to 3 times the half-life. 3 times time taken to complete 50%. Similarly, time taken to complete 99.9% of the first order reaction is equal to 10 times the half-life. And all these results which are mentioned over here, these are valid only in case of what? In case of first order reactions. Only in case of what? In case of first order reactions. So for a first order reaction, T 87.5 is equal to X times T 50. X here comes out to be what? X here comes out to be 3. So the value of X here comes out to be 3. So it's option 1, which is going to be the correct answer of this particular question. But do remember these things, they are valid only for the first order reactions. I believe I'm clear. I believe I'm clear. I believe I'm clear. My dear students, moving on. Moving on to a few more questions. Okay. This sort of a question, I believe you would have seen many a times, being asked in different examinations, mainly in JEE examination, JEE mains, right? So what do you think about this one? What do you think about this one? You have got lithium, beryllium, boron, carbon, nitrogen, oxygen, fluorine. You have to talk about their first ionization energy. If I talk about lithium, beryllium, boron, carbon, nitrogen, oxygen, and fluorine, right? Generally, what do we observe? When we move from left to right in the periodic table, ionization energy increases. Right? This is the expected order. This is the expected order. But if I ask you, is this the actual order? Is this the actual order? This is not the actual order. How the actual order looks like? You have to interchange the positions. Boron will be here and beryllium will be here. Similarly, oxygen will be here, nitrogen will be here. And what is the reason for that? In case of nitrogen, because of half-filled configuration, 2p3 configuration. In case of nitrogen, you have got 2p3 configuration. In case of oxygen, you have got 2p4 configuration. And you know half-filled configuration are more stable than partially filled configurations. And you know if half-filled configurations are more stable than partially filled, I would say to take electron out from this configuration, we have to give more energy. That means its ionization energy will be more. Same goes for these two as well. Perfect. Here you have to take the electron out from 2s2 configuration. Here you have to take electron out from 2p1 configuration. So where energy required is more in case of beryllium. So this is the actual order. So do remember this particular question as well, right? Do remember this particular question as well. This is super important. This is super important, right? This is super important, people. All right. What do you think about this one? What do you think about this particular question? Look at the question carefully. For a metal ion, the calculated magnetic moment is 4.9 Bohr magnetrons. the metal ion has dash number of unpaired electrons? What do you think? You know, your magnetic moment is given by the result under root of n into n plus 2, right? And as per the equation, magnetic moment is 4.9. What should be the value of n here? What should be the value of n here? Quickly. What should be the value of n? I would say n value has to be 4. 4 plus 2 is 6. 6 4s are 24. Root of 24 comes out to be 4.9. So what is n? N represents the number of unpaired electrons. N represents the number of unpaired electrons. So the number of unpaired electrons in the species has to be 4. Again, one simple basic question. A direct question, I must say. A direct question, I must say. A direct question, I must say. I hope this is clear. Okay. Look at this particular question. The question is from the chapter atomic structure. You know, when we talk about atomic structure, velocity of the electron in the nth Bohr's orbit, you know it is V naught Z upon n. And if you need to compare the velocities, it is V1 divided by V2 is equal to Z1 divided by Z2. And this is going to be N2 divided by N1. This is the expression with the help of which you compare the velocities of the electrons moving in two different Bohr's orbits, right? Moving in two different Bohr's orbits, right, people? Now, as per the question, if the velocity of the electron in the first orbit of hydrogen, so hydrogen is given on one side and on another side, Li di positive is given. Let me call this as number 1. Let me call this as number 2, right? First thing, if I ask you the atomic number of hydrogen, it's 1 here. Atomic number of lithium, hydrogen, helium, lithium, that's 3, right? If the velocity of the electron in the first Bohr's orbit, of hydrogen is V, what will be the velocity? What will be the velocity V2? What will be the velocity in the third orbit? In the third orbit of Li di positive. So you have got all the parameters. Now it's pretty much simple. Put all these values in this expression. Instead of V1, I'll write V divided by V2, I'm writing as such, is equal to Z1 is 1, Z2 is given to me as 3, N2 value is given to me as 3, and N1 is given to me as 1. So 3 got cancelled. So I would say V2 is nothing but it is coming out to be V. So this is the result. V2 is equal to V. So option 2 has to be the correct answer of this particular question as well. I hope this is clear. I hope this is clear. I am moving on to one more question. Let me see if we can solve this one or not. Just a second. Okay. This sort of a question, expect again. The question has been asked in NEET 2020. But these sort of questions you can easily, right? These sort of questions you'll be easily getting in the upcoming NEET examination as well. Questions based on nodes, right? So my dear students, how do we calculate the total number of nodes? There is a result. Total number of nodes are calculated by the formula n minus 1, right? Now, how do we calculate the angular nodes? How do you calculate the angular nodes? Angular nodes are calculated with the help of L, where L is the azimuthal quantum number. You should know it. Similarly, if I talk about the radial nodes, or you call them as the spherical nodes, radial or spherical nodes are calculated by the formula n minus L minus 1. Now, everything is in front of you. You can easily kill it. So, if you talk about 3s, we have to calculate angular nodes and radial nodes. Angular nodes is equal to the value of L, and L value for your s orbitals is 0. So, angular nodes, no angular nodes. Now, if you talk about the radial nodes, radial node is n minus L minus 1. What is the value of n? N value here is 3. What is the value of L? L value here is 0, minus 1. So 3 minus 1 comes out to be 2. So it is going to be simply 0, 2, right? 0, 2 respectively are the angular and radial nodes in case of 3s orbital. Have a look on these sort of questions as well. Have an eye on these sort of questions as well. Okay. Let me move on to one more question. Similar sort of question, but you should be giving it a try. Similar sort of question, but you should be giving it a try, people. Quickly. The question is orbital having 3 angular nodes. Orbital having 3 angular nodes. Angular nodes are given by the value of L. So angular nodes are 3. That means L value is 3. Total nodes are given by the formula n minus 1. Total nodes are 3. So n is equal to 3 plus 1, 4. N is equal to 4. Now, L is equal to 3 means, L is equal to 0 means S. L is equal to 1 means P. L is equal to 2 means D. L is equal to 3 means F. So we are talking about F here. So the orbital has to be 4F. So is there any option as 4F? Yes, absolutely. It's option C. It is option C, which is going to be the correct answer. Right? My dear students, let me move ahead. Let me move ahead. Can you solve this one now? I would want answers in the chats. I would want answers in the chat. Someone is asking, why is live saved only for 20 minutes? When the stream ends, when the session ends, after that only you'll be able to see all the bio questions, physics questions as well, right? That happens basically. That happens when you take the same session from different studios. That happens. So at the end of the session, you'll be able to see all the questions. Right now, you won't be able to see all the questions. Anyways, what is the answer of this question? I would want you guys to write it in the chats. Quickly. People are coming up with option C, right? I'm not going to solve this question again. Again, you'll be using the same formulas. You know it, right? Total nodes, angular nodes, radial nodes. Perfect. So majority is coming up with option C. So I believe it has to be option C only, right? I'm not solving this one again. I'm not solving this one again. Okay. One short question will be there from oxidation state as well. Okay. One short question is going to be there from oxidation state as well. Now, the question is saying the oxidation number of the underlined atom in the following species is given. Identify the incorrect option. Have you solved this? Have you solved this? Quickly. Quickly, people. Incorrect. He's asking about the incorrect one. He's asking about the incorrect one. People are saying 4. How come 4? He's not asking about the correct one. He's asking about incorrect one. Incorrect one is second option, guys. If you talk about Cu2O, right? Cu2O. 1, just crisscross them. Make this 1 as the charge here. Make this 2 as the charge here. Right? Make this 2 as the charge here. So I must say oxidation state of copper here is +1. But it's given as -1. So this is the incorrect. It's incorrect. It is just crisscross them. That's it. That's all. Nothing else you need to do. Okay. Moving on to one more question. Do you remember this question was asked in your NEET 2019? So you need to remember few structures. Your CrO6. Your CrO6. I believe you remember its structure. Right? I believe you remember its structure. This is your CrO6. And the oxidation state of chromium here is +6. Right? Remember the structure of your CrO5 as well. CrO5 looks like this. This is your CrO5. We say it's a butterfly structure, but in reality, it's not butterfly structure. You should know it, correct? So, what is the oxidation state here? Oxidation state of chromium here. Again is +6 only. So, remember it. Remember it. Yeah? So, people, I'm moving ahead. I am moving ahead. Just give me a second. Okay. Look at this particular question. If you look at this particular question, you have to check the substances that are oxidized and reduced in the reaction. Which substances are oxidized and which substances are reduced in the reaction? How do we check it? It's again pretty much simple. The question has been asked in AIMS 2016. And this sort of a question again can come in the upcoming NEET examination for sure. See guys, if I talk about this particular oxygen, its oxidation state here is -1. It's hydrogen peroxide, right? Oxidation state of oxygen here is -2. So is the oxidation state of oxygen increasing or decreasing? -1 to -2, it's decreasing. You know, decrease in the oxidation state is what you call as reduction. So this H2O2 is undergoing reduction here. If it is undergoing reduction, that means this species is undergoing oxidation. This species is undergoing oxidation. Substances that are oxidized. N2H4 is oxidized. H2O2 is reduced. Right? So it has to be option 2, which has to be the correct answer of this particular question again. Right, people? Let me move ahead and let me try to solve a few more questions. Talk about this particular question. The question can be in the format of this. Calculate the N factor of ferrous oxalate, or the question can be calculate the equivalent mass of ferrous oxalate. Right? Right, people? You should know equivalent mass of the substance is always equal to molar mass of the substance divided by what? Divided by N factor of the substance. Molar mass divided by N factor. Molar mass divided by N factor. Now, first of all, how do I calculate N factor of this substance? Let me write it again. This FeC2O4, it gives Fe di positive plus carbon dioxide. Okay. Now, my dear students, if I ask you about the oxidation state of this iron here, it's +2. Oxidation state of this carbon, when you calculate, it is +3. Oxidation state of this iron here is +2, right? Sorry, this +3 here. It is +3, and oxidation state of this carbon here is +4. It is +4. Now, if you think carefully, if you look carefully, oxidation state of iron is increasing from +1 to +3, sorry, +2 to +3. Oxidation state of carbon is also increasing, +3 to +4, right? So this is a species in which oxidation state of more than one element, is simultaneously increasing. And whenever you see such kind of the species in which oxidation state of more than one element will be simultaneously increasing, how do you calculate its N factor? Its N factor is calculated as N1 plus N2. Whenever you see a species in which the oxidation state of more than one element simultaneously increases or decreases, simultaneously both increases or simultaneously both decreases, this is how you calculate its N factor, N1 plus N2. The point is now, what is N1 and what is N2? Let's get to know. See guys, +2 to +3. +2 to +3, what is the change in oxidation state? This is final and this is initial. Final minus initial comes out B1. So your N1 value here is 1. Similarly, +3 to +4, change, final minus initial. The value comes out B1. But that one is the change in the oxidation state for one atom of carbon. But I have 2, right? So N2 value is equal to 2. If N1 is 1, N2 is 2. So overall N factor of this particular species is 3, right? Overall N factor of your ferrous oxalate is 3. And if you got the N factor of ferrous oxalate, its N factor is going to be molar mass of ferrous oxalate divided by 3 over here. That's going to be its equivalent weight. Right, people? Is it clear? Is it clear? And people, at the same time, if you see a species in which oxidation state of one element increases, other one decreases, at that point of time, how do you calculate its N factor? It's going to be mod of N1 minus N2. Okay? Right? That can be one more question. I am moving on, people. I am moving on. Can you let me know the answer of this particular question now? See, you are given with Cu2s. Cu2s gives copper dye positive plus SO2. If I'm asking you to calculate the N factor of this particular species, can you do that? Check it out carefully, right? The oxidation state of copper here is +1. Oxidation state of sulfur here is -2. Copper here is in +2 and sulfur here is in +4. Now check it out. +1 to +2, increase in the oxidation state. -2 to +4, again increase in the oxidation state. So again, this is a species in which oxidation state of more than one element is simultaneously increasing. So how do I calculate its N factor? Its N factor is going to be same, N1 plus N2. Now it is the point to calculate the value of N1 as well as N2. Now how do I calculate N1 and N2? +1 to +2. +1 to +2. Change is 1. 1 is the change for one atom, but I have 2. So N1 value is 2, right? -2 to +4. -2 to +4. What is the change? Final minus initial. The value is 6. 6 is the change for one atom. I have only 1, right? And 6 plus 2 makes it 8 here. So I would say N factor of Cu2s over here is just going to be 8, right? This sort of equation, I believe you would have already solved. I'm moving ahead, people. I am moving ahead. Look at this sort of equation as well. My dear students, if you look at this particular reaction, your Br2 is giving BrO3 negative. And with BrO3 negative, you are getting Br negative as well. Right? Now, what am I supposed to calculate? I'm supposed to calculate the N factor of Br2. If you look carefully, the oxidation state of bromine here is 0. The oxidation state of bromine here is +5. The oxidation state of bromine here is -1. Right? Now, 0 to +5 means increase in the oxidation state. 0 to -1 means decrease in the oxidation state. So can I say oxidation state of same element is simultaneously increasing as well as decreasing? And whenever you see oxidation state of same element simultaneously increasing as well as decreasing, you say that particular species undergoes disproportionation. So I would say in this reaction, Br2 is undergoing disproportionation. And whenever you see a species undergoing disproportionation, how do you calculate its N factor? It's N1 multiplied by N2 divided by N1 plus N2. Now, it is just the matter of calculation. Get the value of N1 and N2. How do I do it? 0 to +5. 0 to +5. Change in the oxidation state. Final minus initial. 5 minus 0 is 5. Right? But that 5 is the change in the oxidation state for one atom of bromine. I have 2. So 5 2s are 10. So N1 value comes out to be 10. What about N2? 0 to -1. 0 to -1. Change. Final minus initial. The value is 1. 1 is the change for one atom, but out 2. So N2 value is 2. So you got the N1 value, you got the N2 value. So this has to be 10 multiplied by 2 divided by 10 plus 2. Divided by 10 plus 2, right? So it's just going to be 20 divided by 12. So this is 4, 5, 4, 3. So 5 by 3 is going to be the n factor of Br2 over here. Now, it is not necessary you'll be asked to calculate its n factor. They can ask you to calculate the equivalent mass of Br2. Equivalent mass of Br2 means molar mass of Br2 divided by n factor of Br2. So the final answer will be 3m divided by 5. This is the equivalent mass of Br2 in this particular reaction. This sort of equation can come, people. This sort of equation can come for sure, right? This sort of equation can come for sure. Okay. All right. Moving ahead. Moving ahead, guys. If you look at this particular equation, this sort of equation can also come. Equation based on equivalent concept. The equation based on redox titration. The equation based on redox titration. As per the equation, what volume of 0.1 molar K2Cr2O7? So you are starting with K2Cr2O7. You are making this K2Cr2O7 to react with sodium oxalate, Na2C2O4. First of all, people, you should remember their n factors in acidic medium. N factor of K2Cr2O7 in acidic medium is 6. N factor of this sodium oxalate in acidic medium is going to be 2. What happens basically? This K2Cr2O7 in acidic medium, it converts oxalate ion into carbon dioxide. It converts oxalate ion into carbon dioxide, from where we get the n factors. So I do not discuss about the n factors here. Let me directly jump into the question. Molarity of K2Cr2O7 is given 0.2. Volume of K2Cr2O7 is to be calculated. Perfect. Molarity of sodium oxalate is given, and volume of sodium oxalate is also given. That is 50 ml. Now, my dear students, if I ask you one thing, do you know how do we calculate normality? Normality is nothing but molarity multiplied by n factor. So molarity multiplied by n factor here, 6 multiplied by 0.2, that comes out to be 1.2. So normality of this solution is 1.2. What will be the normality of this solution, which I'm representing with N2? 0.3 multiplied by 2 is 0.6, right? If I just use the equivalent concept, your equivalent concept says that milli-equivalence of K2Cr2O7 reacted has to be equal to the milli-equivalence of sodium oxalate reacted, right? Their milli-equivalents are supposed to be same. Milli-equivalence of this reacted has to be equal to milli-equivalence of this reacted. Now, how do we calculate milli-equivalence? Milli-equivalence of first means normality of first multiplied by volume of first in ml. Milli-equivalence of second means normality of second, volume of second in ml. Now, what is N1? N1 value is 1.2. What is V1? That's something to be calculated. What is N2? 0.6. What is V2? That's 50 ml. So V1 comes out to be 25 ml after doing the calculation. So what is the meaning of this 25 ml? I can say V1 is 25. That means 25 ml of 0.2 molar potassium dichromate is required to oxidize 50 ml of 0.3 molar sodium oxalate in acidic medium, right? So the answer of this particular question came out to be 25 ml. So this sort of equation, again, you should have an eye on it. You should have an eye on it. You should have an eye on it. Guys, there is this sort of equation. It is already asked in NEET 2021. You can expect the similar type of question. Match the following. You can expect the similar sort of question. Expect the similar sort of question. Okay? Expect the similar sort of question here. The question is PCl5. If I talk about PCl5, what is the type of this molecule? It's AB5L0. SF6 is going to be AB6L0. BRF5, it's going to be AB5L1. BF3, it's going to be AB3L0. Since you have selected the type, you can easily talk about their geometries, etc. So AB5L0 means trigonal bipyramidal. This is trigonal bipyramidal. AB6L0 means octahedral. Octahedral. BRF5 means AB5L1. AB5L1 is square pyramidal. So this is your square pyramidal. BF3 is nothing but it's going to be trigonal planar. Now it is just you need to match them. That's all. That's all. Correct? That's all. This sort of equation can be asked. This sort of equation can be asked. Just give me a second. Just give me a second. Okay. Tell me the stability order. This is the first question. This is the second question. I need the stability order of these carbocations. What do you think? I need the stability order of these carbocations. What do you think? Stability order of these carbocations. Quickly. Quickly, guys. What do you think? Stability order of these carbocations. You know, carbocations are stabilized by electron donating groups, right? This is electron donating. This is electron donating. This is electron withdrawing, right? So its stability will be least. Stability of C will be least. Now, among A and B, which one shows more plus I? CD3 shows more plus I. If it shows more plus I, so its stability will be maximum. So B and A and followed by C, right? If you look at this one, give me the stability order of these carbocations here.